Dr. Story Landis, our NINDS Institute Director, will do her opening remarks. I just wanted to give some quick reminders. The meeting is on videocast, so please use your microphone before asking questions so that everybody can hear remotely. Also, the bathrooms are going to be at the end of this hallway on the left, where you see the sign, the green sign, store, conference store, something like that. Yes. Okay, thank you. Okay, thank you very much for coming, and here is Dr. Story Landis.
So I am delighted to kick off this conference and welcome you to the Small Blood Vessels, Big Health Problems workshop. And it's amazing to see so many people whom I know, which is great, from the work in small blood vessels in the brain, and also so many people I don't know who work in small vessels in other parts of the body. I was trying to figure out if this is a first. I don't know if there's been a conference like this sponsored by NIH that's collected people from so many different disciplines to look at one common, I don't know if you can call it an organ, but system, which is so important in maintaining normal physiological function across all these multiple organ systems. And recognizing that they are also responsible for a whole series of pathological events. Now, from my, wouldn't say narrow-minded perspective, cerebral small vessel disease is a major contributor to age-related cognitive impairment, and as a number of us, all of us are aging, it's more and more important that we understand how they contribute and what we might do about it. Just to give you a couple numbers, small asymptomatic brain infarctions, that's the kind that they see when you get an MRI, and I am never going to serve as a normal volunteer in our MRI studies in the clinical center. There's small brain infarctions that you have no evidence of clinically in 25% of the population over age 60, and that can rise to 40%. And we're only beginning to understand the cognitive impairment. So we may think of them as asymptomatic, but in fact they may not be. And I'm sure everyone here who works in a different organ system could make the same case for the importance: heart, lung, blood, kidney, eye. And so this is one of those topics which is of interest to many different NIH institutes, each with their own mission. And sometimes those are the areas which really don't get the kind of attention that we want. So a number of years ago, extramural program staff across NIH from all those institutes came together to make a trans-NIH small vessel biology and disease working group, which I think was spectacular. And it's co-chaired by Francesca Bossetti, whom just spoke, and Zarina Gallus from NHLBI. And I really want to thank them for spearheading the development of this workshop along with the other members of the working group. The main goal is to bring together experts from across different disciplines, but with a common interest in small vessel biology. And we hope that you will identify common challenges and share your lessons learned. And so one of the goals is to figure out how we would actually come up with a list of really important questions and topics to pursue and the kinds of tools that we will need to do them. And that's going to come out in the form of recommendations. Now, I know many of you have come to workshops in the past when NIH had a better budget. Usually associate a workshop that develops recommendations with RFAs and FOAs, and I would say that what NINDS at least has done over the past decade, as the budget's undoubled, is to try to think of having these workshops not simply as a way to get RFAs and FOAs, but to inform the whole community about what you as experts, leading scientists in the community, think are the most important questions and issues that need to be addressed. That's not to say there won't be an FOA. I'm just saying when you write the recommendations, don't think you're writing an RFA, please, because, well, you know why not. And I also want to really recognize the travel awardees. This is the future. These are students and postdocs, and this is the future of science in general, the next generation, and I think we really need to be attentive to including them in important meetings like this, and also to recognize the people who funded this. We were very lucky to have funds from the Microcirculatory Society for sponsoring these travel awards. And so in addition to coming to the meeting and getting to interact with the more senior scientists, everybody who is on this list will get a certificate. And as I'm thanking the Microcirculatory Society, I also want to thank the Neural Environment Cluster Program Analysts in NINDS. We don't have divisions and branches in our extramural program. We have clusters, dates from an earlier time. Ipolia Ramadan and Patrick Hussman, who actually worked really hard to make this meeting happen. So today's agenda is really full, and so I'm not going to take any more time with introductory remarks, just to say thank you all for coming. It's a really important topic, and it spans many, many institutes and many organ systems, and to the extent that you all can synthesize common issues, common challenges, that will be wonderful, but we also recognize that there will be challenges and issues that relate to specific organ systems. So turn the podium back. Yes, thank you very much, and have a lovely meeting.
Hello, everybody. I'm Serena Gallus from the Heart, Lung, and Blood Institute. We are really delighted to see the congregation in front of us of so many bright minds coming from various domains and funded by various NIH institutes. So we really are looking forward to a great meeting. I want to introduce a little bit the logistics. The speakers are going to be asked to please stay on time. The chairs don't like to be bad cops, but if we have to, we will. We want to enhance the communication with the audience, so please plan on 5 minutes at the end of your talk for questions. We also ask everybody to summarize their talk for the last 2 slides, so if you somehow fall asleep or have to get a cup of coffee or something, make sure that you see the last 2 slides. So we want to see those and don't run over time. Otherwise, I think we are all in for a treat. And I'm looking forward to chairing the session together with Dr. Jerry Lutty from Johns Hopkins. And we will kick off with the keynote lecture by Bill Aird. And he is going to help us see the endothelium.
All right, thank you. That's not a good start to be disoriented to time.
One more request is to use your mouse to indicate we are on videocast. So there are hundreds of people watching us on videocast, and the only way they can see what you are showing on the screen is by using the mouse to move around. So I think,
Where is the mouse?
Yes.
Okay,
here.
thank you.
Would you like to take it?
Yep,
here?
great.
Okay.
All right, well, thank you to the organizers for this invitation to speak today. Since this is the opening talk, I thought that rather than showing a lot of primary data, that I would take this opportunity to take some creative license and to tell a story to help set the mood and to at least begin a dialogue. And the story is about how, as scientists, we identify and choose to see objects that are worthy of scientific attention, in this case, the endothelium, and how that prism changes not only between individuals, but also over time, according to the prevailing methodologies and technologies of the day, to historical constraints, particularly subspecialization, and of course to prevailing therapeutic imperatives. So all vertebrates have a closed vascular system in which blood is maintained within the confines of blood vessels and is propelled around the body by the action of a central pump or the heart. Now, the very term system implies cohesion, interaction, actually in this case, continuity. But it's our natural tendency to break systems like this into its component parts, into bite-sized, manageable pieces that allow us to train our focus on one level or another. How and where we choose our lines of cleavage, however, very much depends upon our own individual idiosyncratic interests and biases. So the oldest classification system, which dates back to the discovery of the circulation in the 1600s and is favored by our pulmonary colleagues, separates the cardiovascular system into a pulmonary or lesser circulation and a systemic or greater circulation. Now, the importance of this schematic is that it emphasizes the role of the lung in the pulmonary circuit in revitalizing or reoxygenating blood as it makes its journey through the body. Another classification which was introduced by Bichat in the early 1800s and found a staunch advocate in August Krogh in the early 1900s, separates the system into a propulsive organ and a series of distributing, exchange, and collecting pipes, the arteries, the capillaries, and the veins, respectively. This system recognizes the importance of the capillary bed as a structurally and functionally distinct entity. Now, cardiologists are necessarily focused more narrowly and intensely on the heart and its vascular supply, whereas our blood-brain barrier colleagues often refer to the rest of the circulation simply as the peripheral circulation. Now, another way to cut the system at its joints is to start peeling back the layers of the heart and the blood vessel wall. And in doing so, we find that the endothelium is the single entity that enters into the composition of the entire system. It's the common thread, the one piece that ties everything together. And viewed from this kind of delightfully endothelial-centric perspective, the endothelium is the fundamental vascular tissue, and the heart really is nothing more than a great muscular thickening around a portion of the system of endothelial tubes. Now, how we name and classify phenomena in nature is not a matter of semantics. It really matters. It influences how we think about and understand the natural world, what questions we ask and how we go about answering those questions. So in that spirit, I would like to submit that the endothelium, and by extension the capillaries, as I will show, should be viewed as a systemically relevant organ system, one that is teeming with life, every bit as active as any other organ system, and one that has largely untapped but enormous diagnostic and therapeutic potential. In fact, I would go so far as to say that the endothelium is an organizing principle in health and disease. Now, the goal of my talk today is not so much to convince you to share this point of view, I suspect some of you already do, but rather to ask the question, why don't more people see the endothelium in this light? So with that introduction, I'd like to focus our lens squarely on the endothelium. The endothelium is a spatially distributed organ system that reaches to almost all recesses of the human body. In an average-sized human being, it weighs about 1 kilogram. It covers a total surface area between 4,000 and 7,000 square meters, and it spans over 100,000 miles of blood vessels, the vast majority of which, the capillaries, are invisible to the naked eye. The endothelium is not an inert layer of nucleated cellophane, as it was once thought to be, but is involved in many physiological functions, some of which are included on the slide. For example, control of barrier function, leukocyte trafficking, the maintenance of blood fluidity and the cessation of hemorrhage, the control of vasomotor tone, innate and acquired immunity, and of course, new blood vessel formation. Endothelial cells secrete. They contract, they swell, they buckle. Some of them scavenge, others undergo transdifferentiation, and yet others are critical for organogenesis during development. Endothelial cell phenotypes are not uniform, but rather vary in structure and function in space and time, as well as in health and disease. And this gives rise to the phenomenon that we term in the vascular biology community endothelial cell heterogeneity. For example, endothelial cells that line most blood vessels, particularly capillaries, are extremely thin, whereas those that line the high endothelial venules of the lymph nodes have a cuboidal, plump, or tall morphology. Endothelial cells that line the straight segments of arteries but not veins are oriented along the longitudinal axis of blood flow. The endothelium of arteries and veins forms a continuous monolayer with its cells held together by tight zipper-like junctions. The endothelium of capillaries may, like their arterial and venous counterparts, be continuous, or they may be permeated by tiny holes or fenestrae. Or they may contain frank gaps within the endothelium and a poorly formed underlying basal lamina, the so-called discontinuous endothelium, depending upon the needs of the underlying tissue. For example, fenestrated endothelium is characteristic of those organs that are involved in a high degree of secretion and filtration. From a functional standpoint, endothelial cells display a remarkable division of labor. For example, endothelial cells that line arterioles are primarily responsible for mediating vasomotor tone, and they do so through the regulated release of a number of vasodilators and vasoconstrictors, whose balance mediates the diameter of the blood vessel lumen and hence peripheral resistance. On the other side of the capillary bed, endothelial cells that line venules participate in the host innate immune response, promoting plasma leakage as well as leukocyte trafficking. This latter process, in turn, is mediated by the selectable site-specific upregulation of a number of adhesion molecules which create a Velcro-like surface that leads to rolling, firm adhesion, and ultimately transmigration of white blood cells. Heterogeneity is particularly prominent at the level of capillaries, where endothelial cells are adapted to the needs of the underlying tissue. From a molecular standpoint, endothelial cells from different sites of the vascular tree express different transcriptional profiles. In vivo phage display have uncovered site-specific repertoires of cell surface proteins or receptors that have been referred to as vascular zip codes. And this is a really nice example of metaphor, which has a very rich history and tradition in the field of vascular biology to describe what can't be seen with a corporeal eye, but rather with the mind's eye. So terms like cellophane, gatekeeper, vascular zip code comes to mind. And at the end of this talk, I will rely on the metaphor very heavily on the metaphor of endothelial color palette. Now, the arteries and veins are the conduits or highways of the system, serving to deliver blood to and from various tissues of the body. In keeping with the conduit function, the walls of veins, and particularly arteries, are extremely thick, composed of multiple layers of smooth muscle cells as well as elastin that allow the vessel to withstand high systolic pressures and to provide elastic recoil during diastole. If the arteries and veins are the conduits of the system, the capillaries are the business end, or the marketplace, so to speak. This is where all the exchange takes place of nutrients and gases between blood and underlying tissue. Since the primary purpose of the circulation is to maintain and promote such exchange, then viewed from this perspective, the heart, the arteries, and veins are essentially subservient to the functions of exchange carried out in the capillaries. To drive this point home even further, consider that arteries and veins require their own capillary supply, the vasa vasorum, for nourishment as well as oxygenation of their walls. Now, based on these and other considerations, August Krogh, who was one of the great physiologists at the turn of the 20th century, concluded that it will be difficult to challenge the proposition that the capillaries constitute the most important part of the whole circulatory system. Now, in keeping with Fick's law of diffusion, capillaries comprise the vast majority of the surface area of the vascular tree. Moreover, they're extraordinarily thin. They're essentially three-dimensional tubes of endothelium that are telescoped within a sheath or a sleeve of basement membrane and then wrapped to a variable degree or cradled by highly branching pericytes, which have a contractile function which provides stability and integrity to the capillary wall and which represent a potentially important niche for mesenchymal stem cells. Now, a typical capillary is just wide enough to accommodate a single red cell, which march in single file, one by one, from arteriolar to venular end, with each cell in a position of reciprocal barter with the underlying endothelium, which further enhances the diffusional capacity of the system. However, there's some interesting exceptions to the canonical arrangement of the microvascular unit. As an endothelial biologist, these are really interesting because each one of these variations entails underlying differences in endothelial cell phenotypes. So for starters, there are two vascular beds or circulations in the body in which the oxygen gradient in capillaries is reversed. That is the pulmonary circulation and the placental circulation. So in these cases, endothelial cells on the arteriolar end of the capillary are exposed to high rates of flow of relatively deoxygenated blood. Not all capillaries are perfused all the time. August Krogh won his Nobel Prize in 1920 for the discovery of a reserve of unperfused capillaries in skeletal muscle, which then become recruited when there are increased metabolic demands, as occurs, for example, with exercise or contraction. Now, Krogh believed that this phenomenon was mediated at the level of the single capillary. However, today it's more widely held that capillary perfusion is mediated or regulated primarily by arterioles. There's some interesting exceptions, including the vasa recta of the kidney, and based on some really elegant recent studies on the blood-brain barrier in capillaries of the brain. Now, in those capillaries that are perfused, flow is generally pulsatile. So, for example, in the dermal capillaries, pulse pressure has been measured between 0.5 and 12 millimeters of mercury. A really cool exception, however, are the cochlear capillaries in the inner ear, which are fed by highly convoluted and tortuous arterioles that are believed to dampen the pulse wave, converting a pulsatile flow into a more linear stream. And without this adaptation, we would be faced with having to listen to our heartbeat day in and day out, a consequence that would not only drive us crazy, but would also mask important auditory signals. Not all arteries are connected to veins by capillaries. In some cases, blood is shunted from the arterial to venous side through AV anastomoses. These are normally seen in the skin, particularly in the fingers and the toes, where they serve as heat exchangers, diverting up to 85% of the blood flow to the dermis. These are also seen in pathology where they're called AV malformations, for example, in certain hereditary states, as well as in tumors. Not all capillaries are teeny tiny hair-like tubes. In some cases, arterioles empty their blood into lakes or sinuses. So we're all very familiar with the sinusoids of the liver, the spleen, and the bone marrow. A more striking example is the placenta. In this case, in humans, a series of about 100 to 120 open-ended spiral arteries pump blood into the intervillous space, which, in a nice case of vascular mimicry, is lined not by endothelial cells, but rather by fetal trophoblasts. Maternal blood then bathes chorionic villi, which contain capillaries from the fetus, allowing for exchange to take place. These capillaries are fed by the umbilical artery and are drained by the umbilical vein. Maternal blood then returns to the system through the uterine vein. Now, normally during pregnancy, fetal trophoblasts invade the spiral arteries, remodeling it into a high-capacitance, low-resistance vessel. In preeclampsia, remodeling and invasion by trophoblasts is defective, leading to hypoxia of the placenta and the release of circulating factors then spill over into the maternal circulation and have downstream effects. Perhaps the most extreme, if not dynamic, situation is that of the male erection. So in a response to appropriate neurophysiological stimuli, blood rushes through dilated penile arteries into the corpus cavernosum, which is essentially one enormous spongy sinus. At the same time, there is compression of venous outflow, leading to complete obstruction, rapid filling, expansion, and engorgement of the sinusoidal spaces, increased pressure, and ultimately rigidity. Now, all organismal pleasures aside, consider this from the standpoint of the endothelial cell. So at the drop of a hat, with absolutely no warning, endothelial cells are called into action, jolted from their usual state of tranquility and serenity to cope with this massive infusion of blood, which causes enormous pressures, up to 300 millimeters of mercury in human, up to 600 millimeters of mercury in the bull. And because that blood can't escape, rapid stagnation as well as deoxygenation. So it's no wonder that priapism, which is essentially a compartment syndrome, is a medical emergency. Viagra works as a phosphodiesterase inhibitor by increasing cyclic GMP levels within the arteriolar smooth muscle cells that allows increased flow of blood into this sinus space. Now, not all capillaries connect arteries to veins. For example, the hepatic sinusoids are perfused not only by the hepatic artery, but also by the portal vein, which carries deoxygenated blood, and particularly in the postprandial period, is full of partially digested food. In the kidney, glomerular capillaries connect two arterioles, the afferent and efferent, which then is distributed into yet another capillary bed, the peritubular capillaries. Now, the glomerular capillary is an ultrafiltration unit. There's very little uptake of oxygen and nutrients, whereas the peritubular capillary is more of a traditional nutrient bed. Now, in some cases, the efferent arteriole dives into the depths of the inner medulla. In this kind of no man's land, endothelial cells have to balance the competing needs not only of delivering oxygen as well as nutrients to the tissue of the inner medulla, but also to function in countercurrent exchange and to adapt to and survive in extremes of environment. For example, oxygenation levels PO2 of less than 10, a hyperosmolar and a hyperkalemic environment. Now, all of these variations are important reminders that while the primary purpose of the circulation is to transport substances, they have a number of ancillary functions. Heat regulation, I've mentioned, hydraulic force generation, ultrafiltration, and finally countercurrent exchange. I'm going to switch gears now from the level of the capillary to the level of its most important constituent, the endothelial cell. So here's a recent PubMed search in which I entered the terms endothelium and endothelial cells that yielded a total of 155,000 and 140,000 articles, respectively. This amounts to an enormous expenditure of resources, not to mention sweat and toil on the part of largely basic scientists, but to some extent translational researchers. Collectively, the data provide compelling evidence that endothelial cells are involved in most, if not all, human diseases, either as a primary determinant of pathophysiology or as a victim of collateral damage. However, if you then peruse the table of contents or the index of the common or popular medical textbooks or on-call references, one finds little reference to these terms. And one of the more striking examples is Scientific American Medicine, which publishes a hard copy of about 100 pages of index with over 10,000 entries, not a single mention of endothelium or endothelial cells. I think it's fair to say that very few clinicians are attuned to the health of this cell layer. There is no syncope, diarrhea, jaundice, or bleeding equivalent for the endothelium. There's no endothelial box that we check off as we move through the review of systems. And the endothelium is not amenable to the traditional physical diagnostic maneuvers of inspection, palpation, percussion, and auscultation. While renal function is readily assayed by BUN and creatinine, liver function by bilirubin and the transaminases, hematological function by the complete blood count, the endothelium continues to defy convenient diagnostic interrogation. So this amounts to what I find and feel is the most striking example of a bench-to-bedside gap in biomedicine today. How is it that we can go from over 1 million pages of published literature to a virtual blackout at the bedside? Well, there's several considerations, 3 of which I'd like to touch on today. One relates to the hidden enigmatic nature of the endothelium. The endothelium flies under the clinical radar screen. It rarely shows its hand, at least in ways that we as clinicians are trained to detect. So while, like blood cells, circulating blood cells, the endothelium is distributed throughout the entire body, the endothelium is tightly tethered to the blood vessel wall. Therefore, it's inaccessible for diagnostic purposes. Moreover, unlike solid organs like the lung, the liver, the heart, the kidney, the endothelium is not well circumscribed. And in fact, its diffuseness and paper-like thinness makes it virtually impossible to image using conventional diagnostic techniques. While there has been some progress over recent years in developing novel diagnostic technologies, for example, measurement of circulating biomarkers, the enumeration and phenotyping of circulating endothelial cells and blood outgrowth endothelial cells, and more recently, molecular imaging, these are all investigational and have yet to have a major impact on clinical care. Well, the second consideration is our tendency to think big, that is to focus on the large vessels, and to do so in ways that compartmentalize the body into organ-specific disciplines. So when we throw out the term vascular, whether in the context of medicine, surgery, biology, or research, most of us do think about arteries and veins. That is the sites of atherosclerosis and deep venous thrombosis, respectively. And for good reason. After all, acute myocardial infarction or coronary heart disease is the leading cause of mortality in the Western world. But it's noteworthy that we are focusing on just a few, albeit critical, inches of the vascular tree, really just a quantum dot on the surface area map relative to the vast expanses of microvessels that permeate every organ. And I think this is a really important take-home message from this talk, and I think from other talks that we'll hear in the next 2 days, and that is that an understanding of the structure and function of these microvascular beds holds important clues to the understanding the underlying pathophysiology and therapy of virtually every other organic disease state that contributes to this mortality pie chart. This idea is not new. So consider, for example, this prophetic statement that was written 130 years ago. Capillaries are the true markets of exchange. Hence the paramount importance of these vessels in the maintenance of life and health. Hence also the direct practical utility of knowing their minute anatomy and physiological dignity. Every practitioner of medicine will see the important relation this branch of histology holds to pathology and therefore to therapeutics. Now, 30 years later, August Krogh lamented that it's a little strange that the capillaries have been neglected in an extraordinary degree. Today, every subspecialty diagnoses and treats its own share of microvascular disorders, or at least those diseases that have a significant microvascular component. But they're not always recognized as such. There's nothing about these disease names or the way we think about them that shouts out capillaries or endothelium. Rather than constituting an integrated system, capillaries from different organs fall under the purview of subspecialties whose frame of reference is not the vasculature per se, but rather the physiology and pathophysiology of the organ in which the capillaries happen to reside. Now, this tendency to think about large vessels is deeply rooted in history. So the Greeks in the classical age and the Hellenistic age viewed the arteries and veins as largely uncoupled systems. And in retrospect, it's easy to understand why. They couldn't see the invisible capillaries. Rather, what they saw was a series of thick-walled, deeply situated, pulsating vessels that carried red-colored blood, and a separate network of thin-walled vessels that lay both in superficial and deep tissue planes, didn't pulsate, and carried a darker color blood. So according to Galen, blood is made in the liver by food that is brought in by the portal vein and is then distributed through the various veins of the body, where it replaces tissue that is lost by invisible emanation. Some, but not all of this blood enters the right ventricle, from where it goes through the pulmonary artery to feed the lungs, or through invisible pores within the interventricular septum, where the blood mixes with air that is brought in from the pulmonary vein. This air-blood mixture, or vital spirit, is then distributed to the various organs of the body by way of the arteries, where it provides motion and vitality and heat. These are open-ended, one-way distribution systems in which blood and air simply diffuse at the level of the tissue. Blood is not seen to circulate, but rather to ebb and flow like the tide, and the heart is not a propulsive pump. It's a furnace that provides innate heat to the body. Movement of blood and air within the arteries and veins is mediated by attractive forces that are exerted by hungry tissues. Now, following Galen's death, science and medicine moved into the monasteries, where both of these disciplines kind of languished under the ruling thumb of theology for the next 1,500 years. Galen's work fit well with Christian doctrine. In fact, his greatest work on the use of body parts is one long argument from design. To study medicine was to study Galen. The goal was not to investigate or discover, but rather to collect, to collate, to translate, to study, and to teach the work of the ancients. All of this would change in 1628 when William Harvey, an English physician, published his short book with a long title, 72 pages long, what's considered now to be one of the greatest works in the annals of biomedical history. Using a series of elegant physiological studies that represented more of a methodological rather than a technological advance. For example, the crude quantitation of cardiac output and the strategic use of tourniquets, William Harvey discovered that arteries and veins are functionally, if not structurally, connected, and that blood is conserved rather than consumed. Once this was established as fact, which would take some years, just before Harvey's death, this would change the worldview of the human body and would lay the foundation for all of modern physiology and medicine. Now, like his predecessors, Harvey's could not see the invisible capillaries. He surmised they might exist, but he actually preferred the notion that blood percolated through the tissues, going from the artery to the vein. After all, he thought, how else could nutrients possibly get out from the blood vessel into the tissues? The discovery of the capillaries would wait for a technological advance, and that was the introduction of the light microscope. So in 1661, 4 years after Harvey's death, Marcello Malpighi, who was a professor of anatomy at the University of Bologna, peered down the eyepiece of his compound double convex lens looking at fresh preparations of frog lung and found that blood was not poured out into air spaces, but was always driven through tiny tubules. And then he went on to look at other tissues and other organs and found the same pattern virtually everywhere. Now, what's surprising by today's standards is that the following 200 years was characterized and in many ways paralyzed by a lively debate as to the entity of these structures. Were they simply canals that were drilled into tissue, or did they have distinct walls? And this question would not be answered until yet another technological advance, a really simple one in this case, and that was the introduction of silver nitrate staining, which is an intravital stain that paints the borders between endothelial cells. converting it from a bland, homogeneous, sort of glistening and moist membrane into a pavement of tiles with a cobblestone pattern. And this was the original paper by von Recklinghausen in 1860. This isn't an abstract. This is a paper, 8 lines long, no references, no figures. These were the good old days. Now, 5 years later, Wilhelm Hess decided to name the endothelium, to differentiate it from other types of epithelium. This was a major development because it said that the endothelium is now a thing. It is an object that is worthy of investigation in its own right. A very, very important development in the field of endothelial biology. Now, in the late 1800s, the use of intravital microscopy uncovered an important link between inflammation and permeability and leukocyte transmigration. This was the heyday of the microvessels, or the golden age of the capillary, and for good reason in retrospect, and that is, in this pre-antibiotic era, infectious disease, both the acute and chronic formed, wreaked more havoc on the population than any other disease. And of course, infectious disease targets the microvasculature. In fact, atherosclerosis was not recognized as a disease, let alone a public health concern, until well into the 1900s. But once focus was changed, attention was altered to the large vessels, there was no turning back. And it really is not uncommon to see the cardiovascular scheme depicted as it is on the left-hand side of this slide. Not only does this vastly underestimate the breadth and scope of microvessels in the body, but it dangerously approaches a galenic view of the circulation. Now, the next major advance in the field of endothelial biology was the introduction of the electron microscope. This allowed investigators to break through the resolution barrier of light microscopy, converting the cytoplasm from kind of a bland bag into a rich, compartmentalized, three-dimensional universe of structurally and functionally distinct organelles. This early work was done by George Palade, who won a Nobel Prize for his discoveries. Now, as wonderful as these discoveries were, truth be told, there was nothing that was particularly unique about the endothelial cell, except perhaps the Weibel-Palade body. In other words, endothelial identity in the full sense of the word was not to be found at an ultrastructural level. At the end of the day, no matter how good the resolution or high the power, no matter how good the fixation and the staining or quality of the cutting life, we're still looking down the lens at devitalized dead tissue, and there's only so much information that can be obtained from these types of cellular autopsy. So in this graph, I've plotted the number of publications related to endothelium and endothelial cells on the y-axis relative to the year that they were published on the x-axis. Now, you'll notice smattering of publications in the 1950s and 1960s, hardly noticeable at this scale. And these are largely related to some seminal physiological experiments, as well as basic histology, cytology, and electron microscopy. Only so many stories you can tell. Something then happened in the early 1700s that changed the landscape. And this was the first isolation and culture of endothelial cells from humans, originally from umbilical veins from discarded placentas, described by two independent groups, Eric Jaffe and Michael Gimbroni. Now investigators had a powerful means to dissect, to study highly purified populations of living cells under tightly controlled conditions. And this led to an exponential increase in the number of publications and enormous advances in our understanding of endothelial biology. However, it can be argued that these successes came at a price, that there were diminishing and are diminishing returns. The reason being that cell culture encouraged us to think about the endothelium as a kind of a monopoly of identical cells in a culture dish, rather than as an emergent consortium with properties, emergent properties whose whole is greater than the sum of the parts. And that brings me to the last consideration in explaining the bench-to-bedside gap, and that is the complexity of the endothelium. The endothelial cell is like a chameleon. It marches to the tune of the microenvironment. In fact, so tightly coupled is it to the microenvironment that when it's removed from its native tissue, it's like a fish out of water. It undergoes phenotypic drift and becomes a shadow of its former self, and as a result, we have to interpret in vitro-based assays with extreme caution. I like to think about each one of our trillions of endothelial cells as a miniature adaptive input-output device. The input arises from the extracellular environment, either the luminal side, the abluminal side, neighboring endothelial cells, and consists of any number of biochemical and biophysical signals. The output is the phenotype of the cell, so this depends upon the level of investigation. Single cells may undergo a change in calcium flux, a change in shape, protein expression, mRNA expression. Single endothelial cells may migrate, proliferate, undergo programmed cell death. Monolayers of endothelial cells, for example, in a tissue culture plate, express barrier properties and can be assayed for leukocyte transmigration. And yet other properties are apparent only at higher levels of organization, for example, at the level of the blood vessel, the organ, or the whole organism. And examples of these properties might include endothelial contribution to vasomotor tone or fibrin deposition, perhaps angiogenesis. These are properties that can't precisely be assayed for using in vitro conditions. Now, input is coupled to output by a nonlinear array of signaling pathways that typically begin at the cell surface by way of a receptor and end at the level of transcription or post-transcriptional control. Many endothelial biologists, and I include myself in this group, spend a lot of our career with our heads stuck in the sand trying to understand basic mechanisms of signal transduction. But if we step back and take a 30,000-foot view, what we'll realize is that the input varies across the vascular tree from head to toe. For example, endothelial cells in the blood-brain barrier are exposed to a marinade of astroglial and neuronal-derived factors that are essential for maintenance of barrier function. Whereas endothelial cells that line capillaries of the heart are exposed to specialized regional forces that are associated with the cardiac cycle, as well as paracrine mediators that are secreted by neighboring cardiomyocytes. Since input varies across the vascular tree from head to toe, and insofar as the endothelial cell is capable of transducing a signal, then the output has to vary from one side of the vascular tree to the next. In fact, if we could somehow color code the endothelium, that is, assign it a shade of color depending upon its precise transcriptome, its proteome, its structure and function, then at any given point in time, the endothelium would display a rich color palette. Certainly between arteries and veins and capillaries between different vascular beds, if not between neighboring endothelial cells in the same blood vessel wall. And if we were then to roll the film, insofar as that environment changes over time, these colors may blink on and off like lights on a Christmas tree. Now, what I've described so far depicts the endothelium as a blank slate, kind of marching blindly and slavishly to the tune of the microenvironment. However, as I'll show in the next couple of slides, the story is a little bit more complicated than this. So one way to think about endothelial heterogeneity and its mechanisms is to look at sort of this time-honored dichotomy in the behavioral biosciences of nurture versus nature. So you can consider the following thought experiment. Take two phenotypically distinct endothelial cells from the body, from their intact tissue. It could be from the blood-brain barrier versus the capillary in the heart, but for purposes of illustration, we'll consider two endothelial cells that are virtually side by side in the same blood vessel. Remove them from their native environment and culture them under identical conditions. If these differences in phenotype are dependent upon the microenvironment of their native tissue, then these phenotypes will drift towards one another and ultimately reach identity. An alternative is that these differences are somehow epigenetically fixed and forever retained under culture conditions. Now, it turns out that there's a lot of literature that have indirectly looked at this, and the answer, which shouldn't be surprising to anybody, lies somewhere in between. That is, while some properties drift or wash out in vitro, others are mitotically stable. And the distance between these two color codes is inversely proportional to the plasticity of the endothelium. A really interesting hypothesis, which has found some evidence in the literature, is that disease and aging is associated with divergence of these colors. In other words, less plasticity and more lock-in. Not a good thing. Now, while the nurture side of the equation is readily explained by imminently reversible, environmentally responsive signal transduction pathways, the nature side is a little bit more complicated, but presumably involves some combination of a change in the histone code and DNA methylation. Which mechanism is operative is important because as far as the environment is controlling heterogeneity, one might choose to target that environment, whereas epigenetics, one has to target the cell itself. Well, I asked at the beginning how we look at the endothelium, and I view the field as being separated into two extremes, the lumpers and the splitters. So the lumpers don't really care about the color palette. They want to know what the default phenotype of the endothelial cell is. Tell me what is the difference between an endothelial cell and a smooth muscle cell, or between an endothelial cell and a cardiomyocyte. The splitters, and I'm an avowed splitter, are much more interested in the color palette, and the more colors, the more interesting. How is the color palette developed? How is it maintained? And how does it go wrong in disease? I'm not suggesting that we form a discipline for each phenotype of the endothelial cell. On the contrary, the endothelium, in my opinion, should be viewed as one large, gigantic mosaic whose colors are not randomly distributed, but rather conform to certain patterns that are influenced and shaped by evolutionary, by developmental, and by environmental forces. And these patterns, and this is a very important point to make, these patterns pay no attention to artificially imposed boundaries between organs. They carve their own paths. So I want to finish with just two of many possible examples. I want to take you back to the example of preeclampsia. This disease starts in the placenta, but it spills out into the maternal circulation where it affects the brain, the kidney, and the liver. So what is it about, and the microvessels in these organs, what is it about the color palette that ties these various pathologies together? It turns out to be a phenotype, or color, if you will, of the endothelial cell that makes it particularly vulnerable to circulating anti-angiogenic factors. And that is those endothelial cells that form fenestrae and are dependent upon VEGF for signaling in the choroid plexus, in the sinusoids of the liver, as well as the glomeruli of the kidney. The second example is Goodpasture's disease, which is a rare autoimmune disorder that attacks the lung and the kidney, leading to pulmonary hemorrhage and renal failure. The common denominator here happens to be a component, or in this case, antigen of the basement membrane that is released by endothelial cells only in the lung and the kidney. So it's not good enough for the pulmonary folks to say, well, I'll take care of my vascular bed and you renal folks take care of your vascular bed. I think it's important as we move forward to be able to navigate much greater swaths of territory of microvessels, either independently or preferably in teams, and to be able to decode the color palette and identify novel patterns in the color palette that might be leveraged for therapeutic gain. So I'll end on that note. Thank you for your attention. Happy to take any questions.
Gary Meininger from University of Missouri. I wonder if you might comment on endothelial microparticles and their potential for clinical use, and also gender differences in the endothelium. Do you have any comments about that?
Yeah, we have two talks from now. We have a talk on gender differences, so I'll leave that to the speaker, and I wouldn't be able to answer that with any justice. In terms of microparticles, absolutely, and I should have included that in potential novel diagnostic technologies. And I think those technologies with phenotyping microparticles, with novel sort of FACS technology are advancing. So absolutely.
John Lederer, University of Maryland. Question, lumpers, splitters. Isn't there a third possibility? I would call it that, local control. Suppose each of the endothelial cells really sort of starts off the same, more or less, and is controlled locally by its environment. It would be good for, say, neurovascular coupling, that sort of thing.
Yeah, so I mentioned the two extremes, and there are obviously some middle ground, and that middle ground sometimes involves patches of color.
But isn't this conceptually different? It's not programmed, it can be adaptive. So if you have a pathologic environment, you can actually change the endothelial cell and its function under local control.
Right, but that color palette doesn't depend upon the relative roles of the environment regulating it and the epigenetics.
Ariel Gomez, Charlottesville. I really enjoy your talk. Tom Davis alerted me to one of your wonderful papers, a review paper. I can't remember the title at the moment, but in that paper you ask questions about how versus why. We found using a different methods that in the kidney vasculature, the renin cells as well as the smooth muscle cells actually are arranged in a mosaic pattern using chimeric mice as well as the confetti mice, and show this multiclonal as well as a mosaic distribution. So, given that you are interested in asking why and with the purposes of the discussion, I wonder if you care to speculate how this might be determined even at the developmental level. I mean, what would be the purpose of this they are arranged like one side by side, and they are multiclonal in that regard. I don't know the answer.
Yeah, so I'm not familiar with that particular phenotype. Maybe we could talk after.
Well, it's not published yet, but...
Okay, yeah. So we'll talk more about it, and I might be able to give some why answers, but I'm not familiar enough with it.
Okay.
Beautiful talk, Bill. Bill Chelian, Northeast Ohio Medical University. Just one question. What would you do with the lymphatic endothelium?
Oh, yeah, so I knew that question was going to come up. And I didn't have enough room in that first slide when I talked about classification, where I really should on the left-hand side, before I even got into pulmonary versus systemic circulation, have that open-ended lymphatics coming off. Absolutely critical component of the cardiovascular system, sort of the poor second cousin that gets ignored and shouldn't be. So thank you for pointing that out.
Marilyn Cipolla from Vermont. Really beautiful talk, really great opening session. I really appreciate you bringing up preeclampsia. And I want that theme to run throughout here because I think it's an incredibly important one that we're, disease that we're just, kind of recognizing in terms of the cardiovascular risk, right? I just want to point out, however, that it's not all about the placenta. And it's not, many women who have preeclampsia do not have placental ischemia. And I think the thinking on this in terms of the spiral artery modeling and the lack of it has really pigeonholed our thinking. And it has really gotten us away from really understanding the flip side, which is the maternal endothelium, which is dysfunctional. It's not just about SFLT and VEGF. There are many women, especially who have eclampsia at term, who don't have placental disease. And so as we move forward and talk about preeclampsia more throughout this workshop, I just want everybody to remember that while there are women with placental ischemia that you're talking about, there's a large proportion of women who have preeclampsia without placental disease. And if we're going to ever solve this disease, we need to start thinking about it in terms of the maternal endothelium as well as on the fetal side. So,
Thank you for pointing that out.
It's a really beautiful talk, Bill. Hong Wang from Temple University School of Medicine, Philadelphia. You touched two fundamental points about the plasticity of the endothelium in the microcirculation, brain versus the heart. So we understand that different microenvironment surrounding cells contribute. And the second point you mentioned is the epigenetic difference. Do you have any thoughts about what determines the difference of the epigenetic environment in different organs that may contribute to the plasticity of the endothelium functional structure?
Well, I think most of the epigenetic programming occurs during development. Although there's a lot of exciting recent data that implicate a role for epigenetics in physiology and pathophysiology in the adult. For example, in atherosclerosis, some great work by Peter Davies.
Tissue difference? Anything?
The tissue differences, it's very... So we know about the importance of epigenetics, and we have understanding of mechanisms underlying epigenetic differences between lineages, endothelial cells and non-endothelial cells. The extent to which epigenetics contributes to endothelial heterogeneity per se, which is your question, for example, brain versus heart, is a completely open area that's ripe for study. I don't know, I'm not familiar with any study that has actually addressed that question and define mechanisms that are responsible for that. But by definition, they exist. We know that they exist because a lot of these cells have memory in culture.
Vesna Garovic, Mayo Clinic. Actually, I want to extend a little bit more on preeclampsia and placental dysregulation because 10% of women with preeclampsia develop disease postpartum when placenta is long gone. So, just extension of the previous commentator, Dr. Cipolla, that, endothelial dysfunction as a consequence of placental dysregulation is very important, but that maternal endothelium, maternal sick endothelium, especially as we see more and more women having a first pregnancy in their later age when they may have more cardiovascular risk factors in general. We see an explosion of obesity and increase in BMI in pregnancy. So that maternal endothelium really should be focus of investigation as pivotal in causing this disease, especially in postpartum cases when placenta is long gone.
Right. So I think the important message here is it's not just about soluble endoglin and soluble FLT. It's not just about the acute syndrome, but also long-term effects in cardiovascular disease. in these
Which makes the
individuals.
study of maternal endothelium even more important.
Thank you.
Thank you, Dr. Aird, for that great talk to get us going. I'm Jerry Luddy from the Wilmer Eye Institute at Johns Hopkins, and it's my pleasure to introduce the second speaker, Shane Pierce Kotler, who will talk about the secret to growing microvascular network. It's all about forming the right connections.
While we're getting set up here, I just want to thank the organizers for putting together what looks like a really interesting and stimulating conference. And I'd also like to thank the organizers of the session for inviting me to participate as a speaker. So my name is Shane Pierce Kotler. I'm in the Biomedical Engineering Department at the University of Virginia. I'm also in the Cardiovascular Research Center. And as shown in an earlier slide, I'm a member of the Council of the Microcirculatory Society, and I'm currently serving as art treasurer. In 360. Yeah. Thank you. Okay. So I want to start out with acknowledgments. I have a number of people to acknowledge for all the work I'm going to talk about, and I'm also going to keep this at a very high-level overview. Members of my lab, one of whom is here today, Molly, my collaborator in ophthalmology at UVA, Dr. Paul Yates, my close collaborator at Tufts University, Ira Herman, at UNC Chapel Hill, Vicki Bouch, at Johns Hopkins, Phelan McGowan, Virginia Tech, John Chappell, at Yale, Jay Humphrey, and at University of Texas, Dallas, Heather Hayenga. And fortunately, as I said, a lot of these people are here today. So if I say anything that sounds interesting, you're welcome to follow up with those who are attending. So we're interested in studying the microcirculation in my lab, and we're particularly interested in growth and remodeling. We approach this specifically by using a combination of experimental and computational models to understand how microvascular growth and remodeling plays out and how it might be altered in disease. And so we use these tools to try to figure out what the levers and switches and knobs are that we might be able to turn using drugs or cell-based therapies. So just to orient you, and is it possible to get the lights down a little bit? I have a lot of pictures and movies that will show up better if there's a dark room. Is anybody standing near a light switch? Am I? Scared of pushing buttons. This looks really complicated. Okay, well, we'll go on with it. So just to orient you to the types of things we look at in the lab on a day-to-day basis, we tend to be very, oh, that's perfect, thank you very much, very tissue agnostic. We look at a lot of different tissues, and we look at the whole microvascular network structure. So this is a micrograph from the rat mesentery. You can see the scale bar down at the bottom. And what I want to impress upon you is the fact that it's a very complex structure. Of course, we have the arterioles and we have the venules shown over here at the top left. Let's see if my mouse will work. There are a few branches off the mesenteric artery and the mesenteric vein, and they come into this tissue, and of course the arterioles branch off further and form the capillaries, and these are the structures shown here in green, these sort of green tendrils, which then ultimately come and link back up to the venules. You can see here. So this is the rat mesentery. The pattern looks quite different in the mouse retina. Here we're looking at a whole retina, and you can see the central arteries and the central veins come in at the optic disc, and they have this sort of radial pattern. They kind of alternate artery, vein, like the spokes on a bike wheel, for example. And in between these, we have the capillary networks connecting. In the mouse ear, we also see somewhat of a different pattern. We see the arterioles and the venules coming in, and they branch together, but they course together for quite some time, down to many levels of branching before they radiate out into the capillary beds that ultimately connect them. And then last but not least, we have a skeletal muscle shown here. This is the mouse spina trapezius. And we see, again, a lot of branching. We have arteries and veins being paired as they enter the tissue. They come down to the level of terminal arterial, terminal venule, and then they branch into capillaries, which are invisible here at this level of magnification. So when we look at these different patterns and different types of tissues, the obvious question is, how did they form and how did they adapt during different diseases? So microvascular growth and remodeling is a complex collection of different morphogenic behaviors. The two that I'm going to focus on today are angiogenesis and arteriogenesis. So this is a lovely movie from my collaborator at North Carolina, Vicki Balch, of an embryoid body that's spontaneously undergoing angiogenesis. And you can see very nicely endothelial cells dynamically protruding into their space and connecting up with one another to form a nascent vascular network. So angiogenesis, again, it's new capillaries sprouting from parent vessels. Arteriogenesis, on the other hand, another aspect of microvascular growth and remodeling, is when arterioles and arteries enlarge. And this is work from Phelan McGowan showing that in a spina trapezius muscle, if you induce a ligation or an ischemic event, over a short period of time, you can actually get enlargement, and that's depicted here with these red arrows of some of the feeding arterioles that perfuse this tissue. So this is at an early time point immediately after ligation, and this is a few days later, and you can see the diameter of these vessels pointed out with the red arrows almost doubles. So one thing I want to impress upon you is that these remodeling events happen on a very short time scale. In the case of angiogenesis in this embryoid body model, it can be on the order of hours. And in the case of this muscle, on the order of days. Now, granted, this is in a mouse, and it's questionable as to how this translates to humans, but even if we're at the same order of magnitude, it still suggests that these processes are very dynamic and can happen quite quickly. And of course, angiogenesis, from the disease standpoint, is implicated in diabetic retinopathy, cancer and tumorigenesis, and endometriosis. And arteriogenesis is usually affiliated with the ischemic disorders, such as stroke, cardiovascular disease, and peripheral vascular disease. So just to impress upon you the cell biology, we typically think of angiogenesis being a result of endothelial proliferation and migration, and arteriogenesis being the hyperplasia and hypertrophy of smooth muscle cells that line the arterioles. Okay, so I really have just 3 points that I'd like to make. The first key point is that the structure of the network matters when it comes to forming the right connections or growth and remodeling. The second point is that composition matters. And the third point is really that the integration of these two across spatial and temporal scales matters. So point number 1, how and where vessels connect to each other really affects angiogenesis. And I mean no disrespect to our earlier speaker, but it's not all just capillary soup. And that is a notion that I find comes out in conversations with my clinical collaborators, particularly in cardiology, ironically. So this is some work that we did a while ago in collaboration with Dr. Murphy down at Tulane. And basically we showed that there is polarization or heterogeneity in expression of perivascular NG2, which is neuroglial antigen 2, which is a proteoglycan expressed by smooth muscle cells and pericytes. So this is the rat mesentery again. And what we see if we zoom in is that there's expression of NG2 on the arterioles, but it's completely absent in veins, at least in the quiescent network. What's really fascinating, though, is that when you induce a stimulus, this polarization of expression of NG2 changes very rapidly. So just after 3 days after a wounding event in the mesentery, we see upregulation of NG2 along venules, where previously venules that were not expressing NG2. And along with this upregulation, we also see an increase of incidences of capillary sprouting that you can see here. And we'll zoom in, you can see again, venules now expressing NG2 in the smooth muscle cell layer and along pericytes, and also this is commensurate with the capillary sprouting that's coming off of these venules. We think this might be important because about 90% of the time we see the capillary sprouting occurs from the venule tree as opposed to the arterial tree. So again, in terms of arteriovenous polarity, it's important to appreciate because it has implications on where angiogenesis can happen in the network. The structure of the network also matters when it comes to arteriogenesis. And so this is work done by Phelan a few years ago when he was in my lab, and he was looking at the spina trapezius muscle of two different strains of mice. On the left, you'll recognize a C57 black 6 mouse, and on the right is a Balb C mouse. Two different inbred strains, same muscle, but very different pattern of network, especially when you look at the arteries and arterioles. So you can see on the left, we get a lot of interconnecting loops in the arterial tree. And on the right, in contrast, it's much more of a dendritic structure in the Balb C. And Phelan showed that this is important because if we ligate one of the feeders that provides oxygen and nutrients to this muscle, the remodeling that happens is very different depending on the structure. So in the C57 black 6, this is where we get what we call the arteriogenesis in those loops, those interconnected loops. And in the Balb, the arteriogenesis happens at interconnections between the arterial trees, but these are occurring at the capillary level. And you can see that here, where capillary-sized vessels are recruiting smooth muscle cells that are shown in red in response to the ligation and alteration of blood flow. So here, the key point is that the location of pre-existing connections between arterioles affects where arteriogenesis can happen, either in the big vessels, if that's where the connections are, or in the capillary-sized vessels. So along with this, if you've been paying attention, you've probably noticed these blue arrows, which represent the paired venule in this particular network. And we showed recently that it matters whether or not a venule is next to a remodeling arteriole, because it affects the recruitment of monocytes and the extent to which they can come into the tissue to affect arterialization. And so this is a time course. Sorry, there was a time course. There we go. It's a time course, again, in the spina trapezius muscle where we've ligated that upstream feeder. And you can see after 6 hours, those green cells on the left are infiltrating into the lumen of the venule and adhering, we think. And then after 24 hours, they're starting to extravasate into the tissue. And by 72 hours, these cells are really collecting, they're sort of making their march over to the arteriole that's undergoing remodeling and collecting there. And so, again, the key point is whether or not that arteriole that needs to remodel is close to a venule may affect the extent to which arteriogenesis happens in that vessel and how quickly it might happen. Okay, so key point number 2, composition matters. So the heterogeneity of cell types and their interactions with one another affects growth and remodeling. And it's not just in endothelial tubes. There are other cells that interact with each other, such as pericytes and microglia in the mouse retina, for example, that you can see here depicted on the left, all in close proximity and likely transducing signals to one another to affect how angiogenesis will occur in this tissue. So that's a healthy retina. And on the right here, we're showing endothelial cells labeled with a dye in a pathological retina. This is a Kimba mouse, which is hyperglycemic and also has pathologically elevated levels of VEGF. And as a result, we see what is commensurate with diabetic retinopathy phenotypes in humans, which is vascular dropout, as indicated by these red perimeters. So we found that we can actually affect the morphology of this network or the structure of this network by injecting cells. And you can see the results of an intravitreal injection into the contralateral eye of the same mouse that has disease when we've provided human adipose-derived stem cells. And what's interesting is that we don't think these cells are becoming endothelial cells. We do think instead they're becoming perivascular support cells or pericyte-like cells, which are affecting the endothelial structure of that capillary bed and the pathological retina. I should also mention there's some very nice work recently published by my collaborator Ira Herman that showed that in fact the cytoskeleton and the mechanotransduction capabilities of the pericytes can very much affect the phenotype of the endothelial cells. So there's a lot of molecular biology underpinning the fact that pericytes and other cell types can very strongly affect endothelial cell morphogenesis. And also, as was mentioned too, in the previous, as was mentioned earlier, there's a suggestion that these pericytes can also be a source of stem cells, which is also very provocative. Oh, 1 minute. Okay, sorry. So this is a good place to stop and summarize. When you put it all together, the structure and the composition and how it affects function and remodeling, I think it's important that we broaden from our experimental tools and also include computational tools. And that's what we're beginning to do with the collaborators shown here on the left. So the idea of simulating these cells in space and time, where we can simulate individual endothelial cells and pericytes and collections of those cells, and also the growth factors and the receptors that bind growth factors in order to increase our repertoire of tools that we have for trying to synthesize and integrate across scales. So the lessons learned, structure matters, identity matters, and certainly dynamics. And moving forward, I think the tools that are going to help facilitate our learning in this area include computational modeling, but also non-invasive imaging, high resolution. Thank you.
Jerry Meininger, I have one question for you. There's a lot of emphasis on angiogenesis and arteriogenesis. How important a role is vascular regression in pruning
Critical.
in a network?
Absolutely critical, yeah.
Because it's clearly this happens in many disease states.
Yeah.
There must be a whole host of mechanisms that are just as important for that.
Absolutely. I completely agree with you. And I intentionally focus on angiogenesis and arteriogenesis, but not at the exclusion of vascular regression, intussusception, and other modes of vascular growth and remodeling. Thank you.
John Lederer, University of Maryland. I was interested in the parasites on the one of the later slides you showed, with the parasites being highly populated, I think, around the endothelial capillaries, maybe?
Correct.
How important is their contractile function in these developments?
Extremely important, and I would reference you to my collaborator sitting there in the white shirt, Ira Herman, who studied that incredibly well and has a great publication just came out in American Journal of Cell Physiology last month.
Manfred Böhm, over here. Manfred Böhm, Heart, Lung and Blood Institute. Excellent talk. I was wondering, when you show this embryonic body and the capillary network formation in there, this capillary network is forming without any blood, right? It's just formed in. And I was wondering if you can comment on a very different function of endothelium, not only to be the wall of the blood vessel, the inner lining, but also to instruct other tissue or the other cells to become what they are. Like, I think it's important for hepatocyte maturation, for liver cell, for lung cell maturation, and hematopoietic maturation is completely independent of its function to keep the blood in the vessel.
Absolutely. I think that's a wonderful point as well. It's important not to consider the vessels in isolation, but also include the tissue in which they're interacting. So the myocytes and muscle and hepatocytes and so on and so forth. Yeah, there's a lot of crosstalk going on that's affecting the phenotype of the vessels, but also the vessels are affecting the phenotype of those stromal cells in the tissue. Absolutely.
Very nice talk. When you address the connection or composition using the cell perfusion, maybe stem cells, you show the parasites. I find this very interesting. Would you imagine that the stem cell or progenitor cell perfusion would firstly contribute to the endothelial cell or arteriogenesis instead of forming the surrounding parasites, which may mostly contribute to the vascular remodeling, which may be a pathological consequence? Would that be a very serious issue to be avoided?
Yeah, I think it's absolutely important to consider the crosstalk of pericytes with endothelial cells. There's a long line of evidence, particularly in diabetic retinopathy, that implicates the pericyte endothelial connection, and perhaps, not just whether or not the pericytes are present, but how functional those pericytes are in disease and their ability to modulate angiogenesis.
Thanks very much. For the sake of time, I think we need to move on. So the final speaker of this session is Virginia Miller from Mayo Clinic, and she'll talk to us about, are there sex differences in small vessel diameter? I'm sorry, small vessel disease. Sorry.
Okay, thank you very much for the opportunity to talk to this group today. And where's Jerry? Thank you, Jerry, for setting me up with this topic. And I've asked a question, and I think you probably already know the answer. And I'm also going to sort of give you a high-level talk of some concepts you already know. And I'm not saying these things to insult you, but I'm saying them to remind you to keep them in the forefront of your mind as you consider the systems that you're investigating, because sometimes these concepts tend to get put to the side. So here are my disclosures in terms of funding, and thank you very much to the National Institutes of Health who are keeping us going. And I have 3 very simple objectives in this 15-minute talk today. First of all, to define how sex is an important biological variable. Second, to list some points of investigation of mechanisms into sex differences. And finally, to paint the big picture. And as I go through these today, I think you're going to see some repetitions and themes. Already, I'm hearing the same kinds of words and concepts that are infiltrating, and I think you'll hear that through the rest of the presentations today. So what are sex differences and what are sex biasing factors? We're talking about sex here today, not gender. Sex is what? Biology. The basic presence of an X and Y chromosome, the sex chromosomes, or an XX chromosome in females, and the reproductive organs that result from that in the hormonal environment. Gender is everything else. It has an impact on what these genetic processes might be. But for today, we're focusing on biology, that is sex. So in males, we have an X and Y chromosome. In females, we have 2 X's. And when I gave this to my group, they said, gee, Virginia, you need to bold the other X. And I said, no, I don't, because one X, to consider the dose of the X chromosome, is inactivated. So if you have genetic polymorphisms on the X chromosome, they're going to show up in the male, but maybe not so in the female. Now, we used to think that the mosaicism in the presentation or inactivation of X was random. We're now considering this that this may not be as random as once thought, and I'm not going to dwell on that today. The presence of the X and Y chromosome in every cell dictates the nature of that cell. But on top of that, and even structural organization of some of the organs prior to the development of the sex organs and chromosomes. However, on top of these chromosomes, you have the sex hormones. And let me remind you that both testosterone and estrogen are present in males and females. Aromatase, the presence in some organs, the heart and the brain, for local production and conversion of estrogen in men is becoming quite important, but also to keep in mind that the relative concentrations of these in the body varies over the lifespan, which I'm also supposed to talk today about aging. So what you have then is a sex biasing factor that varies across the lifespan. So we consider the cardiovascular system of men and women. It's a given that they shouldn't be expected to act the same, because as you can see here, a female's body is programmed, the circulatory system is programmed to be able to adapt to this very complicated situation, physiological process, which we call pregnancy, which involves adaptation of the circulatory system not only to maintain the health of the mother, but also to maintain the health of the fetus. So when we're talking about basic molecular mechanisms which contribute to health and disease, is a given that these systems may not be the same in men versus women and vary across the lifespan given changes in the hormonal environment. So we need to take an integrative approach to how we consider sex in terms of a contributor to cardiovascular disease. It's going to affect the brain and neural control of the circulatory system, the organs that contribute to the heart to circulate the blood, the kidney which help regulates it and clears the blood, and also all components of the blood vessel wall, including those elements which circulate within the blood. As we go forward today, we need to remember that every cell within this whole system has a sex, and that the hormonal variation that you're going to see affecting the expression of these genes varies across the lifespan. So how does this play out? Well, let's consider some conditions which may have sex differences, cardiovascular disease and incidence and presentation. Chronic kidney disease, vascular dementia, ischemic, non-obstructive ischemic heart disease, all have sex differences in either incidence or presentation. But we also have sex-specific conditions, which the first speaker mentioned in terms of erectile dysfunction and preeclampsia in women. The sex-specific cardiovascular conditions usually are associated with reproduction. But the question is, how do these sex-specific conditions influence the sex differences across the lifespan? If these sex-specific conditions influence the vasculature, the blood elements, the endothelium, the smooth muscle cells, and the parasites, which were mentioned in the previous talk, then how do they affect organ damage in the heart, brain, kidney by changing blood flow, altering structure and function across the lifespan? To give an example of how this might play out, let's look at the sex-specific condition preeclampsia in women. With my colleagues Vesna Garovic and Michelle Milkey looking at the Rochester Epidemiology Project, which is a medical record link system where we're able to evaluate what's documented in the medical record in the population that lives in Olmsted County, they evaluated hypertensive pregnancy disorders relative to future cardiovascular disease. So these were women who gave birth between 1976 and 1982, and we're now pulling their records to see what happened to them in the intervening years, but they're now in their mid-50s. The hypertensive pregnancy disorders were defined as preeclampsia, and these were medical codes that were pulled from the records. Those women many years later had a significant increase in hypertension, a significant increase in reported dementia and stroke, but not coronary heart disease. You might say that this is because these women were too young to have coronary heart disease, or that the codes may not have differentiated the particular types of coronary heart disease, or more importantly, there might be different etiologies between the two. And I see one of our pregnant ladies here in the front who's shaking her head. Okay, so health to you. Okay, so I want to go back. Let's see. Why can't I go back? There we go. There, okay. So related to hypertension, future hypertension, we also see two conditions which affect the brain. Well, let's look at how this might be manifested in the brain in terms of small vessel disease, and using structural MRI, magnetic resonance imaging of white matter hyperintensities, we're using this as a surrogate for small vessel disease in the brain. So this is a horizontal image through the brain. The nose is up here, the back of the head is here. Here's normal brain tissue, and you can see nice myelin sheaths and healthy cells, where if you look at this area called considered white matter hyperintensity, which is a damaged area or ischemic area, we see loss of myelin and also loss of cells, including capillary density. What happens to these women as they age? Well, we used another condition specifically to women to evaluate this in a prospective manner, and that is the other female-specific condition of menopause, of loss of hormones in midlife. And in the Cronus Early Estrogen Prevention Study, we imaged women between the ages of 42 and 58. The mean age was 52. Again, we have this horizontal look through the brain. The white matter hyperintensities here are masked so that they can be better quantified in green. The average age of the women that we investigated in this study or examined was 52. I consider this pretty young relative to aging, And what is amazing to us was that every woman we imaged within this age group had white matter hyperintensities. And as we followed them over the 4 years, these increased, such that we saw a linear progression of the white matter hyperintensities over the 4 years of the study. The other amazing thing within this study of the early estrogen prevention study was that these women were screened for their pre-existing cardiovascular risk factors, all which were within the normative range. These were women at low risk for cardiovascular disease and had no calcium in their coronary arteries. So this was pretty remarkable to us that this starts early. It's well known that mean arterial blood pressure may increase the risk for development of white matter hyperintensities. But when we evaluated the mean arterial blood pressure in these women, the remarkable thing was that we did indeed see an age-related increase that the white matter hyperintensities increased with increasing blood pressure, mean arterial blood pressure. But look where we are. These mean arterial blood pressures is not what you're going to be considered as hypertensive. So we have a relationship of blood pressure as a continuous variable with the development of white matter hyperintensities in the brain. These were adjusted for age. What does this age adjustment look like? Well, if we break it out, we see that the women who were in the middle 50s, here is their relationship to white matter hyperintensities and blood pressure for age 55. Here it is for the age 60. So indeed, there is an age interaction with blood pressure and development of white matter hyperintensities. But what seems to be remarkable is that it decreases with age over time. And so when we do statistical adjustments for some of these interactions, we may be missing a critical time point where intervening to slow progression of some of these diseases might be missed. Do these increases in white matter hyperintensities or blood pressure have any effect on end organ function? Well, if we look at the cognitive ability of these women using auditory attention and work and memory tests, we see that the only conventional cardiovascular risk factor that correlated with a deficit in these two cognitive parameters was blood pressure. Again, within a pretty normative range, indicating that the relationships between some of these peripheral systems and what's going on in the brain needs to be paid attention to, and particularly the critical time points within menopause or with individuals with hypertensive pregnancy disorders to be intervened. We heard some activity questions from Jerry Meininger regarding microvesicles, and I'd like to say that we can look beyond conventional cardiovascular risk factors for mechanisms that might contribute into some of these factors. The one that we were particularly interested in are activated platelets, because platelets have been implicated in the development of cardiovascular disease. Can we take this a step further into brain activity and brain disease as well? Microplatelets are derived from megakaryocytes in the bone marrow, which have a nucleus, and therefore they'll be responsive to hormones. Their activation and production of microvesicles will affect the capillaries and the vascular wall to increase the development of lesions and perhaps change capillary activation and permeability. This will affect end organ structure and function, and hormonal environment will influence this because of the genomic effects of the hormones on the megakaryocytes. The microvesicles are derived from activated cells, but we need to consider what might activate these platelets in the circulation. First of all, we have agonist-stimulated activation of the platelets, but also they are activated by things like systemic blood pressure, fasting blood glucose, triglycerides, and inflammatory cytokines. These are conventional cardiovascular risk factors which change with sex and over time with aging. They're also influenced by pregnancy disorders and menopause. So we have the sex-specific conditions also affecting either directly or indirectly platelet activation, thrombin generation, vascular structure and function. What actually are these microvesicles? They're cellular membranes of, which carry signature markers of the cell of origin. They also provide a surface for the generation of thrombin, and they contain bioactive molecules which, when adhered to a cell surface, can release their cargo, affecting subsequent target cell activity. We evaluated the association of white matter hyperintensity with the platelet activation in the women in our Cronus early estrogen prevention study. Again, these women were within the age range 42 to 58 when they came into the study, and after the study was finished, some of them were 62. We looked at the platelet characteristics of these women when they came into the study, that is, at baseline, and found that the expression of fibrinogen receptors correlated to the white matter hyperintensities at the time they came into the study. This reflects their potential thrombin-generating activity. We also looked 4 years later at the white matter hyperintensities and found that these correlated with the platelet-derived microvesicles, not at the time we imaged them, but 4 years before, which tells us that perhaps there is a temporal sequence, a time-dependent factor of when cells become activated in the blood, the time it takes for them to interact with the vascular wall, and actual structural changes in the end organ. So how do we wrap this up into the big pictures that we've learned over these studies? That sex does matter. We need to pay attention to it in every experiment, and I would challenge you as you go forward today to listen to the subsequent speakers, that if they actually talk about the sex of the material they're investigating. We've heard about taking an integrative approach. I think we need to take an integrative approach to causality of small vessel disease, thinking about how changes in the kidney affect the brain and the heart, and actually consider the temporal sequences of the events that we're studying, that what markers we're measuring today may not indicate the factors which are happening in that organ we're imaging, but going forward. Finally, as we go forward, I think we really need to better define what we mean by small vessel disease. We need to figure out, excuse me, that when sex matters and how it matters. And finally, is small vessel disease a continuum of large vessel disease, or is it an independent entity? And how is the feedback worked out? And finally, we need to develop better diagnostic and prognostic tests and markers to indicate when small vessel disease occurs and the subsequent changes over time. Thank you very much.
Thank you, Virginia. Any questions?
Yeah, Paul Nyquist from Johns Hopkins. I just want to congratulate you on your great work, and I would like to discuss not only the issue of white matter progression within women, but in specific brain regions. And there's this new terminology that has come up about subcortical structures and classifying them as deep white matter hyperintensities versus periventricular hyperintensities. And there's been a couple small papers and longitudinal studies that show that in female subsets there's greater expansion of the deep white matter hypervolume. We've also now seen this in our study, where we've had 800 individuals, and there's a strong association that is independent of age, suggesting that deep white matter hyperintensities at periventricular are not only independent from age, but that deep white matter hyperintensities are strongly associated with female sex.
Thank you very much for that comment. We tried to evaluate our white matter hyperintensities with brain regions in the first study we did. Our imaging technique was not on, I'm telling you what my radiologists tell me, okay? The technique that we used and the machine that we used in the initial study was not sensitive enough for us to be able to break down in the areas of the brain that we would like to see. As we're calling these women back 3 years after they came out of KEEPS, we're using a different machine that's going to allow us to better analyze the subcortical areas where where these white matters may occur. It's a very important question. We understand it. I'm glad you're finding it, and I'll have to talk to you later about exactly how we can go forward.
Great. You're working with Dr. Jack, is that correct?
Yeah.
Mm-hmm. Yeah. I think he's working on this issue aggressively now.
Yeah, I know. That's why we're we've changed some of the techniques that we're using to image the women coming forward. And actually, the pregnancy cohort the epidemiology study that I mentioned as part of our SCORE project, we're actually calling these women back 30-some years or 20-some years after they've had their pregnancies, and we're giving them a complete cardiovascular workup, as well as cognitive function and brain imaging as well. So we'll really be able to understand within these women that had a hypertensive pregnancy disorder what the subsequent cardiovascular and cognitive challenges are for these women.
Well, thanks for your work. It sounds like a very important clip.
Please keep your questions very short. I think we can only take one more. We have a panel coming up.
Okay. Zina Wexler, University of California, San Francisco. A quick question. Microvesicles, thrombin-containing microvesicles, are those exosomes or they are microvesicles?
Okay, we defined our microvesicles in terms of size, so it's anything less than 200 micrometers. And we have specific, what's hard is to separate out markers from microvesicles and exosomes because some of them still carry the same kinds of markers. We're working on that now with different types of centrifugation and gradient separation to try to find the exosomes. What we have identified in this study were those microvesicles that were defined by having an Nexin 5 positive carriers, P-selectin, endothelial markers, and platelet markers. We haven't in this study separated out the exosomes, but we're working on that when we're looking at the products in the urine that may be affected subsequent by kidney disease to really pull out the exosomes, the internal organelles of the cell compared to those of the cell surface.
Thank you.
Thank you very much. I invite the speakers, and we have two discussants, Dr. Gomez from University of Virginia and Davis from University of Arizona. And our request to them was to bring brief additional points or even challenge the speakers. And hopefully we'll have enough time to get, again, some questions from the audience for your interventions. Please come forward. Yep, you see your name somewhere there? Okay, good. So the discussants, please start us off with brief remarks. 5 minutes, not more than 5 minutes. There's a button.
Push this. Thanks.
Yes.
There you
okay.
go. You got it. Okay. Thank you. Technologically impaired, as I am. So just a very brief remark. There are lots of experts in the audience, and I think Tom, my colleague Tom Davis, and I would like hopefully to stimulate the discussion. And please participate as much as you can. And then the other thing is that we would love for the speakers and yourselves also to be more respectful. Think about it and maybe come up in a few minutes with hope this permeates some of our discussion. Now, we talk about connections today, and Dr. Pierce Cottle gave a wonderful talk about it. And it is a remarkable feat that blood vessels connect perfectly. And again, I'm going to bring something from the kidney as well as the brain. Tom will talk about that, in which the arterioles of the kidney make this perfect connection with each glomeruli. And this is incredible because there are about 1 million glomeruli, and there are no known, at least I'm surprised that there are no more microvascular abnormalities. So this is very tightly regulated. Now, the questions that I have are going to be broad, that we have, are broad on purpose to stimulate the discussion. And one is, so what are the mechanisms? We didn't talk about the mechanisms, be it molecular mechanisms or physical factors that determine this precise temporal and spatial arrangement of the vasculature in any organ, and its shape, its branching, as well as its size. And I can start, Dr. Pierce Cotler, to ask my question from before was, is it random or is it organized, and how much of each?
Okay, so I guess this is a question about the role of stochasticity, randomness, versus deterministic signals like growth factors and
I think that may be the problem. Maybe that's the problem. It's not Joan.
Right? I mean, I don't think we have a good answer. You said something that's really interesting, that we get perfect connections and a beautifully, exquisitely, perfectly formed network. And I always find myself thinking the same thing. And then I wonder, is it maybe that, or perhaps is it a very noisy system where there is stochasticity and randomness and chaos sort of bubbling up, but it's kept in check by the right feedback control systems? And I don't think I don't think we have an answer. I can just end by saying we are trying to use computational approaches where we can simulate these processes using stochastic rules, if you will, or more deterministic rules. And by doing that, we can see sort of what outcomes or predictions our models make. And by comparing that to what we observe in our experimental systems, we can try to get a grip. I think the answer is it's going to be a combination of randomness and deterministic.
Thank you. How about some remarks from the audience regarding these issues? Does anybody? Please.
Well, I can project.
Sing.
Nobody wants us to do that.
Question by asking differently.
Okay.
Oh, well, I think the big deal is to make one circulation in one glomeruli. So if the nature organized that, then the rest is just replicating, right? But not to replicate too many. So that's also one way to look. And I'm not saying I know the answer, but this is just an opposite way.
Okay, that's a good idea to go that way. Tom, somebody else has any ideas about this? How is shape determined? How is the branching pattern determined? Many of you work on this day and night. Question? Yes, please.
A question regarding the microvascular disease and the effect of estrogen or other hormones along with the glucose. So Dr. Miller's presentation was excellent from the hormonal point of view. But besides the effect of the hormone, the glucose also plays a very important role in diabetic retinopathy. So how do they, both of them, come together to accentuate the effect of the aging effect and other pathology on the white matter hyperintensity as well as other microvascular disease in the kidneys and retina?
Okay, maybe. Is this working? Okay. That one, all righty. Here we go. Here we go. Close up and personal. Thank you for that question. Clearly, there are interactions between how the sex chromosomes, the presence or absence of the sex hormones, and metabolism are going to affect the cell. The development of type 2 diabetes in women is changing with the demographics of obesity in this country. So we used to think about type 2 diabetes risk being increased at menopause. I think now we need to go back and say, how does ovarian function affect the development of type 2 diabetes over the lifespan, especially in individuals, for example, that have polycystic ovarian syndrome, who may have a metabolic syndrome but be not prone to have some type 2 diabetes activity and vice versa. So the interaction of them, I think, needs to be studied. Typically, many of the models for diabetes have been male animals. I think we need to be able to use female animals who have either are virgins or have had in pregnancy or after pregnancy, dams that have experienced multiple pregnancies, because clearly if you look at the clinical literature in terms of risks for some future disease, whether or not you've experienced a pregnancy, multiple pregnancies, or a pregnancy disorder is going to affect your propensity for disease going forward. All these interactions are incredibly important. They haven't been studied and need to be.
A short question regarding the white matter intensity to the neuronal death. Is it because of the microemboli from the platelet vesicles?
We think it's a combination. We haven't been able to sort it out. So it's an effect of how the microvesicles may affect capillary function, how it affects the larger blood vessels in terms of structuring endothelium develop arterial lesions in the larger blood vessels, which will affect blood pressure. We have to think of it in terms of an integrated system. And if there's one thing I would like for you to take away, two things. One, sex does matter. Sex is biology. And two, we have to think integration. We can't just pull out the brain independent of the rest of the circulatory system.
I would like Dr. Thomas to make his comments.
Marilyn, go ahead.
Okay, just one quick comment, Virginia. I really appreciate pregnancy being involved in all of this, and I think it's really important, but be very careful about gestational diabetes is very different than what you just talked about, which is having diabetes during pregnancy.
Okay, thank you. I have an additional perspective I'd like to add, and it's mostly regarding the cerebral microcirculation. And I'd like the whole group to think about this, and then I have a question and a gap I want to fill. Let's not forget that the cerebral endothelial cells of these cerebral microvessels have open to them a 20 square meter interface with the blood. This is an opportunity to deliver drugs. If we're going to deliver drugs, we're going to develop therapeutics that can get into the CNS. We need to stop blaming the blood-brain barrier, because one of the biggest gaps I see from bench to bedside is our thought that this barrier cannot be permeated by drugs due to the lack of success for a number of years. I think the lack of success, if you go back to 1918 when Stern and Rothland developed the term blood-brain barrier, a lot of people hung with that for a long time thinking that nothing would get in and basically walk away. Fortunately, in 2002, when NINDS had their stroke progress review group, they came up with a concept, well, they coined the term neurovascular unit to show the interaction of the various cell types. So what's the point? These specialized cells that have the cell-cell adhesion complex that obliterates that paracellular space with these tight junctions, I see it as an opportunity. I also see it as a moving target. So to get at that, my question is, for Dr. Aird mostly, actually, to be truthful with you, endothelial cells. Does the blood-brain barrier formation in the cerebral endothelial cells coincide with the early vascularization of the growing CNS? And when does the endothelial cell develop this barrier actual morphology? When does it actually function as a barrier? And what can we learn from that can help us understand the flexibility of this barrier and its moving target aspects as we try to develop therapeutics that can get across
it? Is that a test? I'm not a blood-brain barrier expert. I do. So I I do understand that the blood-brain barrier forms late during development or even postnatally, that the endothelium is not tight during development, and that maybe we can learn something about that transition. But is that wrong? Well,
We have a session on blood-brain barrier, so we could wait. Wait, go.
so please.
Go to the microphone. Microphone again.
Oh no! Please, rescue me here.
This is a group discussion.
Okay, well, no, I know Richard Hanneman paper, for example, showed that blood-brain barrier forms embryonic day 12, 13, and the pericytes invade and astrocyte came later.
What species are we talking about?
We're talking about mouse.
Mouse, okay.
Yeah. So of humans, I don't know. I mean, I don't know. But just there is some other papers also showing recently similar situations. So it's developed embryonically.
Okay, so, but yeah.
I think.
There is a convincing evidence that at least by birth in term humans, blood-brain barrier is formed without fenestrations in the most of the areas, with relatively few exceptions. So when does it happen exactly in humans? It is still being studied, but blood-brain barrier is a lot more formed at that point than many people think. And I want to bring another point, and I think Dr. Pierce already alluded a little bit, that microglial cells that enter the brain, the developing brain, fetal brain, much earlier than the blood-brain barrier is being formed. And these are the cells that help blood-brain barrier formation through multiple different mechanisms. So this is something that we as a field should also consider, as when we study mechanisms.
Yeah, maybe I want to Bill to comment a little bit more about the heterogeneity of different organ vessels in different organ. We from the first two talk, Bill and Shan, really focus on the connection of the endothelial cells with surrounding cells. And we know that the therapeutic response is different, the disease is different. So if we emphasize the connection of surrounding cells, so I would imagine the signaling or the communication, a molecular flow from the surrounding cells play a major role determining the disease difference, organ differences in the vascular disease. Should we focus more on the signaling communication from the somatic cells towards endothelial cells? Do we know anything about tissue-specific signaling on this microenvironment?
Zarina tells me there's a session on this coming up, but I would just point.
I invite everybody to actually look up the program. So try to focus your questions for this specific topic. At the end of the day, we'll have a general topic and very challenging, interesting questions such as those asked. Very challenging questions you can think and discuss over lunch, and we can come back, all of us, at the end of the day. Please focus your question to...
But I would just echo very quickly something that I pointed out, and that is that those phenotypes that are mediated by the microenvironment, for example, by cell-cell communication, are amenable to therapies directed at that interaction. Absolutely. I see David at the microphone. He may remind us that in dogfish shark, that actually the blood-brain barrier is mediated by the glial cells and not by endothelial cells.
Yeah, in sharks and hagfish, in sharks, hagfish, in the very lower vertebrates, the elasmobranchs, it's a glial barrier, as it is in the fruit fly, in fact. Since I've got the mic, I want to move away from that question to another one that sort of troubled me. Many of us are living off of mouse models, and there are lots of strains of mice, and the question that I'm thinking that's forming in my mind is, if you look at the development of the microvasculature, not just in the brain, which is where I live, but in other organs, could the differences in the structure of the microvasculature in those organs explain some of the propensities of some of these strains to have specific diseases that are much more common or much more severe than other strains? So there.
Okay, I would like to just add to that about the strains of mice. I mean, how many of you actually pay attention to what the sex of those strains of mice might be, or these fancy-dancy? Some of you, there's a few hands going up, but if you pull out a lot of papers that use genetically altered mice, they mix together males and females, and some of them, I've been on IACUC, I know what the protocols look like. It makes my hair curl without a curling iron. And you know, we need to pay attention that these might provide us important clues if the particular genotype is not expressing a phenotype in a male compared to a female. That may provide us really important information rather than just killing all the females as they come out because they're too expensive to keep.
So in general, genetic background, including sex, is very important in those things. There's a number of other people that want to ask questions.
Yes, we have a break coming up now. We're actually over time, so they're welcome to ask questions to the speakers directly or to keep them for the end of the day where we'll have more opportunities to discuss. So we care about your questions.
Okay.
Just keep them until later.
They were warming up. They were warming
yes.
up to the discussion.
So once again, speakers, please keep on time. Otherwise, we don't have questions.
10 after 10, we'll resume the session.
And get organized for the second session this morning. I'm going to go ahead and begin introducing the first speaker of the second session. It's Dr. Kleinfeld from UCSD. He's going to talk to us about the smallest stroke and insights into brain dysfunction. So, Dr. Kleinfeld.
Okay, it's a pleasure to be here. I should note that I had Story Landis as a teacher in a Woods Hole course in neurobiology 30 years ago. So if you don't like to talk, you can blame her for her influence. All right, how do I, which is forward? Okay, so I'm going to talk about studies that performed over the last 5-10 years that had to do with blood flow within cortex, and some of these studies are related to perturbing flow in individual cortical microvessels, and these I suggest give us some insight into brain dysfunction. And I'm going to talk about work from my lab and talk about work from Andy Shee's lab at Medical University of South Carolina. And as you'll see, Andy has done some beautiful work introducing high-field MRI into the detection of single vessel defects. So here is Team San Diego and Team South Carolina. You can figure out which one is blue and which one is red. And I'll take the opportunity to thank the NINDS and the NIBIB for funding us really for the last 10 years and making this all possible. Okay, so we need a target function and objective. And I think this is seen very nicely in this work that came from this pathology work that came from Harry Vinter's lab at UCLA. And what you notice in autopsy tissue are these sort of dead regions. They're about maybe 500 microns in diameter within the gray matter of patients or people who had suffered from some form of dementia. So let's keep this in mind as an objective. What is the nature of the defect that has caused this dead tissue? And let's keep this size in mind, this sort of 500 micron diameter size. So that's a target, and we want to see if we can get some insight into this from looking at the vasculature in rodent models, in mice and rats. So I just want to give you a little bit of background about what the vasculature looks like. So over the last number of years, work primarily spearheaded by Pablo Blinder and Phil Tsai, we've reconstructed every vessel, okay, within many cubic millimeter chunks of cortex. I want to emphasize that there's this modern push to reconstruct everything, whether it's neurons or vasculature, whatever is in the brain, so you're not biased, okay, by any sort of systematics and statistics. And the features I want to talk about are the surface vasculature, which we'll see in a moment forms loops, and then these penetrating vessels that bring blood from the surface down to the parenchyma, these microvessels, and then sort of the penetrating venules that bring blood back up to the surface. And I've left out the neurons here, but the bands of gold are basically the clusters. This is in the vibrissae field of the mouse, and these are sort of the so-called barrels or vibrissae columns. So what are some of these features? If you look down on the surface of a rodent, what you see is the sort of arterial network. This is the network that's branching off of the middle cerebral artery. And every vessel on the surface here has been labeled in green, but then we sort of chased this labeling with this sort of labeling in black. And the key feature is that the vasculature on the network, on the surface, forms a network. It's not a tree structure, it's a network. So critically, if you need blood to get to this point, it could come from a complete variety of different routes from a given point. So it's sort of like a roadway, like a grid system, where you could flow in traffic from all directions. So I don't want to, I'm not going to go into the details of the surface vasculature, but both the surface vasculature and the subsurface microvasculature form short-range interconnected loops. And this is work that we finished over the last couple of years. So I just want to show you the lessons. So you don't see long loops. It's not like a small-scale network where you have these large pathways like you have, between airports and transportation systems. It's just these local vascular networks. The number of edges in the networks, well, for the subsurface vasculature, it's somewhat deeper. It's maybe 8 edges. For the surface, it's maybe 5 edges on average. But the point is that these networks facilitate the rapid redistribution of blood. And when we made lesions at a single point in these networks, okay, there's no neuronal degeneration. And that's probably because the blood is observed to rearrange itself immediately in the vicinity of these defects. So what I want to focus on, and I think is potentially relevant to small vascular disease, are the penetrating vessels. And the penetrating vessels take blood from the surface, and they bring it down to the parenchyma to feed all the neurons. And there's an interesting question is, are you dominated by flow from a given penetrating vessel? Or in fact, if you were to block this penetrating vessel, could flow come in to these neighboring subsurface microvessels, so the neighboring capillaries? And keep in mind that these capillaries form a continuous network. So it's kind of like in that old movie, Fantastic Voyage. Wherever you are, you can go wherever you want within this network, okay? So, and just, if you look at this numerically, this is data from Andy Shih. You look at sort of the median flux through a penetrating arteriole, it's about a nanoliter per second. That number is not very important. If you look at the flux through the microvessels, it's about 2 orders of magnitude less, but the point in says hundreds of microvessels. So the numbers are close. And when numbers are close, really, I think in biology, calculations fall apart, and you have to do an experiment. So our idea is that we could come in, and we want to actually quantitate what's going on. So we need 2 tools. One is the use of 2-photon microscopy, Winfried-Denk's technique. This allows us to look deep into the surface. I mean, people are pushing now maybe down to about 1 millimeter deep. Here we're looking about 300 microns deep. We pick up an individual blood vessel, and then what we could do, we could scan, you take little snapshots, and we could actually see individual red blood cells against a bright background. The bright background is just fluorescein in the plasma of the blood, so it's bright, and every time a red blood cell moves, we see this dark spot moving over here to the right. So we could quantitate that, and in fact, Pat Drew, when he was a postdoc in the lab, was able to automate this process substantially. So the second thing we want to do a perturbative experiment. We want to be able to block one vessel, and this is, I think, is a very important tool in the study of small vessel disease, because you could have an animal, it could be transgenic animal with all sorts of indicators or factors going on, but then you could measure all the physiological parameters. You could go in, you could block a specific vessel, and then you could remeasure parameters in that area. So this is a technique worked out by Nozomi Nishimura and Chris Schaefer, both professors at Cornell now. And we map the surface vasculature, again, using 2-photon microscopy. Each of these red dots are where a vessel dives. Nature was very kind to us. The penetrating vessels actually have these short little stubs. So we could come in and use a linear illumination scheme and make a blockage. And then we can just check. We could just go back and check that we actually blocked a penetrating vessel. So this is X and this is Z going deep, and you can see that the vessel here is blocked. Okay, so when you put this together, what you have, we can now ask this question. We can look at the flow through a microvascular network. In this case, we're going to look at vessels that have sort of a straight segment anywhere between about 100 and 300 microns below the surface, layer 2-3 in neocortex. This is the work of Nozomi Nishimura. So she found a penetrating vessel that was going to be targeted. She measured the speed in every vessel that she could. She actually measured it twice to get the variability. Came back, blocked the vessel at this point, and asked what happened to the flow. And unlike what happens on these interconnected networks on the surface or at depth, then right close to the block, the flow goes to zero. Blood flow just stops, okay? And it doesn't really pick up again until we're out about a radius of about 250, 300 microns, or a diameter of about 400 to 600 microns, around 500 microns, like you saw in the original image from humans from Harry Vinter. Okay, so the lesson is that the penetrating arterioles are a bottleneck in the supply of blood to the surface microvasculature. Now, I just want to show you what are one of a number of neurological consequences of this blockage. So this is data from Pat Drew. This is a mouse in which the chemokine receptor in microglia is labeled with green fluorescent protein. So again, this is a surface vasculature. This is a YZ projection. This is a penetrating vessel dropping down. We make a clot on the surface. We block flow in this vessel. We come back 6 days later and do histology, and you can just see sort of the cylinder of death. One vessel led to this cylinder of death. Okay, so one of the questions is, we formed this cyst, and is there anything we can do actually to mitigate this effect? Is there maybe some silver lining to this thing? So we looked into the possibility of using blockers of excitotoxicity, such as those that were prescribed to treat Alzheimer's disease. And so there are two blockers of excitotoxicity. One is MK801, which of course is a very strong binding constant. The other is memetine, Stewart-Lipton's drug. And these bind to the NMDA receptor when it's in the open state. So the critical thing is, if we make a microinfarct, like I showed you before, we could outline it histologically. We get a certain volume. But if we block the NMDA receptor, just shortly after we do this, the size of the microinfarct is decreased. So blocking one vessel leads to cell death, but it's mitigated, in fact, by excitotoxicity blockers. And over a very large sample of data, we could look at the cumulative probabilities. And what's shown here in this black curve is sort of the many, many defects. Again, we see a sort of a cumulative radius of around 250 microns on average. And this is extremely mitigated by the application, post-occlusion mitigation of memetine. Okay, so that's great. But what about cognitive defects? I mean, is this something we have to worry about? Or is this, it's so small. Why worry about it? Well, if you teach a physics class, you worry about it. You might forget the minus sign of Faraday's law or something. So we need to have a task. And the thing is, we want a task that really highlights a specific, something we can really grasp on in the lab. So we chose a sensory motor task, and we chose to do this in the vibrissus system of rats because nowhere in cortex is a representation of one sensory input as sharply defined as it is within the whisker system. And we used a task that was invented by Hudson and Matheson. It's a beautiful paper in 1986 where they defined the multiplicity behavioral task. And the idea is this: if there's a gap and a rat can stretch its nose and touch the other side, it'll jump. And if it can touch the other side with its whisker, it'll jump. But if it can't touch the other side, it won't jump. So it's very simple. This is the work of Andy Shih. This could be automated. So the idea is that we have a normal rat, and we get a psychometric curve. So there's a gap here of about 15 centimeters, and the rodent normally jumps to that distance. And then we come in, and we make a lesion at a single penetrating vessel. This post-hoc histology, this is the damage due to that single penetrating vessel. And now what happens is the animal has a deficit. It won't jump across the gap, basically loses the ability to jump about a distance of the length of the whisker, which is about 1 centimeter. And there's one potential control now. If it can't feel its whisker, then you should be able to cut the whisker off, which is shown here. And that's the blue curve, and that just matches very nicely with the red curve, which is due to the lesion. So the lesson is that occlusion of a single penetrating vessel leads to a cognitive deficit in a sensory motor task. Now, we said that the size of the deficit is shrunk by blockers of excitotoxicity. So let's see if this actually improves this cognitive deficit. So Andy repeated this experiment now. We get this gray curve, which is the normal psychometric curve. We make a single vessel lesion. We chase it with post-hoc histology, and you can see it's a smaller microinfarct. Okay, if, in fact, you give memantine 30 to 60 minutes after you block the single vessel, when you check this psychometrically, you get the red curve now. It's very close to the normal curve. And then if you snip off the whisker as a control, you lose this sort of centimeter of sensitivity. So the lesson from this is that one vessel will cause a cognitive deficit. So this opens up a number of questions. It opens up basic sort of questions in fluid mechanics. Why is the blood not coming into the side? But it also opens up another sort of questions in biomedicine. And we want to see are these microinfarcts as seen in dementia a consequence of this kind of activity, of occluding penetrating vessels. So I just sort of leave this as an idea. This is Winter's work in pathological tissue, and these are the kind of defects that we see from making a single vessel occlusion. And it looks very close. But now the question is, can we get, this is histology, can we get actually a better handle of this in real time? So work done in Andy's lab with Joe Halpern, okay, has started to use MRI, high-field T2 MRI. So what I want to note is that this is a microinfarct that was only published in the last year that came out of Gert Bezos' lab. You see these defects, these gray matter defects. And what you see, okay, in the rodent model, again, is the same kind of defect. So the thought is that you can start to get some insights into 7 Tesla MRI on humans from these rodent models. And I just want to note that the advantage, one critical advantage of the rodent model, okay, is that you can see these defects that were formed. This was that I showed before in terms of optical imaging. You can see these very nice in T2 images, in FLAIR images, in effectively diffusion tensor images, and then you could chase them at the end with post-hoc histology and verify what you see. You can make a comparison between MRI and the rodent models. So I'm just going to, skip ahead. So the lesson is that blockage of a single penetrating vessel leads to death of a cylinder of cortex. Another thing that happens that I didn't show is that individual occlusions can coalesce to form a large cyst. You block 2 vessels, you get a small region. You block a 3rd vessel, and the regions in between all of a sudden could coalesce, and it's interesting that you could form a cyst. And what I would say are gaps in the field is one, if these sort of large neurological insults actually result from losing a single vessel, and finally you get some critical concentration, okay, and the intervening tissue just dies for excitotoxicity, for lack of blood flow. It's not clear. And I think one of the things that's really missing is all these are gray matter. So I think we need to try to push into the white matter. I realize there's not a very thick white matter layer in rodents. The callosum is not that thick. But I think with imaging is pushing itself into the white matter. And I think the kind of optical techniques we use can start to make these white matter strokes. So thank you.
Take a couple questions in a shorter time period.
That was a terrific talk. Thank you. I'm wondering how you understand the memantine effect, especially in the whole rodent model. Once that initial 30 to 60 minutes pass, are you proposing that there is then some rerouting to the penetrating arterioles?
No, we looked. What appears to happen is you're just preserving the microvessels. Okay.
Speak in the microphone.
Oh, sorry.
Because nobody can hear.
So I have to answer this in 2 ways. The reason that that you don't get very effective flow from the microvessels on the side is because the bulk of the resistance in the vasculature is in this microvessels, is in the capillary bed. And the feeder arterioles and the return venules are relatively low resistance. So the return veins are actually sort of, in a very literal way, are sopping up the blood and keeping flow coming in from the side. Okay, but it's not, this is not a crystalline lattice, so some blood will get in, and for whatever reason, okay, the tissue you can just watch over time, just the vessels start to lose blood and the vessels start to disintegrate. So somehow, in a way I don't know, okay, just adding excitotoxicity blockers is just slowing the deterioration of the vessels to the point where, the microvessels will live long enough and they'll supply the corollary blood. Yeah.
Randy Marshall from Columbia. I'm a non-basic scientist, and I, at the risk of making a non-scientific point, I think one of the reasons for being here is to make sure we're talking the same language. So I want to point out two things that we might want to talk about how to use taxonomy and semantics. First, a cognitive deficit. What you described there, I think, is very analogous to a small vessel stroke that's usually an embolic stroke in a human. A small cortical infarct would produce a sensory motor, but not a cognitive deficit, as we think of cognition in memory and other higher level cognitive functions. The second is small vessel disease itself. When we talk about it, it's usually deep white matter penetrators from larger vessels in the basal ganglia, the deep white matter, and the brain stem. So just from a terminology standpoint, I think it would be useful for us to recognize there are differences in the way those terms are being used.
Okay, I just want to defend myself over cognitive, which you mentioned cognitive and you get in trouble. I mean, the output from primary sensory cortex in the rodents, projects down to premotor nuclei that causes the animal to move its whiskers, and it'll cause the animal to jump. So there's a decision that's being made, in this area. I mean, the nomenclature is primary sensory cortex, but the sort of the field says that that's where the decision is being made. So I think we did this because there's sort of no cognitive task the way you could ask somebody to recognize a clock or objects in a field that we could actually manage to do on rodents.
Do we have time for just one more question, please?
Hi, I'm Hans Ackerman from the NIH. Both sickle cell disease and cerebral malaria cause a tremendous amount of neurovascular disease. Yeah. But what's unique about both of those diseases is that the primary occlusion may actually be happening on the venular side. And so I wanted to ask if maybe by accident or by intention you did any of these single vessel experiments where you blocked the venule and saw the consequences.
Okay, that's a great question because I could answer it. What we did, what Andy did, and actually what Nozomi Nishimura did separately was to block veins, and you do get a deficit in flow. And what's interesting is the same model I talked about where veins will prevent blood from getting in, gives you a sort of semi-quantitative understanding of both this venule deficit as well as the arterial deficit. So we did that, and in fact you do get a deficit in flow, right? So it works, yeah.
Start your seat.
I'm happy to introduce Dr. Gary Gibbons, who's the director of NHLBI, who's one of the sponsors for the meeting.
Thank you very much. I'll be brief. I don't want to stop the flow of the science. As a cardiologist and a vascular biologist, this is something that's very exciting for me. I was hoping I could hang out even longer to hear some of the science. It's good to see some old familiar faces in the crowd as part of this meeting. We're excited to be part of this collaborative effort at NHLBI alongside our colleagues at NINDS and the NEI, as well as the Office of Prevention and Women's Health. And we've come together because we recognize that Congress, in its wisdom, did not always follow nature in recognizing the interdependency of systems. And so hopefully this isn't publicly broadcast anywhere. Don't want to affect our appropriation, but what you are exploring is that interface between our various institutes as we come together and recognize the incredible systems that are aligned, whether it's the nervous system or the vascular system, and their interplay. And we're excited about this momentum that's in this room because I think it has very critical effects on our mission particularly in heart, lung, blood, and sleep disorders, where we see a great opportunity, particularly with the momentum of the BRAIN initiative, in which NINDS is going to, along with other colleagues, map out the brain. And I must admit, I get a little bit of organ envy because indeed I hope that as you map out that great circuitry of all those trillion connections, that you don't forget about the blood vessels that are intimately integrated in making that system work. And that's what I love about this conference, is showing that intimate relationship that we see every time you guys do a functional MRI image of how these two systems engage each other. Similarly, I'm also intrigued even by that last presentation of the public health significance of this as we battle the challenges of chronic diseases like dementia and Alzheimer's, and intrigued by the notion that those who carry that clinical diagnosis of Alzheimer's often are riddled with microvascular disease and microinfarcts. An appreciation that perhaps some of our targeting of how the blood vessels are may have an impact in cognitive decline. And hopefully some of those things will bubble up as part of this interaction. Similarly, we're intrigued and hopeful about actually the last question because we think about our whole portfolio. And one of the things that is a high strategic priority for us relates to sickle cell disease. And as a cardiologist and vascular biologist, certainly coming from Atlanta, I became intimately familiar with children who, because of that blood disorder, lose 5 to 10 cognitive IQ points because of that interaction between brain development and having this disease, and have, as you know, characteristic vasculopathy that affects their brain, such that in 25 to 30% of these children, you can see the scars of a silent ischemic infarct. And I've put forward as a challenge for our institute that we think about bringing all the science together from basic translational and clinical and population research, and even as part of a trans-NIH collaborative, to think about what we can do to create that stroke-free generation. And I've sensed that a lot of that progress may relate to what you'll be discussing today, in how small vessels in the neurovascular unit work together as complex systems that interact, and finding out those pathways in a way that can have an impact of turning discovery science into enhancements in human health. And that's our mission. So we're very excited. I wish I'll try to steal a few moments and listen in to some great science. And we appreciate your collective efforts to attack this important area. Thank you for being here.
All right, we thank Dr. Gibbons for his input. Our next speaker will be Dr. Chilian, who's going to tell us about the coronary microcirculation. Dr. Chilian?
Thank you. It's a pleasure being here, and I have to extend my thanks to the NIH people who put this together and who invited me. So I'm going to talk about the microcirculation of the heart. I'm deviating a little bit from the brain, but just think of the heart as a cerebral support system, and then we'll all be in the same pathway. So this is a model of the microcirculation and the circulation of the heart, in which various control schemes are outlined with a flow-sensitive element in a series coupled to a myogenic element, in a series coupled to a metabolic element, and the aggregate is a network response. Now, I do not have time to discuss all that. What I will focus on is the metabolic element, because I have time to really go over that. Let's see. Okay, this is the relationship that I'm going to discuss. This is the relationship between oxygen consumption and blood flow. And in just about every organ system that relies on aerobic metabolism and has active function, in the brain, the function, the work that it's doing is chemical work, and the heart's mechanical work, this relationship is imperative for our survival. It allows us to think, it allows us to absorb and digest foodstuffs in the gut, it allows us to function, the heart to respond to various challenges. And yet, here we are in the 21st century. We've known the human genome now for almost 15 years, and we still don't know the exquisite links in this relationship. And this, I believe, is paramount to our understanding of a variety of pathologies. Let me first describe the classic scheme that we think of for the control of organ blood flow in response to metabolism. And this is a scheme that we teach medical students. It's in most medical textbooks. And if we just think of it starting out with a relationship between tissue PO2, which most would consider a controlled variable, and myocardial oxygen consumption, if we start out at a point of X and we increase oxygen consumption to Y, PO2 goes down. We haven't yet increased flow. So we're consuming more oxygen, flow is the same, PO2 in the tissue drops. Then we would proceed to this. So as tissue PO2 would drop from X to Y, production of some vasodilator would go up, okay? Then we would have this scheme where the vasodilator factor, as it relates to coronary blood flow, we go from X to Y, more of this vasodilator being produced, higher coronary flow. Coronary blood flow, tissue PO2, we go from X to Y, and the end result is this. So in the star, X and Y are overlapping. Now, as I tell my students, what's wrong with this picture? We have lost our error signal, but flow remains high. Now, we can argue that we could have a cyclical event, similar to the way the thermostat controls the temperature in a room, but the problem with that is that means that the heart would be living on the brink of ischemia. And we know in the normal person, the ability to increase coronary blood flow is 4 to 5 to 6-fold. So the heart does not live on the brink of ischemia. So I think we need an alternative way to think about some of these processes. And I'll present a little bit of that in a second. So let's first talk about some of the who's who in the microcirculation of the heart. Who are some of the players? And by the players, where is resistance to flow controlled? This is a piece of work that I did a number of years ago with the late Mel Marcus, in which I had made measurements in the beating heart using micropuncture techniques. And I like to tell all my trainees that I had a full head of hair before I started that, in which we used micropuncture techniques with a three-dimensional electromechanical micromanipulator that we could train to move in synchrony with the beating heart. And we measured pressures under control conditions, the solid dots, open circles, or during diapridamol-induced dilation. The point that this slide illustrates is that in the coronary microcirculation, roughly 20% of the total resistance is in vessels larger than 200 microns in diameter, which is not as much as it is in the brain, with the PO circulation being a huge component of resistance. But nonetheless, small arteries do contribute a fair amount. The vast majority of resistance is in the arteriolar component. That's roughly 60% of the resistance. And that component is extremely reactive. Notice with diapridamol what happens to the pressure drop. Pressure drop under control conditions is from here to here, across the arteriolar bed and the capillaries. And if we dilate, we essentially have the pressure drop at the time we're increasing flow 6-fold. So that means resistance goes down about 12-fold in that area of the circulation. So that's where flow is controlled, in the arterioles primarily. Who are the players in this control? So let me refer back to the negative feedback scheme that I showed you, which I don't believe works for aerobic dilation. I think it works for anaerobic dilation and ischemic dilation. That's a different scheme. But aerobic dilation is different. And let me present this scheme to you. So one of the hypotheses we entertained a number of years ago is that mitochondrial-derived hydrogen peroxide links myocardial metabolism to coronary blood flow. How would this work? Was, you know, in the mitochondrial electron transport chain, oxygen can be converted to superoxide by its reduction by an electron. So in the process of electron transfer, some electrons leak. There's a finite leak of electrons, and that superoxide is rapidly converted in the mitochondria to hydrogen peroxide. So the thesis is, during increased cardiac work, because the flux of electrons is greater in the mitochondrial electron transport chain, the amount of ROS produced, specifically in this case hydrogen peroxide, is also greater. So what's the evidence for that hypothesis? This shows work that we did a number of years ago, isolated cardiac myocytes. In a chamber, we could stimulate them with an electrical field to get them to beat from essentially quiescent up to 400 beats per minute. You sample this conditioned buffer, you analyze it for hydrogen peroxide, Non-stimulated myocytes, those stimulated 200, those stimulated 400, levels of hydrogen peroxide rise. And through various pharmacological tools, we could determine that this is of mitochondrial origin, not from some cytosolic NADPH oxidase that would produce superoxide, which then could be converted into hydrogen peroxide. Is the amount of hydrogen peroxide produced in this manner sufficient to cause vasodilation? Again, our isolated cardiac myocytes, stimulate them in a field for 20 minutes, collect supernatant or conditioned buffer from those myocytes, apply it to a microvessel preparation. This is an isolated arteriole. Down here in the open square, non-stimulated myocytes, myocytes that are stimulated at 200, or I should say buffer from myocytes stimulated at 200, buffer from myocytes stimulated at 400. And then administration of catalase. And you can see there's a lot of dilation. B, most of that dilation is inhibited by catalase, suggesting that it's hydrogen peroxide. Now, it's not all inhibited by catalase. That's the point I'd like to make. So if it's hydrogen peroxide working on a blood vessel to induce vasodilation, what is the target? In our hands, that's blocked by 4-aminopyridine, which is a Kv channel antagonist. I'll get back to 4AP in just a second. So this is the nice scheme in vitro. Does it work in vivo? This is more or less a series of studies in which we drove myocardial oxygen consumption with either norepinephrine infusion or with pacing. And pacing is shown in these black diamonds. Norepinephrine is shown in the open squares. We started out at a baseline. Measurements of myocardial oxygen consumption versus tissue levels of hydrogen peroxide. And notice we have a nice relationship between these two variables. Now, one thing you'll note is, and this actually gave us pause, was the levels of hydrogen peroxide are very high, higher than what we would have anticipated, up to 200 micromolar. And one might say, my God, that can cause death in cell culture. Yeah, but that's cell culture. But the other thing that I'd like to emphasize is that this is a very ephemeral event. You turn off the pacemaker, you eliminate the norepinephrine infusion, hydrogen peroxide levels drop immediately. So this is not a sustained increase. It's a very, very ephemeral event. Hydrogen peroxide levels versus coronary blood flow, again, a nice linear relationship. Now, these are correlations. It's not causal. Is there any way that we can further interrogate this to try to show causality in this process? This is what I showed previously with the 4-aminopyridine blocking the effects of the conditioned buffer, the dilator effect of the conditioned buffer. Down here is the effect of 4-aminopyridine on the relationship between myocardial oxygen consumption and coronary blood flow, and this was in a large animal preparation. Now notice 4-aminopyridine shifts the relationship downward and shortens it. To me, that is extremely important when we try to understand links between metabolism and flow. Because in the heart, if you don't shorten this relationship, chances are you're not studying the right thing. Now, what do I mean by that? In the heart, myocardial oxygen consumption is driven by the product of blood flow times the A-V difference. Since the heart is already extracting most of the oxygen coming out of it, the only way for it to increase oxygen consumption is by increasing coronary blood flow. And if we limit that, by definition, we'll limit the increase in oxygen consumption. So this, to me, suggests strongly that 4-aminopyridine sensitive ion channels are critically involved in this coupling. The good news is that. The bad news is there are a whole lot of ion channels blocked by 4-aminopyridine. How can we interrogate this relationship further? So we went to mice. Now, when I started graduate school, I never thought I'd be working on mice, but I will tell you it's been a fun experience. We can measure myocardial blood flow in the beating mouse heart. This is an anesthetized mouse with contrast echocardiography. We don't measure myocardial oxygen consumption, but we measure a very good surrogate, cardiac work, heart rate times stroke volume times blood pressure. And these are the types of relationships we can get. This is a myocardial blood flow versus cardiac work in wild-type mice and in mice that are null for a Kv 1.5 channel. KV 1.5 channels are a redox-sensitive, voltage-activated potassium channel. But the redox-sensing element is really critical in its regulation. Of course, hydrogen peroxide, being a reactive oxygen species, can modulate redox-sensitive proteins. And so you can see that this relationship is greatly attenuated. Now, what I haven't shown you is the knockout is global, so what about a rescue? Well, we can re-express that channel only in smooth muscle under the control of doxycycline, and we can completely restore this relationship. So this is a smooth muscle event. Not to slight the endothelium, Dr. Aird, but it's a smooth muscle event. Let me just back up. Now, one could say, if there's a cynic in the audience, say, well, Chilean, it just looks like this heart is more efficient. It's getting as much work done at a lower flow. The point I'd like to make on the next slide is this. We make our measurements at sort of a quasi-steady state. In a wild-type animal, you administer norepinephrine, blood pressure goes up and stays up. Notice what happens in the KV 1.5 null. It goes up and then it falls. There is insufficient flow to maintain the level of metabolism. And if we keep the norepinephrine infusion going on, the animal dies. So what about a chronic stressor? Normal mouse heart beating. This is a mouse heart. I'm going to shut this one off. This is a mouse heart from a KV 1.5 dull animal that has what we call a Takotsubo-like cardiomyopathy, apical ballooning syndrome. This is the consequence of microvascular disease in the heart, insufficient flow to maintain the needs of metabolism. And down here, this shows the male-female differences in death due to heart failure. So, Dr. Miller, I hope you're going to be happy about that one. Okay, so who are the players? Hydrogen peroxide, many to-be-named components. I think there's a purinergic component involved in this process. I think nitric oxide is also involved, but involved in the recruitment of big vessels or large small arteries. Who are the transducers? I think KV 1.5, we have evidence for 1.2 and 1.3, and probably many more candidate channels as transducers. Lessons to be learned. Transgenic and gene knockouts, I think, are really effective ways for us to study the molecular signals that connect flow to metabolism in any organ system. Coordination of neurohumoral signaling, I didn't go into that. But let me just mention down here, a large fraction of patients, this is from clinical observations, including the WISE trial, show symptoms of ischemic heart disease without disease of large vessels. So this really takes me back to Dr. Aird's comments when he showed the way a physician might look at something in terms of billing. I think one of the real hindrances to this area in its advancement, many things like microvascular disease in the heart are not perceived as a clinical problem because the cardiologist doesn't have something to check. It's not a box to check. And if the patient has signs and symptoms of ischemia but no vessel disease, then it's stopped there. They don't interrogate it further. And I see I'm being squeezed out, but let me just say that in 1997 we had a workshop on the microcirculation of the heart, and we perceived then that there's a discrepancy between experimental studies in the microcirculation and the needed clinical investigations. And I would say in 2014 that need still exists. Thank you.
Time for a few questions.
So, Bill, I really enjoyed that. I'm going to ask a question that an undergrad might ask, and that is, you pointed out, or you claim, that the heart extracts as much oxygen as it can, and so that oxygen consumption is dependent upon flow. My understanding is that the PO2, leaving even the hungriest heart, is still around 30 or 40 in those veins, so it doesn't appear to extract everything. Is that true or false?
No, that's false. The PO2 in the coronary sinus is typically around 20. So there's still... Yeah, but if it's zero, it would probably be dead. Okay? You can't extract it all. You need something. Because remember, if it's zero, that would be probably zero in the mitochondria. Yes, Virginia.
Yeah, Bill, thanks for putting a male-female slide in there. And I'd like to pursue that just a little bit further. I mean, Takasubus is typically seen more frequently in women compared to men, especially older women under stress. And you have elucidated some of the molecular mechanisms associated with oxygen consumption. Have you dissociated those mechanisms between male and female animals in terms of the channel expression or the redox state relative to mitochondrial regulation by estrogen and so on?
The quick answer is no. I mean, there's a lot of work to be done.
Just want to ask a quick follow-up question, kind of on the undergraduate level. Can you define for us, put us all in the same playing field in terms of what a definition for a microvessel is, or the microvasculature, and what a definition is for the capillary, and how those are the same or different?
Well, being a card-carrying member of the Microcirculatory Society, I'll have to tell you that if you ask 10 microcirculationists what a microvessel is, you're going to get 20 opinions. So, but generally, you know, I view a microvessel as a vessel not in terms of a binary system that this is a micro, this is a macro. I view it as more of a continuum. So when I refer to microvascular disease, it's probably a bit of a colloquialism, but it's principally those resistance vessels that are the flow controllers.
I'm going to speak out of ignorance regarding, you know, there's always been, with supply-demand matching, there's always this issue of where the production of the item is and where the action is. I mean, you've got the exercising muscle and you've got arterioles which are regulating the bloodstream. Is it freely permeable? Is it acting through a second messenger? How is the hydrogen peroxide actually signaling?
Well, the hydrogen peroxide leaves cardiac myocytes. Whether it leaves through aquaporins or not, we don't know the answer to that. But it leaves cardiac myocytes. We can collect it in tissue or in a bath. So it's obviously getting out somehow. And it's interacting with the KV channels and probably other potassium channels. I think Dr. Gutterman has lots of data suggesting that H2O2 works on channels other than the KV channels. So whether or not it's reacting with an extracellular component of the channel or an intracellular component or some type of regulatory protein that affects channel activity, don't know those answers yet.
Last question.
Yes, it's great. I was wondering, you know, where does the resistance lies in the different vascular districts? So you said you made your case based on the cervical measurements that is really the surface arterioles, which would be equivalent to the PL arterioles in the brain. Now there is a controversy in the brain right now of what controls, you know, vascular resistance. Some people say it's the capillaries, and some people say it's the PL arterioles. And I wonder, in your experience, you know, I know some of those measurements have been made also in the cerebral microcirculation. Where does that fall?
Well, since I'm not in the cerebral field, I feel free to express my opinion. Obviously, the arteries.
All right, thank you.
Well, I'm happy to introduce the next speaker, who's Dr. Gerald Menninger from the University of Missouri. He's going to speak on vascular smooth muscle mechanotransduction, a tug of war.
My thanks to the organizers and the chairs. I appreciate the opportunity to speak before you all. Today I wanted to focus on vascular smooth muscle cells and try to expand a little bit about what mechanotransduction is to me. I want to provide a little bit more of an explanation about what mechanotransduction is, and that I think it's part of a system of responding to mechanical forces that involves, importantly, mechanosensation as well as mechanotransmission. And this mechanotransduction process is basically just a way of translating mechanical forces that moves in a two-way street. It's clear that this process of sensation, transduction, and transmission plays multiple roles in the circulation. For example, it's very important for regulating contractile tone. We know that the regulation of contractile tone is linked to blood flow regulation. We also know it's linked to vascular wall and cellular remodeling and development, for that matter. And we know that it's linked to many different disease processes. And just a few examples here: hypertension, diabetes, and even large vessel stiffening, even though this is a small vessel conference. In today's talk, what I want to do is just briefly introduce some background and traditional concepts, and then try to stress the fact that the vascular wall responses to mechanical force and the cellular responses to mechanical forces are very dynamic and adaptive in nature. And then finally, where do we go from here and some possible future directions? Now, just as a very quick review of what the major mechanical forces are that we're dealing with in the vascular wall, there's shear stress, which impacts primarily the endothelial layer or intima. There is circumferential tension or stress, which is introduced by the intravascular pressure in the wall. And there is longitudinal stress, which is not frequently talked about, but it is a stress that we find in many of our peripheral vessels. The other thing I wanted just to ask you to look at here is when you look at the adventitia, media, and intima, these diagrams we often see in the textbooks make it look like the vessel is a layered cake, that each layer is totally independent from one another. Our smooth muscle cells are nice and uniform in shape and wrapped circumferentially around the wall, and I'll come back to that point. Now, when we think about how vascular smooth muscle cells sense, respond, and transmit force, one of the first things we are driven to think about a little bit more is, well, how are they connected? And how is that force getting into cells? And so this is just a brief summary of some of those major routes. There are vascular smooth muscle cell to matrix connections, which are governed primarily by a family of proteins called integrins. There are vascular smooth muscle cell to smooth muscle cell junctions, adherens junctions. In smooth muscle, this is primarily N-cadherin. And then we have endothelial cell to smooth muscle cell junctions, which occur in the fenestra of the internal elastic lamina between the endothelial layer and smooth muscle cell layer. And finally, a little studied area is we also have smooth muscle, and I'm kind of generically grouping a whole bunch of cells together to mural cell connections. And these could include neurons, they could include pericytes, they could include fibroblasts. And this is a little studied area. Now, one of the most widely used preparations in this field has been the isolated perfused arterial. It has a number of strengths which have allowed many advances in the field of looking at mechanotransduction. These strengths include we can control the mechanical forces. We can control the intravascular pressure. We can control the flow through these vessels. We can fluorescently label different components of these blood vessels. And we can also label proteins and look at their spatial distribution. We can use conventional light microscopy on these vessels, and we can use more advanced fluorescent microscopy techniques like confocal and two-photon microscopy. And lastly, these vessels can be biochemically assessed if we're interested in cell signaling pathways. Weaknesses of this technique: it involves removing the vessel from its native environment. So we are studying it in isolation, but that's also a bit of a strength for the technique as well. Just a little bit of history here in terms of the origins of mechanotransduction in the vasculature. This probably dates back to Sir William Bayliss, who was the first to publish a paper indicating that the vasculature is responsive to changes in intravascular pressure. And this is usually diagrammed in single arterioles with a pressure diameter curve that looks like this, where if the vessel is passive with no tone and you increase pressure, it distends along a passive distension curve. If the vessel has tone, and in a normal vessel, this is about 100 micron arterial, the pressure is going to be between 80 and 90 centimeters of water. As you increase pressure, the vessel will contract. As you decrease pressure, the vessel will dilate. And I also want to point out here that this same relationship exists with or without the endothelium present, indicating that it is intrinsic to the vascular smooth muscle. That's not to say that endothelial cells cannot contribute, but the basic response to the pressure and force is the smooth muscle cell. Now, we wanted to look at the cells within the wall, so we developed an imaging approach that allowed us to look at individual smooth muscle cells. And this shows an arteriole that would be running this way across the wall, and we used a technique called fluorescent dye exclusion, where we simply added fluorescein to the vessel bath. The fluorescein leaks in and around and between the cells, and then we can invert that image to get a nice picture of the smooth muscle cells. And what I want to point out is if you look at these cells, they're not nice and uniform and all oriented in the same angles and directions. They have different shapes. They have different relationships with one another. It really looks like a heterogeneous population of cells, and that's what I believe it probably is. Now, we did a very simple experiment to look at how these cells transmit force and how they sustain that transmission of force. And we did that by adding norepinephrine to one of these vessels and following the shape of the cells. And this next slide just summarizes that experiment very briefly. When you contract an arteriole, and in this case it was using noradrenaline, and we looked at the vessel 5 minutes after we took a through focus image series, and look at the individual cells, and we've pseudo-colored 2 cells here just to look at for a moment. The diameter was 91 microns. The overlap between these 2 cells is about 16 microns. When the vessel contracts, you can see the cells shorten, you can see them fatten, just the way we conceptually would envision this process. However, if you leave the noradrenaline on this vessel for 4 hours and look at those same 2 cells, what we noticed is that the cells are reestablishing their initial length. The overlap between them has increased. They're moving past one another, yet they're still constricted. And we have this change in overlap. So the cells are not cemented in place. They can rapidly adapt, change position, either by changing relationship with the matrix, changing relationship with each other, or cytoskeletal remodeling. And I believe probably all 3 of those are taking place. This next little movie I wanted to show is just to illustrate and attack this layered concept a little bit. In this particular image, the smooth muscle cells are going to be labeled green, as I showed in the previous image. The red is elastin fibers in the vascular wall. And I just want to show how complex this network of elastin is and how it changes as you penetrate through the vascular wall. And what is apparent when you play this over and over and over again and study it is that if you look at the outer layer, most of those elastin fibers run in the longitudinal direction. If you snip them, the vessels lengthen, so they do keep the vessel under a longitudinal compression. The medial layers run mostly circumferential, not longitudinal, and there are penetrating perpendicular fibers that pass all the way down to the internal elastic lamina and attach to it. So it's an integrated structure that runs all the way through the wall. And this structure changes depending upon where you obtain your vessel, what stage of development you're in, and it's quite different when you look at cerebral vessels. I feel I had to bring that up. The cerebral vessels totally lack the external adventitial layer, and all you see is the internal elastic lamina. So they're very sensitive to longitudinal stress. So as a brief summary, not all vascular smooth muscle cells are the same. Cells are dynamic. They have a positional plasticity. The ECM is an integrated functional part of the vessel wall and needs to be understood just as much as we understand the living part, the cellular part of the wall. If the matrix is disturbed, function is disturbed. And we postulate that this dynamic process is an efficient means of tuning the contractile state of the smooth muscle cell to the mechanical state that exists in the vessel wall and the mechanical environment. Now, we took this to the cellular level. To do this, we used an atomic force microscope where we basically, at the heart of the system, is a cantilever with a tip that we can interact with cells. We isolated our cells from microvessels. We can position this tip and we can record the forces that are encountered at the tip. And we can calculate things like the cell stiffness or elasticity as well as adhesion force. This next little movie is one example of how we do this, where we take an AFM probe, we coat it with a matrix protein. It might be fibronectin or collagen. We indent the cell. You can see the deflection. We use this information to calculate the estimate of elasticity using the Hertz model. When we retract the probe, if there are adhesions between the fibronectin coating the probe and the cell, we get a slightly different retrace curve. We can use this information to calculate adhesion or how sticky the cell is. And we did a very simple experiment. We treated these cells with angiotensin II or adenosine, and we looked at stiffness. And this column of graphs right here shows a single cell example. This shows a group example. What I want to point out is if you look at the angiotensin II example, this bit of black here is the control period. A2 was added at the dashed line. And I want you to notice how quickly the stiffness starts to rise in the cell. And I also want to point out that this is cortical stiffness, since we're only indenting the cells about 150 to a maximum of 300 nanometers. So it's just the very surface of the cell. Also interesting, I don't have time to go into it today, angiotensin II throws the cells into an oscillation pattern that you can record. And that's what you see happening here. And with adenosine, we saw just the opposite. We see a decrease in the cortical stiffness of the cell. You can also assess the adhesion of these cells, and we did this by looking at that retraction curve. And what we notice when we look at adhesion, if you look after angiotensin II, there's a marked increase in the density of adhesion events. There's many more adhesions between the probe and the cell, and when they break with higher force. If you look at adenosine, you see just the opposite. We see a decreased density of adhesion events, and the breaking force is declined. So both of these things indicate that when cells contract, they get stickier. When they relax, they get less sticky. We also wanted to look at remodeling of the cytoskeleton. We did this by using the AFM in a scanning mode where we get cell topography, just scan back and forth horizontally. We can reconstruct a height image of a single cell. This is a single isolated smooth muscle cell. And this is a height map, so the brighter area is high. You can pseudo-color it and see that this cell rises about 4 microns above the substrate over the nucleus. And we can also process these data so we get a deflection image, which allows us to delineate the stress fibers underlying the cell surface membrane. Now, just a real quick analysis scheme that we used. This shows pre-angiotensin. We scanned in high resolution a 40 by 40 micron square, shown in blue. And we drew a line across this cell and looked at the scan after angiotensin II. There's an increased number of stress fibers. The stress fibers are bigger, and if you do an analysis of orientation, they show that the orientation is increased. Whereas angiotensin II, we saw just the opposite effect occur. In fact, the not angiotensin II, just adenosine seems to cause a depolymerization of the cytoskeleton and disorients it. And nitric oxide has even a more profound effect. So in summary, smooth muscle cells are very mechanoadaptive. in the wall. The adaptation involves rapid changes in the stiffness, stickiness, and cytoskeletal structure of the cell. And it supports the conclusion that the cells themselves tune themselves to their mechanical environment and the mechanical work that's placed upon them. Lessons learned, mechanotransduction is multifactorial. There's more than one sensory pathway and molecule. There are many signaling pathways that are involved, which I didn't go into. And the process can be modulated by multiple cell types, circulating factors, and cytokines. The process is dynamic. It involves more than changes in contractile tone. It's more than just actin-myosin interaction. And it operates on a temporal continuum, so that anytime you look at a model system and perturb it mechanically, you're looking at a snapshot in time, and it's going to change depending on when you look at that. And then gap analysis. What do we need to improve our basic understanding of mechanotransduction so that we can use this information to fight disease? We need more knowledge of the time-dependent events. We need a much more detailed understanding of the extracellular matrix structure and where cells interact with it and how that structure behaves. We need to look at intracellular interactions. We need to look at how integrins interact with cadherins and how one cell interacts with another. We need more detailed understanding of the cytoskeletal compartments within the cell and how they behave differently. And finally, technically, we need much better visualization approaches, faster ones. We always want more resolution, more speed. That's common for biologists to want that. And I think, in addition, and I heard this earlier, we need models. We have spent a generation breaking the system down into smaller and smaller parts, and it's time to start reassembling this and working as teams, mathematicians and biologists, and building models that are predictive, that can be tested, so we can go back and forth. And I think this will improve the speed with which new concepts develop. Thank you.
Thank you. I think we have time for just one question. Well.
Yeah. Thank you, Jerry. Edensco, University of Alabama, Birmingham. You sort of did a nice job of depicting what the intricacies of this mechanosensation process is like, but what happens to this process under conditions of chronic hypertension or even acute changes, of marked changes in pulse pressure?
Those are good questions. We're looking at hypertensive cells now. They are different from normotensive cells. They're much stiffer, they're much stickier. We haven't yet done any dynamic studies to see how their responses are different.
Jerry, I'm going to ask just a very quick one. The endothelium is critically important in the cerebral vasculature, as you pointed out. You're denuded endothelium, yet you still got smooth muscle effects. What if you damage that endothelium? Hypertension, rheologic factors, tortuosity factors, anything that would affect that endothelium. Then what's going to happen to your smooth muscle reactivity?
Short answer is we never did those studies. It's a good idea, but we haven't done that.
Thanks, Jerry.
So.
Thank you.
Okay, our next speaker in this session is going to be Dr. Nelson, Mark Nelson from the University of Vermont, who's going to talk about small blood vessel disease in the brain. Dr. Nelson.
Thank you. Well, thank you very much for the invitation to speak. I'd like today to talk about some of our recent work on small vessel disease in the brain. And here in the talks this morning, I'd like to link the previous talks up with my talk. I'm going to be looking at the small vessels, the penetrating parenchymal arterioles that David Kleinfeld described as the bottlenecks. I think these are the reasons why small vessel disease in the brain occurs in these spots. We're going to look at, we see an early channelopathy-like event in small vessel disease model that involves KV, probably KV 1.5, next talk that was presented. Probably also involves changes in the extracellular matrix and metalloproteases, the next talk, and something else that coming up later on that I'll show you. So where do I advance them? So it's okay, here we go. Just a little bit of background. I want to give us who we are. We have a program project grant on small blood vessels in the brain. And we want to thank the Heart, Lung, and Blood Institute for that. Marilyn Cipolla and George Wellman are here today. We also have an international network funded by the Foundation Leduc, and some of the members are here. Mike and Niedergard, I saw her in the back earlier, Frank Ferrasi. This is a network to look at small vessel disease in the brain, including basic scientists and clinicians in Europe. So this has been a wonderful opportunity to look at it. A little bit of background. I'm just trying to get every speaker that's being a person in the audience here. The arteries, which you heard about in the first talk that we look at, the arterioles, penetrate from the brain surface into the brain and are surrounded by astrocytes. And these are the key arterioles that we'll be looking at today. So they're in a unique environment. They're surrounded by astrocytes. So that's something different. So small vessel disease, it progresses silently over many years before becoming clinically symptomatic. It contributes to more than 25% of the strokes. You heard something about it this morning, as well as from David Kleinfeld. Leading cause of age-related cognitive decline and disability. No specific treatment. Major risk factors are age, which I'm getting there. High blood pressure, I'm working on that. Blood pressure variability, who knows? And so our noble and lofty goals of the network are to provide progress on understanding molecular basis, obviously markers and therapeutic targets for small vessel disease, train scientists and clinicians to work together, and this is what this meeting I think is doing beautifully. And really what you've already heard today too, that small vessel disease, like any disease of any organ, is not simply the tissue of the organ, the heart muscle or the brain, but is in fact all of them have to work together. And I was long ago in a neuroscience program at WashU, and we didn't mention blood vessels or glia once. I mean, and if blood vessels were mentioned, it was a stroke, and that was something we didn't really talk about. So I think they come hand in hand from, as you heard from David Kleinfeld, in controlling behavior and may relate to a lot of cognitive disorders. So what do we study? We are using one of the few monogenic causes of small vessel disease as a model. It's a very rare but monogenic disease, and we view this as a lens into the bigger sporadic causes of small vessel disease in the brain. And this model is called CADASIL. About 1 in 100,000 people have it. It's autosomal dominant. It's the most common cause of hereditary small vessel disease of the brain. It often starts with migraine with aura in the patients in their 20s. Microinfarcts, which you heard about, cognitive deficit, motor impairment, and usually early death. And these are caused by mutations in the Notch3 protein in the extracellular domain and substitutions of the normal 6 cysteines and the EGFR repeats that to an odd number. And this leads to what we think is of the extracellular domains of Notch3 clumping together that start a cascade that leads to the pathology over years. And these are some of my coworkers in Europe in the Leduc network that provides some of the clinical evidence. So this has been one of these very frustrating things where the gene has been known for years, nothing can be done, and fortunately recently there's been a mouse model developed which I think is starting to provide some insights. This, also should mention, Notch3 is expressed only in the vascular smooth muscle and pericytes in the brain. And so remember, it's the extracellular domain sticking out, and the next cell it sees is our astrocyte end foot. So keep that in mind, although we don't, we're still sort of at early days. These are some of the techniques that we bring to bear in the rodent models for anything from isolated cells from the arterioles, proteomics. As you heard on the last talk, pressurized arterioles. These arterioles are 20 microns in diameter from mouse. EM, brain slices, and in vivo work as well. This just gives you one of the new tools we have. We have also, this is a calcium biosensor, genetically encoded calcium biosensor. This is a parenchymal arteriole, 20 microns in diameter. This is a ratiometric calcium indicator, and you can just see we're adding a KV channel blocker, and it constricts and calcium goes up. And you can see nicely the smooth muscle cells as they penetrate into the brain to go to the terminal arteriole. I think models like this will be very valuable in understanding what goes on in small blood vessels in any tissue. We also have a similar endothelial mouse. Our European colleagues also try to take the information, we try to bi-directionally, take not only proteomics from autopsies for deposits from CADASIL patients, but also there's a number of genome-wide association studies going on in Europe on CADASIL patients and patients with sporadic small vessel disease. So here are some concepts, and I think will resonate with people, the talks you heard this morning. These are things we're just throw out to you for food for thought. We're proposing that the mutated extracellular domain of the Notch3 changes the dynamic landscape of protein accumulation extracellular matrix to alter smooth muscle cell function and astrocyte function. That there are dynamic changes, which you heard about from Jerry in the last talk, in the composition of the extracellular matrix between the smooth muscle over time, and we think that some of these changes are common denominators and perhaps in a broader sense of small vessel disease. And one of the ones I'll focus on is changes in metalloprotease activity and the level of the number of Kv 1.5 channels in smooth muscle cells as being an early defect in small vessel disease in this CADASIL mouse model. So we use a mouse model developed by Andrew Tellem, a European collaborator, and was described recently in the Journal of Clinical Investigation. It's a mouse model that expresses one of the human mutations for CADASIL, R169C. The mouse was developed overexpressing the normal transgene fourfold and the mutant transgene fourfold. And this mouse then developed some of the salient preclinical features of CADASIL, including white matter lesions at over the age of a year. But one of the interesting things is what happens early in the disease process. And I think we're all interested in trying to understand the early changes. And they could be a lot more subtle. It can be because we can induce a microclot, or they could be something as subtle as a small change in how the vessel reacts to pressure, which is what I like to propose today. And these things could happen early. They may have long-term consequences. And Anne in her paper showed that pressure-induced constrictions are reduced in the big arteries, the peel arteries in the brain at 6 months of age. This is at least 6 months before the onset of white matter lesions. And whisker-induced blood flow is also reduced. So we wanted to look at the arterioles within the brain, and this shows one of our micrographs where you can see that this moment the arteriole penetrates in the brain is encased by acidic processes, and the red staining is aquaporin 4 for the end feet, and this is the middle cerebral artery. So this shows the basic study you saw in the sort of the Baylis effect, increasing intravascular pressure on a cannulated 20 micron diameter parenchymal arteriole from a mouse brain. And what you can clearly see here is that the blood vessel constricts to pressure in black, and the CADASIL mouse arteriole constricts the same amount at 20 millimeters of mercury, but at higher pressure, 40 being probably physiological for this part of the circulation, constricts about half of what the wild type does. So what this says, this looks like what Anne saw in the peel arteries, but this says a fundamental part of the process, how the blood vessel senses voltage, pressure, is altered in this model of CADASIL. But it's not altered at lower pressures, only at higher pressures. So it's something that kicks in at higher pressures. And what I want to convince you of, what happened, this difference is due to an upregulation of one of the brakes on contraction, upregulation of the Kv, voltage-dependent potassium channels. You upregulate a brake on the pressure-induced depolarization, you break contraction. And so this happens at least 6 months before white matter lesions. But I should say the extracellular domain aggregates are detected within a couple months of birth of the animal, as well as the deposits around the arterioles. CADASIL and small vessel disease, CADASIL is a disease of the brain, yet the deposits are found everywhere, and the mutations in all vascular smooth muscle. So we looked at the mesenteric arteries and looked at the same properties. And just to make a longer story brief, the mesenteric arteries, there was no effect of the mutation on any of the properties we could detect in the mesenteric arteries, yet they have mutated Notch3 and they have gum deposits. So we then went back to one of my colleagues, George Well and Messiah Cody are here, and they had shown a couple years ago that in a subarachnoid hemorrhage model, that instead of the arteries constricting less to pressure, they constricted more. And this entire difference was due to a downregulation of voltage-dependent potassium channels, and just the channel number. It looked like there was increased endocytosis. And they showed that, this shows that one of their curves, and so we thought there might be parallels here, that they showed a downregulation of KV channels. So we also looked at KV channels in the arterial myocytes from these arterioles. And what you can see here, control, large increase in the current density of voltage-dependent, probably KV 1.5 channels in the catechisal arterioles, which would mean a bigger break on constriction. And analyzing the currents indicated that it wasn't a change in the properties, it was a change in the number of channels. And George and Messiah over here, their work showed 4 days after introducing subarachnoid hemorrhage blood, there was a downregulation of the same channel by endocytosis. So we thought this is an interesting parallel. So this says that the KV channels have to be, their number is critical for how the artery responds to pressure, depolarization, and they're the brake on it. You change the number, you change the set point. So then this sort of gives a cartoon of it. So the idea is membrane pressure causes membrane depolarization. And in the example that was previously published, that you have a decrease in the number of channels, more depolarization, more constriction. And what we see is an increase in the number of channels, less constriction, and less depolarization. Both can be bad because it means the arterioles are sensing pressure in very different ways and inappropriate ways. And their study suggested that it was metalloprotease activity, an increased metalloprotease activity that led to the decrease in channels. So these show an artery from Anne where these deposits around the arteriole. And so we thought, well, so Anne started investigating. This is how science comes together. And she found one of the proteins that was concentrated in the deposits caused by the mutant Notch3 was an MMP inhibitor, metalloprotease inhibitor, TIMP3, both in deposits from human and the mouse model, making us feel more comfortable about both, I guess, certainly about the mouse. So we have a situation here where downregulation of subarachnoid hemorrhage increased MMP activity. In catechisal, upregulation increased MMP inhibitor. So what we did separately, we increased MMP activity in these arteries in the way George Wellman did by adding something that would increase MMP activity, or the downstream product of it. And Andrew Tell decreased TIM3 activity, MMP inhibitor. In both cases, we're able to restore normal myogenic tone. So a fundamental question becomes, what happens when you can correct the deficit early on in the pathology? How does this contribute to the overall pathology? And so take-home message, small vessel disease channelopathy, because of the change in number, not properties, is a regulation of the number of voltage-dependent KV channels in the smooth muscle cells. Probably KV 1.5. This is determined by metalloprotease activity, which is dynamically, and this process dynamically determines blood responses to blood pressure fluctuations. Too many channels are not good, and too few are not good, and may be pathological. And so I had this made up in one slide. Challenges of the study of small vessel disease of the brain. And this is really, you could put it for a lot of things. Requires a spectrum of diverse scientists and clinicians. That's why we're here today. That's what, and our Leduc network and our appropriate PPG make this possible. In the brain and any tissue, it's a unique environment, which you've heard about, in this case encased by astrocytic processes. So we have to think about how the astrocytes change, and particularly their end feet. You'll hear from Mike and Niedergaard later on that there's a flow of fluid right down by these cells that can change extracellular matrix, could potentially change extracellular matrix. I think it's critical to have the appropriate mouse models for disease processes, because we really didn't have anything for humans in small vessel disease. Appropriately genetic encoded biosensors are key, because otherwise putting fluorescent dyes and other things are impossible unless you have them in the right cell type. There's also difficulties in adapting molecular imaging techniques to the mouse microcirculation, which I think have been largely overcome. I think it's important in all these tissues to isolate. I know it's in isolation, but you also should do the brain arterioles as well, or the arterioles from the tissue bed of interest, and the dissociated cells. Culture is an issue. And we need a critical mass of scientific information expertise. Linking up rare Mendelian disorders with sporadic small vessel disease is a key thing. And I would suggest that early functional changes may be very important for the long-term pathology. I'd like to thank all the sponsors and supporters, particularly the Heart, Lung, Blood Institute. George is here. This is subarachnoid hemorrhage people. Collaborators in Europe. Fabrice did most of his work, View from Our Window, and Jutel, her postdoc. Any of the really heavy clinical questions, don't ask me. And here's the lab. Full disclaimer ahead of time. Okay, stop my stopwatch. Sorry. All right, open for questions.
Yes. So I have a question. So, what do you think notch signaling is transcribing?
I don't. Our current hypothesis, it's really the accumulation of the extracellular domain that then oligomerizes and then accumulates other proteins and changes the landscape. So it's not the transcriptional activity per se, but it is, and it's not really maybe the deposit, but how it changes in a dynamic way, as we heard from the previous talk, extracellular matrix protein. So it would be the notch-free extracellular domain cascade hypothesis.
I mean, that's fine, but notch is still regulates transcription, and so could one argue that notch is regulating a transcription of a mechanical sensor, and thereby influencing
it could. And the
reactivity?
interesting thing, when Andrew Telf came first to discuss this program with me, she said KV 1.5 is a target. And I said, why? Because notch 3 intracellular domain is a transcriptional regulator at KV 1.5. Because I told her it's KV 1.5 because the arteries don't constrict. And by the way, 4AP also normalizes the differences. So we were looking at it physiological, and she was looking at it genetically. However, it does regulate trafficking, and I think the trafficking is a big issue, a big component of it. Hey, Bob.
Bob Bryan, Baylor College of Medicine. Nice talk, Mark. So, with this lack of break, the arterioles and acatocil mice are probably going to have a different structure, so that the lumen. Or what about lumen size? What I'm thinking about getting to is the pressure in the capillaries.
Yeah, that's a really good question. I don't know, maybe, but there is some. And it showed there was some, I get it right, inward remodeling of the peel arteries. The diameter was smaller. We can't say because we never take the same. It's hard to get exactly the same piece of the terminal arterial. So there are structural changes that go on as well. Would you argue just in face value here that the pressure fluctuations would not be as buffered as well in this model of the arterioles not constricting as much? But I don't know, and I think our network's trying to investigate that.
Jan Kirievsky from Columbia University. Just to follow up on Azrar's question,
yeah.
We established that notch regulates MMPs transcriptionally in endothelial cells, and we're seeing similar responses in human brain vascular parasites. We also, I could share with you
Notch 3?
Sorry?
Which notch?
Notch 3 and notch 1.
Okay.
And in addition, a compound mutant of notch 1 and notch 3 has severe parasite defects early in development in neonates.
Okay.
That includes accumulation of extracellular matrix, excess MMP activity, and in fact we can even see development of GOMs in the brain vascular parasites of neonates. So I think your hypothesis that there's a key extracellular matrix component is correct, but I would suggest that there's certainly a transcription.
Oh, I didn't even mean to suggest there wasn't a. I mean, it's. So
I'm just sharing it with you a
that's
variety
really interesting.
of unpublished results on that area.
All right, well, and so I was hoping somebody in my lab recorded that, so.
Yes, well, I'll be glad to talk to you.
Okay, great, thank you.
We're seeing notch regulation of tight junction proteins too. Right there at the junction, the notch regulation of the claudins and the occludin is occurring. So, I mean, you've got notch regulation of not just the basement membrane, but also the proteins at the endothelial cell.
These are all smooth muscle cells. So.
I understand, but notch regulation of trafficking is also occurring for those exact proteins that are making up that tight junction. So the trafficking effect of the notch is real at different points in the
we haven't looked at it. I don't think, if I remember correctly, I think at the moment, at least early on, Ann didn't see changes in the blood-brain barrier. But she's looking at it now. Okay, so we got one of our
I just thought I'd bring that up because it seems like a unifying concept.
Yeah, I appreciate it. Thanks.
Yeah,
sorry.
Ariel Gomez again. A comment that I have. Notch 3 deficient mice have a poor regulation of blood pressure. They are poorly responsive to vasoconstrictors, and they have an abnormality of the arterioles peripherally. And I wonder if that doesn't contribute also to the CADASIL in the sense of being hypotensive, compounding the process that you see in the...
It could. I mean, and this is really Andrew Tell's work, but she has provided, I think, fairly compelling evidence that at least that the Notch3 mutation is not due to a loss of function. And I know there's some discussion about that, but I don't...
But in vivo, they just respond very poorly to vasoconstrictors. I mean, that's very well known now.
All right. Thanks. Thank you very much, everybody.
Okay, if we could ask the panel discussants and the speakers to come up and engage in an open discussion.
So besides the speakers, we'd like to welcome David Guterman from the Medical College of Wisconsin and Stephen Macknick from the SUNY Downstate to the panel. And I guess to follow what was done in the first panel, we'll have Dr. Guterman and Dr. Macknick say a few words to start out.
Okay, great. Thank you. All right. Well, I have a whole lot of comments I could make, but I'll just start out with a few because I think the discussion from the audience is really quite helpful. Following up on one of the items that Dr. Chilian talked about has to do with the cardiomyocytes producing hydrogen peroxide that then interact in the smooth muscle. And John Canty published a number of years ago editorial talking about hydrogen peroxide kind of attacking the blood vessel or the smooth muscle from two different angles. And some of the work that we've done and others have shown that the endothelial cells also produce hydrogen peroxide as a vascular vasodilator mechanism for flow-mediated dilation. So you have the endothelial cells as well as the cardiac myocytes in the heart impinging on the smooth muscle cells with hydrogen peroxide. But the point I wanted to make is that, as Dr. Chilian mentioned, probably the metabolic dilation produced by this hydrogen peroxide is a transient phenomenon, and hydrogen peroxide levels go up and down. From the endothelial cells, what we see, at least in patients with coronary disease, this release of hydrogen peroxide seems to be fairly continuous in isolated vessels from the endothelium. So it may actually invoke some pathological changes depending on what's being released from the endothelial cell. If you have a normal patient, what is being released from this huge array of microcirculatory vessels in the heart is nitric oxide. But as soon as a patient develops coronary disease, there's a switch to hydrogen peroxide. So part of the pathology that we see, and we know that microvessel disease occurs in humans, we measure it very roughly or crudely by just measuring an alteration in maximum increase in blood flow with adenosine or other dilators. But we know that this abnormality occurs, has just as bad a prognosis or worse than macrovascular disease does, but we don't understand how that's occurring, and we need to really look more specifically at microvessel function, I think, to understand that. We need better techniques in the patients to be able to understand what's going on at the microvascular level. So that's just one example, I think, where hydrogen peroxide may be a bad player, nitric oxide a good player, and the microcirculation being the producer of these elements that's in bathing parenchymal cells, whether it be heart or elsewhere. So I think this is one of the big gaps in our knowledge with that, in regard to that issue. I also wanted to mention on Jerry Meininger's presentation talking about the heterogeneity of vascular smooth muscles, even in the same microvessel that's pulled out of of a tissue. We see, and I think others have also published, that the content of potassium channels expressed in individual smooth muscle cells dispersed or isolated from fresh tissue samples may vary quite greatly. I'm not sure what the meaning of that is or what the purpose is, but there's quite a bit of heterogeneity just in the expression of various surface proteins in smooth muscle cells from adjacent, presumably adjacent cells and adjacent tissue. I can stop there.
Well, my comments and questions lead largely from Kleinfeld's talk, which I think is a really important anchor for this meeting, really, in terms of the basic neuroscience there, at least in the brain. And I think probably the most important concept that he brought up was this issue of coalescence. So you have one penetrating arterial blockage that leads to a massive behavioral change that can be reversed and shown in, with psychophysics of a whole animal to affect these large behavioral improvements. And I think that one of the things that or rather deficits, and one of the things that suggests is that these small vessel diseases can be the 1,000 little pinpricks that lead to humongous changes. Now, one of the things that I was interested in was the fact that a single penetrating arterial blockage, even though the capillary beds are connected to the surrounding penetrating arterial capillary beds, that the pressure wasn't high enough to allow the blood flow to cross or to feed the cells in there. And I wonder if blood flow enhancers would decrease the size of the umbra from a single arterial blockage, penetrating arterial blockage in that case.
So, all right. Your question was why, if you increase flow, like you've gotten blood into this region that was.
Well, it was more if you increase blood flow with nitric oxide, for example.
Oh.
If you increase blood flow with nitric oxide. So you used.
I think it's years of teaching physics 1 where you got to shout.
So you've used excitotoxic drugs.
We went down a lot of blind. The truth is, we went down a huge number of blind alleys looking for inflammation. We didn't actually increase pressure. It's a fair point. It should have been done. I think though the point is that it's not. The real issue is that the venules have very little resistance, so they should just be sopping up the blood. If there's actually a way, the prediction, if it's possible, would be if there's a way to actually constrict the local veins. Now, I realize I don't have smooth muscle, but if there's any trick out there, right, to constrict the veins, okay, then in fact blood should get in from the side.
I see. So it's not the arteriolar side that's going to get the umbra smaller. It's the venule side.
It's the venule side. It's the drains. And maybe it's worth thinking about. I really have no clue how to do this.
Okay. And my other comment was for Chilean, which was that so you showed linear relationships between blood flow and metabolism, and are those two ways? So, do you have effects on metabolism that leads to changes in blood flow, and will the changes in blood flow happening for whatever reason lead to the same correlated changes in metabolism, or is it really causal only from the metabolism to the blood flow?
In the heart, there's a phenomenon called the Gregg effect, and that is if you increase blood flow and increase the vascular volume of the myocardium, you increase the work that it's done. And it's probably due to the contraction of the muscle. If you have more water in the muscle, so to speak, it takes more energy to overcome and move that mass.
So we can take some questions from the floor.
Connie Wyand from Stanford to Dr. Chilean. Bill, so if H2O2 is very important in regulating this, the immune cells are really born and equipped to make H2O2. They make it better than any other cell. Do they regulate blood flow in the heart?
I don't know. I think that's an intriguing idea because certainly the myocardium has an ability to recruit many types of immune cells under a variety of conditions, even conditions that are not profound frank ischemia, but it would just be, or there could some ischemic episodes, but not enough to cause necrosis. And clearly that could be a mechanism.
Yeah, this is a comment for David Kleinfeld. You mentioned that the bottleneck is at the penetrating arterial. We've done experiments in which we embolize vessels of any size, including like a 10 micron terminal arterial, I guess, and we see a tiny microcore of cell death there.
Yeah.
So basically, I think that even the smallest vessels are actually there's not enough collateral circulation to compensate for those occlusions.
Okay, so 10 microns is these would be, what would you call them? Sort of like primary capillaries, things coming off the penetrating vessels. Mm-hmm. Would that be a fair description?
So they would be deep into the brain.
Deep into the brain,
deep in the brain, branches of.
But big enough to supply multiple.
Maybe
I'm trying to get a hierarchy.
Like a two capillary, basically pre-branch,
Pre-branch.
Yeah.
So maybe, okay, you sort of, it's good to put a word on something because some people remember it, but maybe a fairer way to say it is you block a penetrating vessel, you get a quite extended amount of death. In our hands, as this half millimeter. When you go down to to a very small vessel in the mouse, like things that are like 3-4, do you see a cyst?
You don't see a cyst, but if you do high quality histology, you will see that there is a microcore, maybe a couple of dead cells with gliosis and a clear reaction, basically, pathological reaction.
Okay, so I have to say that when we blocked the smallest microvessels, we didn't really see death, but that I don't think it's very important. A few neurons, what's a few neurons? No, but in a serious way. So maybe there's a progression, but I don't think you get this large-scale cyst. I think maybe that's consistent when you block a very small vessel, and I don't, it may not be a threshold phenomena, right?
Howard Crystal, SUNY Downstate. This is for Dr. Kleinfeld also. Could you comment on the human pathologic correlates, in particular because clinicians are always talking about white matter. To what extent are your findings because it's an artifact that you can study the cortex more easily? And why don't we see these cysts in postmortem material?
Okay, well, I understand. I wasn't trained in medicine, so and I try to avoid the physicians too. But I think what you do see, what you see in Harry Vinter's published pathology data, and now what you see in the 7 Tesla data coming from George Biesel's lab, but it's only the last year and a half, are gray matter lesions. Now, the criticism perhaps is that the 7 Tesla work is still at the level of MRI. These patients are still alive, so there's not a correlation with postmortem tissue. So I think it was a technological reason that people didn't see these. It's only when you have 7 Tesla that you get below about 1 millimeter resolution. But it was already there in the pathology data. So it's just, it's not a proof. You look at a mouse model, or you block one vessel, you see a certain size defect. It's water rich, T2 relaxation goes up. You look in the human, recent human data, you see the same kind of signature. It may take a couple of years to match. I think the reason you saw this in white matter, white matter is very water poor. Okay, so the contrast levels were much higher. Okay, and then it raises another interesting question: is why also the defects tended to spread over a larger region. I think that's going to be a challenge. That's part of the challenges, right, for mouse models. But I have a feeling it wasn't that it wasn't there. It just wasn't seen as a technological issue of resolution in the magnets.
Thank you.
Okay. And so if we could have kind of short answers and short questions so we could get all the people that are currently standing and nobody else. Thank you.
John Lederer, University of Maryland, for Dr. Chilean, but also I was hoping the rest of the panel would have perhaps a comment or a thought. In heart, when heart cells are stretched, they have a physiologic generation of ROS in the form, we think, of hydrogen peroxide that produces local signals, but it goes through superoxide outside the cell. Given the intimate nature of the capillaries to the heart cell, one-to-one, real close, there should be a physiologic ROS source with every heartbeat that should also have an effect. Can you, first of all, consider that? And secondly, speculate, could that account in part for some of the ROS effects that you may be seeing for vasodilation?
I certainly cannot eliminate it. You know, if one would stretch cardiac myocytes and you move up on the so-called Frank-Starling relationship and the heart's doing more work, it's going to produce more hydrogen peroxide.
You get 8% with every filling during every heartbeat, and that's enough to produce a signal.
An 8% change in
8%.
Hydrogen peroxide?
No, of stretch.
Oh, okay, okay. But in terms of beat-to-beat regulation, my hunch would be yes. It must play a role. I think the work that comes to my mind, this is old work, but it was work done by Joseph C. Greenfield many years ago, showing that even a slight occlusion during a portion of diastole resulted in a compensatory hyperemic response. So it can happen very quickly.
So I don't know if anyone else has a thought on hydrogen peroxide in the systems.
So I think that's absolutely right. And if you look at the peripheral vasculature, even outside the heart, just the pulsatility, Gabor Cayley and others in Valhalla have shown that just stretching the vessel and others produce superoxide and hydrogen peroxide. So this probably effect goes beyond the heart into the peripheral vasculature. And a clinical situation might be with the most recent onset of use of LVADs, which now produce a more continuous flow rather than a pulsatile flow in the periphery, and may induce some abnormalities in relation to that.
Manfred Böhm, Hart-Lange Blood Institute. I have a question for Dr. Meininger. Response to injury theory or dogma. There is the switch of contractile smooth muscle cell to a proliferative secretory smooth muscle cell. There is a very rare disease where there's a variant, actually a loss of function variant in soluble granulate cyclase, which is a receptor for NO. The hypothesis would be that these smooth muscle cells have a problem.
Let's try to speak more in the microphone.
So, classic response to injury, smooth muscle cells switch.
Even closer.
Huh? Even closer?
Even closer.
Okay.
Perfect.
In the classical response to injury, smooth muscle cells switch from a contractile phenotype to a proliferative phenotype, secretory phenotype. There's a rare disease where there's a defect in the contractility in smooth muscle cell that does not necessarily respond in a problem of hypertension or regulation of blood pressure. It's a response, a hyperproliferative response. So these patients develop moya or achalasia of the stomach. So my question is, if you have a defect in the contractile ability of a smooth muscle cell, is there a kind of switch to a proliferative smooth muscle cell, very similar to the classic theory, and how would that be regulated?
That's a complicated question. And it's difficult to give you a short answer to that. All of our observations in single cells are done in cells that are in primary culture, so they haven't been passaged. But they still change a little bit. We can't help that, but it's the nature of the technology we use to study them. They still do retain some contractile properties. So they're not entirely just proliferative. If we do a concentration response curve with angiotensin II, you can map the height of the cell, and you can see that the height increases with concentration. So they still retain some contractile properties. So I don't think that the responses I'm seeing are strictly because the cells have switched to a proliferative phenotype. It would be interesting to take cells that have switched and see if they're different. But we haven't done that.
So I'm sorry, I'm getting the signals from the bosses here. I'm sorry if you can find the people you wanted to speak to at lunchtime. So I want to thank everybody, the speakers and the panelists. Thank you.
We start. Hi, I'm Eser Tolunay from NHLBI. This is the session on small vessel cellular interactions. Our first speaker is, before that, I'm sorry, my co-chair is Dr. Greg Dalzoppo from University of Washington. And I'm pleased to introduce our first speaker, Dr. Tanya Mayadas from Harvard Medical School. She will discuss leukocyte recruitment in the context of immune complex deposition.
I would like to first start with some acknowledgments. I would like to acknowledge my very talented graduate student, who's Florencia Rossetti. Let me see if I can see it. Florencia Rossetti, who just recently graduated, as well as my former postdoc now, Taki Suboi, who's now has his own group at Nagoya University. So the kidney actually filters about 40 gallons of blood every day because it basically filters the entire blood volume every 45 minutes. So in order to do this, it has actually developed a very sophisticated and ultrastructurally complex microvasculature. So the filtering unit of the kidney is actually the nephron, and we have about 1 million of these per kidney. And the nephron is composed of a glomerulus and a renal tubule. The glomerulus is fed by an afferent arteriole, which then subdivides into capillaries, which then merge and exit as the efferent arteriole. And then we have the peritubular capillaries, which are important for reabsorption of filtered water, salt, and solutes. Now, today I will really be focusing on the glomerulus because diseases of the glomerulus constitute one of the most important problems in nephrology. So the glomerulus is composed of capillaries, as I just mentioned, and those capillaries are basically lined by a fenestrated endothelium. And then these endothelial cells then sit in a glomerular basement membrane. And on the other side of the basement membrane is the visceral epithelium, which have their foot processes on the basement membrane, and is very important in the size and charge selectivity for the filtration function of the kidney. You then have the parietal epithelium, which is in the Bowman's capsule, and they line the urinary space, where the plasma filtrate initially collects. So immune mechanisms underlie most forms of the glomerular disorders, and glomerular deposits of IgG, often with complement components, are found in the majority of individuals with glomerulonephritis. Now, the mechanisms of immune complexes formation really differs in terms of in situ. You can have in situ immune complex deposition, where antibodies recognize intrinsic tissue antigens in the glomerulus, such as the NC1 domain of type 4 collagen antigen, which is part of the glomerular basement membrane, and that can lead to anti-GBM nephritis, also Goodpasture syndrome. You can have antibodies that are generated to the PLA2R antigen, which is actually on the other side of the basement membrane, and that's in the podocyte. That leads to membranous glomerulopathy. You have antibodies that can bind to mesangial antigens. You also have antibodies that can react to planted antigens, which deposit in the capillary wall. In terms of, for example, circulating immune complexes that are formed in systemic diseases, example would be lupus. Those antigen-antibody complexes can also deposit in the glomerulus, and I wouldn't talk about these last two in the interest of time. So whether antibody actually generates an inflammatory response depends on where the antibody is located. If it's circulating immune complex, one important concept of the fenestrae in the glomerulus is that they're open fenestrae, so that the deposited immune complexes have access to circulating blood elements and leukocytes. So in the case, for example, in lupus, where you have circulating immune complexes deposited, you have subendothelial deposition, as well as in anti-GBM nephritis, where the antibody recognizes, as I mentioned, collagen type 4, you have now a very robust inflammatory response to those immune complexes. When you have, on the other hand, immune complexes that form outside of the basement membrane, such as in the podocyte, such as in membranous nephropathy, you do not have a inflammatory response. So glomerular inflammatory response usually entails hypercellularity, where you have leukocyte infiltration. You result in endothelial mesangial cell proliferation. You also have epithelial cells. This is more the parietal epithelial cell proliferation, which then gives, in combination with leukocytes, the formation of crescents in a glomerulus, which is very, very predictive of end organ, renal failure. You have occlusion of the capillary lumen that also causes some ischemia. Podocyte injury, which is also very bad because podocytes have very limited ability to regenerate. You then result in tubular interstitial inflammation and damage, chronic kidney disease with systemic manifestations. So there are basically two models of immune complex-mediated diseases in the mice that have been exploited by many investigators in the field. That the antiglomerular basement membrane nephritis, it's best actually to talk about nephrotoxic nephritis because there are additional antibodies besides just the glomerular basement membrane. But in any case, you have, when you take a naive mouse, you now give it a rabbit or sheep anti-GBM serum, you now see nice glomerular staining for the IgG, and within 14 to 21 days, the mice now develop huge leukocyte influx with neutrophils, macrophages, and T cells, hypercellularity, crescent formation that I just mentioned, massive proteinuria, and mortality in some cases. Now, this is what you would call the in situ. So this is a fixed antigen, which is a glomerular basement membrane to which antibody is generated. Here in spontaneous lupus, this has more a flavor of soluble immune complexes. Lupus-prone strains of mice exhibit a breakage of tolerance. You now get immune complex deposition. You'll see that this kind of deposition is very different in that it's sort of punctate in terms of, instead of the linear staining you see here with anti-GBM. Within 3 to 12 months, you also get a lot of similar responses of hypercellularity, neutrophils, macrophages, T cell influx, crescent formation, and proteinuria. Now, one of the very proximal events, of course, in inflammation in the glomerulus and elsewhere is the recruitment of leukocytes, which was of particular interest to my laboratory. The classical paradigm of leukocyte recruitment is the activation of the endothelium, then that leads to the leukocyte recruitment through the interactions of ligands expressed on the activated endothelial cells, such as selectins, with their counter receptors on the neutrophil. That leads then to firm adhesion and transmigration. And these, as I mentioned, selectins and integrins and ICAM and VCAM on the endothelial cells have been classically associated with leukocyte recruitment. However, when you've got immune complex deposition, we argued that there's probably additional receptors that are important in the actual leukocyte recruitment cascade. We hypothesized that Fc gamma receptors that are present on the leukocyte will directly be able to lead to leukocyte recruitment in the context of immune complex deposition. So now Take Suboy in the lab, so before I go on, Jeffrey Ravage's group had shown that if you knock out all Fc gamma receptors, that means if you use a gamma chain deficient mouse, These mice are protected in various models of immune complex-mediated disease, both in the kidney. They're protected from arthritis, glomerulonephritis, and many other immune complex-mediated reactions. What we did here is to actually express a uniquely human Fc gamma receptor selectively on neutrophils, and then cross those animals to gamma chain deficient mice to see whether now we were able to understand better what is the role of the Fc gamma receptor 2A on the neutrophil in neutrophil recruitment and the disease pathogenesis. So these sets of mice, the 2A transgenic on the gamma chain background, as well as the gamma chain knockout mice, were then subjected to anti-GBM nephritis. And as you can see here in the gamma chain knockout animals, these are the glomeruli you can see here in low power. And on higher power, everything looks pretty much pristine, pretty much untouched, and no neutrophil accumulation, just as Jeffrey Ravager's group had shown. However, when we express the 2A on the neutrophils alone, and this is not an overexpression system, this is physiological levels of 2A, we found that we had great tubular atrophy, and in the glomerulus, you saw the sort of occlusion of the capillary lumen and adhesion of the Bowman's capsule, and this was associated with significant neutrophil accumulation, suggesting to us that the Fc gamma 2A expression on neutrophils, selectively on neutrophils, can give you neutrophil recruitment and damage. We wanted to know more directly how the Fc gamma receptor may be actually allowing neutrophil accumulation. So we made preformed immune complexes in vitro, gave them intravenously, and within an hour, when you surgically exteriorize the cremaster, Tracy Stockel in my lab had shown many years ago that leads to immune complex deposition in the postcapillary venules due to changes in the permeability of those vessels. So you can see here nice immune complex deposition, and we then now look by intravital microscopy both in the gamma chain deficient mice, and this is a movie, and you can see there is some rolling, and that's due to, because exteriorization will cause the expression of P-selectin on endothelial cells, and you get pretty fast rollers there. Not much adhesion or transmigration. However, if you now have a mouse that is expressing the human Fc gamma receptor on the gamma chain background, I think you can all see that it's, well, there's a lot of slow rolling. There's rolling, but there's a lot of slow rolling. And let me see if I can, it's not buffering. Okay. There's a lot of slow rolling as well as adhesion and transmigration. So many people in the field have used knockout mouse models in these two models of disease, anti-GBM and lupus nephritis. And as you can see, many of them, these inflammatory molecules, whether it be surface receptors, complement adhesion molecules, are pathogenic in nephrotoxic nephritis, as well as spontaneous lupus nephritis. I think what's missing here is the ability to know on what cell types these receptors are important and how they may be actually influencing the leukocyte recruitment, which will require the generation of conditional knockout mice. It will also require the ability to do intravital microscopy directly in the kidney, in the glomerulus, because as is right now, as I showed you, I did in the cremaster, because that's very transparent and easy to analyze tissue. But if we really want to know leukocyte recruitment in the microvasculature, we need to do some intravital microscopy. This is very challenging because the glomerulus is about, the closest glomeruli are actually about 100 microns from the cortical surface, and it's in a very optically dense tissue. So using two-photon microscopy, others in the field, such as Fred Geisman's group in London, has shown that patrolling, and these are fluorophore-labeled monocytes, GFP-labeled monocytes, increase their dwell time in the vascular in response to chemokines generated by a local TLR7 ligand within 3 hours of applying the stimulus. And then Michael Hickey's group also last year from Australia suggested an increase in neutrophil retention and neutrophil-generated oxidants within the glomerulus occurs within 2 hours of injection of an anti-GBM antibody. So this work has certainly paved the way for us to start looking very critically at what are some of the molecular mechanisms of recruitment in the glomerulus. Now, we were interested really in end organ damage and leukocyte recruitment and some of the mechanisms for that. So instead of using the lupus-prone strains of mice, which has loss of tolerance and everything else, but then it takes a long time to develop, we decided to generate a model of passive transfer of human SLE serum in what we call genetically susceptible mice, and we show that that induces organ damage and a role for neutrophils. And I'll just show you 3 slides on this because this work was published. But for reasons that I won't go into, we hypothesized that if we express the human receptors, but then remove a receptor called MAC1 from the mice, we now may make this mouse susceptible to developing disease. And you can see here we introduced human SLE serum and then collected the urine and serum over several days. And you can see that the renal function is significantly increased in mice that are lacking the MAC1, but expressing the human receptors. Those are called non-susceptible and susceptible mice. And the susceptible animals had a lot of endocapillary proliferation as well as crescent formation, as is shown in this histological score. And then one point that I just want to make here is that the human IgG immune complex and complement deposition was very similar in the non-susceptible and susceptible animals, and yet only the susceptible animals that lacked MAC1 had neutrophil accumulation after some of the pathogenesis. So I think this is a very important and interesting point, and that I'll talk about in what we have the gap in knowledge right now. So we think that there's pathway 3 or some elements that are important. So you can have anti-nuclear autoantibody deposition, autoimmune response, but really there's something in the end organ susceptibility factors that allow neutrophil influx. So lessons learned, I think we have identified molecular and cellular mediators, renal injury following glomerular immune complex deposition, and intravital microscopy recently will allow us to get further in that area. There's a lot of gaps in knowledge, and I won't have time to go into it, but I think some of the important points are, what are the target organ susceptibility factors that dictate the extent of the immune response and damage incited by the deposited antibodies? And I'll go down a little bit here, use multiphoton intravital microscopy in the glomeruli to look at the temporal sequence of events in leukocyte-induced inflammation. And then some interesting points is, how does the glomerular inflammation lead to tubular interstitial damage, which is most predictive of progression to end-stage renal disease? And how does glomerular inflammation actually affect increased risk for atherosclerosis, which is a macrovascular disease, independent of traditional cardiovascular risk factors? Thank you.
Open to questions. We have time for a couple of questions.
Malik, Chicago. Tanya, very nice talk. So, one of the factors in permeability across the glomerular apparatus is the charge.
Yes.
So the charge of the matrix and charge is really critical. So how do you think charge modification regulates this process?
So that's very interesting, because the glomerulus is actually very anionic. And so if you're cationic and you're small molecular weight, you're able to go across the filtration barrier. But there are some diseases where you may expect that the glomerular basement membrane charge may actually change. It's very rich in heparin sulfates, for example, in the glomerulus, and those changes may occur during diseases, which may also allow more deposition of extrinsic, trapping of extrinsic antigens.
Hi, Jose Lopez from Seattle. Do you think that there might be a role in humans for the FC receptor gamma R2A and platelets, which the mice don't have?
Yes, I absolutely agree. Definitely, there's a role for that on platelets. And as you just mentioned, since mice don't have that, it's very difficult to evaluate. We had a neutrophil selective promoter that we used. So in our case, we did not have any 2A on the platelets. Yes.
Very quick question. Okay.
So, in your models, did you have a chance to look where the neutrophil extravasation actually occurs? Does it happen on the fenestrated areas or away from those?
We don't know yet, but hopefully we're going to get to it once we start doing some intravital. Yes.
Thank
you very much, Tanya, and thank you, Esser. It's my pleasure to introduce our next speaker, who is Gary Rosenberg from Neurology at the University of New Mexico, who's going to speak on biomarkers in Binswanger's disease, targeting small vessel inflammation for treatment. Gary?
I want to thank the organizers, Dr. Bossetti and Dr. Galis, one for putting this very interesting conference together, which is, I think, a critical stage, and also for inviting me. So dementia is, due to vascular cognitive impairment, is really one of the major health problems. And it's projected to increase greatly because of the confluence of increasing aging of the population and the increase in vascular risk factors. But in spite of its huge importance, we don't have any treatments for the vascular forms of dementia. And I think one of the major reasons for that is that we're not dealing with one disease. We're dealing with a heterogeneous group of diseases. And in all medicine, if you cannot define what you're treating, you're really unable to treat unless you have huge populations. So I think if we're going to get anywhere, we have to narrow this down. And the most common form of vascular cognitive impairment is the small vessel disease, and I'll try to convince you of that. And I'll also try to convince you that it's an inflammatory disease and not necessarily stroke. I'll give you some evidence for that. So how do we deal with that? Well, I've decided the only way is with biomarkers, and to have biomarkers that are more complex or complex enough to deal with the complexity of the pathological changes. But the big problem there is to verify a biomarker, you need an autopsy, which is not necessarily available in this group of patients. So we chose to collect a large number of biomarkers at the beginning of the illness and then follow patients for multiple years to try to get as close as we could to the best clinical diagnosis. And I'm going to show you that that approach at least narrows it down to multiple subgroups, some of which I think are going to be amenable to treatment, targeted treatment, fairly soon. So what I'm going to do is describe a set of validated biomarkers that identify the form of vascular cognitive impairment that's due to small vessel disease. And these biomarkers are collected as the patients come into the study. We take everybody with suspected vascular cognitive impairment. They usually have an MRI that shows the quote-unquote white matter disease, which I'll show you is really not a very good marker. And then we follow them to try to get a more accurate diagnosis. And I think the data that we're collecting and that I'm going to show you is going to show that the group that we want to target is a group that has an inflammatory process, which is disrupting the blood-brain barrier. And because of that, we have not only a marker to follow, but we also potentially have some targets for treatment. So what is this thing that we call vascular cognitive impairment? It's clearly a heterogeneous process. Large strokes, embolic strokes are going to cause it. Thrombotic strokes are going to cause it. But the major form is this small vessel disease related mainly to hypertension. And there are rare forms. You heard about CADASIL, 1 in 1,000. Hypertension is everybody. So this is the major form. And because it's been lumped all into one form, we've never been able to get a focused treatment going. And I think we fell behind. 20-some years ago, Alzheimer's group got all together. They have 38 centers. They forgot us. And so very little research was actually done on the vascular risk factors. And I think their approach is the appropriate one. So I'm going to talk about two forms of this disease, or basically one form of this disease. So you have large vessels, multiple infarcts, and lacunar strokes in the basal ganglia, or you can have small vessel disease, which leads to white matter change. And the white matter change, I'm going to use the term Binswanger's disease, and I'll try to argue that that's the best way to do that. If you deal with large vessels, you have a sporadic course. Clinical studies are going to be very difficult because you don't have a predictable progressive process. Whereas if you deal with white matter injury, you have a potential target. This is one of the only studies, there's very little epidemiology. This was a wonderful study out of Lund where they looked at vascular dementia autopsies. If they were pure vascular dementia, most of them were small vessel. If they were multiple types, and as you know, when people die, they have all sorts of pathology, small vessel was again the number one. So small vessel disease is arteriolosclerosis, hypertension, diabetes. There's progressive growth of the white matter lesions, executive dysfunction, and gait imbalance. It's a classic patient. We've now followed a bunch of these patients. And I'll show you that this is an inflammatory process. Otto Binswanger described this disease in 1892. He was born in 1852. He was a student of Alzheimer. Actually, Alzheimer named this disease Binswanger's disease. It was probably syphilitic white matter disease due to vascular disease. This is the classic picture. Unfortunately, 20% of normal individuals can have this picture when they're in their 70s and 80s. And we've done that study in a very healthy group of people. You cannot use this as a diagnosis. I get sent patients all the time saying, I have Binswanger's disease. They're perfectly normal. Some of them. The question is to weed out the ones that are not. And the ones, and we had, this is actually a very advanced brain from the Radcliffe Infirmary of a patient when they weren't treating hypertension very well. And you can see gliosis, huge gliosis. This is dead tissue. And the demyelination spares this U fiber. And that's the key to this not being a stroke. If it were a stroke, cortex, white matter would be involved. But this is the classic pattern. And what's the pathology? This is the pathology. This is the most common form of arteriolosclerosis, fibrohyalinosis, all kinds of different terms. And there's inflammation around these blood vessels. These are PG1. I had the opportunity to go over to Radcliffe and work with Margaret Asiri, who had a large number of vascular dementia patients, and we stained them all for metalloproteinases a few years ago. And here you see the macrophage, and this is MMP2 in the astrocyte, and this is the GFAP. We've recently done dual labeling now with immunofluorescence, so we know that these are astrocytes. But more interesting, we found stromatolysine, MMP3, in the macrophages. So the metalloproteinases open the blood-brain barrier and they break down myelin. So they're the perfect molecule to cause this process. So what are these metalloproteinases? These are an interesting group of 26 molecules. They were discovered for cancer actually by Lance Leota, who gave me the original type 4 collagenase to use. It's a signal peptide covering the zinc active site, and depending on what things you hang on the end, you give it different names. The simplest one is matrolysine. It's only the zinc. The one that is most studied because it can be measured is the metalloproteinase 2 and 9 gelatinases, and they attach, they have a hinge here, and they're a fibronectin binding site, so they go to the basement membrane. And then there's a transmembrane type. The neurovascular unit is the site of action of these molecules. This is a MMP2 immunostain around an astrocyte. And you can see that it's in the foot process, and it wraps around the foot process. It modulates your barrier all the time. It's not open or closed, it's modulated. And some years ago, we developed this model In a normal person, metalloproteinase bound to the astrocytic foot process is constantly being activated. The MMP2 is constantly being activated, breaking down the basal lamina, restructuring it. That's a normal process. But when you have hypoxia, you activate NF-kappa B and IL-1 beta, and you turn on the MMP9 and MMP3, which really are very toxic. And I won't go into how we work this out. We have an animal model, spontaneously hypertensive, stroke-prone rat, which develops everything that the Binswanger patient looks like, but we accelerate it. And what happens is hypertension leads to hypoxia, and we've actually measured this with electron paramagnetic resonance in the animals. HIF gets turned on, the FUR gene turns on the MMP membrane type, making the active MMP2, attacks the blood-brain barrier, causes edema, demyelination, white matter damage, and then there's a repair process. So now I want to show you how we've used this to develop biomarkers. The first biomarker that we looked at were the metalloproteinases in the spinal fluid, and we now use a thing called the MMP2 index, which is indexing the MMP in the spinal fluid to the MMP in the blood, and you need to do that to tell whether it's endogenous or exogenous. But you can see in the other name for the Binswanger's disease is subcortical ischemic vascular disease. I flip between those two depending on which audience I'm talking to. We found that this goes down, and the way that we've correlated that is with the albumin in the spinal fluid. Albumin is made by the liver. It's never in the spinal fluid except in very small amounts. And as the concentration of albumin goes up, the MMP2 index goes down. So that's one of our biomarkers. The other important one is to measure the blood-brain barrier permeability. And I think this has been the major advance that we've been able to do. The early studies, mainly by Anders Wallen and his group in Gothenburg, showed that albumin was elevated in vascular dementia patients. And then there was a series of patients with MRI, one from Japan, one from Wardlow's group in Edinburgh, and then from Marcus's group, showing the blood-brain barrier was abnormal. But these were qualitative. These were not actually measuring. And we developed in humans the dynamic contrast enhanced MRI with gadolinium. It was first done in animals by Ewing, and actually Sahit Tahiri, who's here in the audience, was a postdoc and worked this out. These are rapid T1 measurements. Every 3 minutes we take a T1 measurement for 21 minutes, and we get a PATLAC graphical plot, which people who work on the blood-brain barrier will appreciate, and we can calculate an actual K for every single pixel. We worked it out in controls first. This is a work that we published a couple years ago with Dr. Tahiri. The histograms show the permeability spread in the brain of a normal person, and we picked a number in 20 patients. And these are the normal scans, and these are scans of patients with Binswanger's disease, and you can see there's elevated the shift of the permeability curve in the VCI patients. So that was work we did at 1.5 T, and I'm going to show you some unpublished data now that we've now collected in about 30 patients with 3 T. It's really very much more interesting. This is the MRI of one of our typical Binswanger patients. I want you to note here that this is the periventricular. This is the subcortical. Periventricular changes are normal. Normal aging gives you periventricular changes. You can't use those as pathological changes. The real changes are out here in the subcortical area. And the other thing I want you to note, this is very symmetric. This is not a stroke, stroke, stroke again. And this is the permeability map from that patient. And you can see that the permeability is increased. Here is a control group, 6 controls. Some of them even have white matter disease here outlined here. And these are 6 patients. And the permeability is around the edge of the white matter lesion. So it's sort of an inflammation around the edge. These are the biomarkers we use: albumin index. We measure the spectroscopy to look for ischemic tissue. We measure the MMP2 index and the permeability. And when we get all of these together, we say the patient has the Binswanger form of the disease. And so far, in our first 61 patients, about a third have that type. A lot of them have what we call leukoariosis. We don't know exactly what that is, large vessel disease. And we've now compared that with our a score that we make with 10 different factors, including the Alzheimer factors, and we found that it does correlate well with our final diagnoses. And this is total permeability matched up to the score, and you can see in our patients the permeability is open. So we now are trying to find some outcome measures to use to test drugs. There's functional, blood-brain barrier. There's structural, diffusion tensor imaging, which is much better than just the FLAIR MRI. The FLAIR MRI is not a good measure. And then the behavioral and the time frame is different. So in conclusion, we think that biomarkers based on the pathophysiology can select a more homogeneous subpopulation of small vessel disease patients with this inflammatory form, which we'll call Binswanger's disease, for targeted therapy. And it will now be possible, I think, to find the group of biomarkers that will help us tailor or target our treatments. And the gaps, of course, are to validate this biomarker approach in a much larger population, to identify some outcome measures, and then the major thing will be to develop a group of anti-inflammatory drugs that can be tested. And as you know, these kinds of studies, if I can get this slide, oops, where'd it go? I thank NIH for funding for both our Clinical Translational Science Center and for R01 grants. The group that has been working on this is my clinical group. The early collaborators were Lance Liotta, Stetler Stevenson, John Shaw did the... So anyway, thank you very much.
Thank you very much, Gary. We have about 3.5 minutes for questions.
Hi, very nice talk. Radostan from Dartmouth. Did you have a chance to carry out any structural studies to see what's going on at the level of the endothelium? What's the basis of this permeability, enhanced permeability? Are the junctions open? Are there structures appearing there?
Yeah, I didn't have time. We have an animal model, and I didn't, in the 15 minutes, I couldn't put them both in. But the animal model, which we've published, does have changes in the tight junctions. I don't know if that's been published yet, but there are clearly changes in the tight junctions. We're studying angiogenesis, and the animal model is a hypoxia-induced blood-brain barrier opening through MMP9 primarily in the rat. So there are structural changes in the endothelial cell, very significant.
Thank you. Please.
Howard Crystal, thank you, Gary. So that subject that had a lot of abnormality on FLAIR that did not have Binswanger's, I assume they did not have any abnormalities in blood-brain barrier, and what cause, what are we looking at?
Well, that's, of course, that's the most important question. Well, anyway, we published the first CT scan of Binswanger's disease some years ago. And then there was an editorial in Lancet that there's an epidemic of Binswanger's disease. This was a disease I'd never heard of in medical school. So I knew they were wrong, and we had an MRI machine that we could use anytime we wanted. It was a research machine. And we had 100 normal people, age 65 and over, and I scanned 100 of them. There were 500 in the group, up to 90. 30% had white matter changes. And we found 7 that had complete white matter changes. We did spectroscopy on those 7. They were perfectly normal. They were in their 80s and 90s. Nothing. They had no ankle jerks. That was about it. So I think this is a potentially, this is a normal change. You know, just like in the lung, you see some fibrosis, you see stiffening of arteries, aging. I think people who are calling these strokes little strokes are misleading us.
Okay. Thank you. David, a short question, please.
Yeah, so you showed data from this dynamic MRI technique where you had gadolinium leaking. I just was curious, if you looked with diffusion tensor imaging of something of that ilk, do you actually see at the same time or earlier or later sort of a change in the white matter?
Yeah. That's what we're doing. We work at this amazing institute, the Mind Institute, connected with the university, and we have all that data. And my imaging people are telling me we're seeing changes, and I think other people have said this is it's much more accurate to look at diffusion tensor imaging. And I think once we get the diffusion tensor tractography,
yeah,
we're going to see some amazing things, because this is basically a disconnection syndrome. And if we can understand, I mean, the brain is put together by connecting white matter to the cortex. And as people get this disease, they break down the white matter. And so we'll start seeing these patterns once we have sophisticated, and the center has all that ability, free connectivity, all that sort of stuff.
Okay.
But that's going to be another 6 months.
All right.
I'm going to have to call a halt, because to be on time for lunch in the next session, maybe the last two questions you can come up and talk to Gary.
So quick question, how is this related to multiple sclerosis? You're talking about, demyelination, inflammation, and, flare, hyperintensity.
Yeah, multiple sclerosis. Can you handle that in 4 sentences?
Well, actually these lesions, which are around the edge of the white matter damage, look like progressive chronic multiple sclerosis. And I used to run the MS clinic, and I would get sent people who were in their 50s, and they said they have MS, and it turned out to have vascular disease. That's how, one of the ways I got interested. So I think from a flare, it's very hard. But there are characteristic features of MS in terms of locations that are different than these patterns.
Okay, that's 4 sentences. Francesca, did you want to say something about that?
Thank you all. I think we'll take a break for lunch. If we can come back by 1:35, it would be great, so we can get started and hopefully finish on time. Thank you very much.
It's my pleasure to introduce Maiken Nedergaard from the University of Rochester, who's going to speak on her work on the lymphatic system, long-term effects of lacunar strokes. Maiken?
Yes, thank you. And if I can get full dark slide. As everybody else, I just first want to thank the people who collected the data and my collaborators. So in this case here, it's particular 2 postdocs, Jeff Liff and Lulucy, and my collaborators that really have expanded the work has been Dr. Benveniste, Dr. Nicholson, and Steve Goldman. So there is one very peculiar feature missing in the central nervous system. The brain and spinal cord are completely lacking a lymphatic system. The lymphatic system would, in all other organs, basically be the second circulation, and the density of the lymph vessel would reflect the metabolic demand of the tissue. So really high energy demanding organs would have lots of lymph vessels, and others would have less. So being a neuroscience, it's kind of peculiar, because the brain have one of the highest metabolic state, but we don't have lymph vessels. We all know that the lymph vessels play an important role in immune function, but lymph vessels are also engineered to transport proteins that are present in between cells in the interstitial space back to the general circulation. And if you have loss of lymph vessel, you get edema because you have protein accumulation in the interstitial space. So this is basically how we started out. We asked, why does the brain not have a lymphatic system? But the brain has something no other organ have, to my knowledge, and that is that it has a very well-defined perivascular space. So the perivascular space is formed, as in all other organs, by the endothelial cell on the inner membrane, but on the outer membrane we have perivascular astrocytic end feet that totally plaster along the entire vasculature in the cerebrospinal system. So we have 99.7% of the vasculature covered by vascular infeed by astrocyte. What does that do? It creates a space that's highly defined, and it's basically a donut-shaped space that surrounds all the vessels, the perivascular space. And this has another feature that has been puzzling people in the glia field a lot, and that is that they have, at the inner membrane of the astrocyte infeed, you have expression of aquaporin 4, a water channel. There's actually so much aquaporin 4 expressed here at the vascular infeed that it fills 50% of the surface of the infeed facing the endothelial cell. So why is that strange? It's strange because brain endothelial cell express absolutely no water channels. So you have a closed faucet facing an open faucet, and it hasn't really been explained what are the function of that. Smart people many years ago have suggested that the cerebrospinal fluid have a role that is almost like the lymphatic system. And the idea has been that if you are a protein like beta amyloid present in the interstitial space, you can actually diffuse out to the cerebrospinal fluid, and then you can be transported away from the brain. But other people have calculated that this is probably not a realistic pathway for removal of waste product, because it would take amyloid 100 hours to diffuse just 1 centimeter. It's far too slow to really matter. But one thing you often forget about, and that is that brain is not just about diffusion, which is a very slow process, and it's driven basically by random events, and it matters how big a molecule you are. The brain also have a lot of pulsatility, because we have tremendous big artery feeding into the brain, going into the brain and surrounding it. And that set up a dynamic movement of the entire brain. And one thing that's kind of interesting is that as, for example, the arteries dip down into cortex, they actually connect directly with the cerebrospinal fluid. So if you look here, let's see here, what way should I go? So if you look here, so this space here, the perivascular space, is directly linked to the cerebrospinal fluid and contained in the upper part cerebrospinal fluid. So one of my collaborators, Helene Benveniste, she's asked, can we just image cerebrospinal fluid fluxes by injecting contrast agent into cisterna magna, and then image the fluxes of cerebrospinal fluid, testing the idea that we have very dynamic fluxes that not just follow the textbook on cerebrospinal fluid movement, but actually you have a recirculation by the perivascular space back into brain. And this is one of her movies. We inject contrast agent here just below cerebellum, and the movie is 1 hour long, but I hope you would agree that it's almost over in 5 minutes. So you see the contrast agent passing along the vessels here, and you actually see it exit in the olfactory bulb already a few minutes after we have injected it. So we have some very dynamic fluxes of cerebrospinal fluid, which we postulate is driven by the vasculature, and especially the pulsation of the arterial wall. To study this in more detail, to see what's going on, we looked at the perivascular space. And for this purpose, we use 2-photon imaging, and basically what we do is we train the mice to sit still on a microscope stage. Then we have implanted a little cannula into cisterna magna, so we're repeating Helene's experiment, but injecting fluorescence tracer instead of contrast agent. We would record EEG to measure the state of brain activation, and we would just basically use the 2-photon excitation to follow the influx of cerebrospinal fluid. And the big question is, if it occurs, how fast is it? How dynamic is it? Does it really play a role in normal brain function? So in this case here, we are looking 180 micrometer below the surface, and this is in barrel cortex, and we have filled the vasculature with a fluorescence tracer in plasma that's red. And this is just to identify the penetrating artery, and all the arrows point to penetrating artery, because our hypothesis is that cerebrospinal fluid would be pumped down by the pulsatility of the arterial wall. Remember, pulsatility is not just standing like this. This is actually wave that derives from the heart and travel all the way into the cortex. So when we do that, you can see, and this is a 15 minutes movie, so 15 minutes. I hope you agree that you see a very rapid influx. First along the PL artery, you see in a negative outline here, and then you see it dipping into the entire cortex, but first and primarily along the penetrating artery. Let me just show it again. So these events occur within 15 minutes. You have basically a complete exchange of cerebrospinal fluid with interstitial fluid at this time point. This movement has to cost the brain a lot of energy. It's similar to kidney. It takes a tremendous energy to pump fluid around because it's not just a matter of water, it's also a matter of transport of ions by transporters and channels. So we ask the question, is this transport of cerebrospinal fluid is dependent on the state of brain activation. And in order to do that, we basically trained the mice so well we could have them sleeping and we could have them awake on the microscope stage. And we could inject two different tracers in the same animal, image exactly the same field, and look at, do more tracers go in when they're awake versus sleep or opposite? To validate the different brain states, we basically measure EEG in the opposite hemisphere, and we validate that we either take a awake mice and we anesthetize it here with ketamine xylazine, and we validate this change in brain activity by measuring what's called the relative power of the delta wave, the slow delta wave. Similar, we can start out with a sleeping mouse, and we can wake it up. How do we wake it up? We simply have a young investigator sitting at the microscope, and she would put her hand in under the curtain. This is all done in dark, and wake it up by moving the tail. Mice hate to get their tail touched. And this is Lucy. And how does it look like? So if you look here, this is exactly the same field. In a mouse, we started out with awake, and then we give ketamine xylazine anesthesia. So we injected a green tracer, about 3 kilodalton in the awake state. The same tracer tagged with a red label, Texas red, also 3 kilodalton. And again, it's 15 minutes. So the blue is the vasculature, just to have an outline. And you can see, again, the penetrating artery. First is outline, but already within 5 to 10 minutes, you have a complete exchange in ketamine xylazine anesthesia with in between cerebrospinal fluid. You see here, you can see how it's coming in here. And you see the penetrating artery here. In contrast, in the awake mice, we see almost no influx. Only at the surface vessel, we see a little bit of the tracer coming in here. So based on these studies and several other set of analysis based on reporter mice where we can separate arteries from veins in the living animal, we developed this concept of the glymphatic system. And the glymphatic system is basically that brain pulsatility drive in the Virchow-Robin space, cerebrospinal fluid in. And you can look at the perivascular space as a highway that has low resistance toward the movement of cerebrospinal fluid in. There's no cellular element, or very few cellular element, compared to the very compact no pill outside the perivascular space. Then the cerebrospinal fluid would flow into the brain parenchyma. It would meet a tremendous resistance because of the complexity of the brain. In order to facilitate the movement into the brain, astrocyte and the astrocytic end feed express a very high amount of the water channel, again aquaporin 4, to basically direct the flow into the brain. It would then literally swipe any solutes, and this is including amyloid beta, out of the brain. As the convective flow passes to the brain tissue, it would collect around the deep vein. It would exit the brain and basically empty out in the cervical lymph vessels, and by that way reach the general circulation. So is this important? I think it's important because we show that 65% of amyloid in young animals are removed by this pathway. So the brain basically exports its waste in our mind. So it's a macroscopic clearance system. Another thing that's probably as important for this audience is that it's long been known that perivascular diseases or small vessel diseases in brain is linked to an enlargement of the perivascular space. It has been somewhat of a mystery why you have specific atrophy in these areas, because you did not really have a function before of the perivascular space. What we are stating is that the perivascular space play a very important role in what we call the glymphatic system. So we call it the glymphatic system because it's glia-driven, it depends on glia water channel, and it has functions similar to the lymphatic system in other organs. But we are claiming now that these fluxes in the perivascular space have an important role in clearance of molecules present in between the cell, in between the interstitial space. This is interesting because the field of neurodegeneration have basically established very systematic over the last 5 years that all the proteins that have been linked to neurodegeneration, so this is synuclein, it's tau, it's amyloid, etc., that although most of these proteins are used as intracellular cytosolic proteins, they're also present in the interstitial space, and they're thereby available for removal by the glymphatic system. We can ask a couple of other questions about the system. For example, what happened in aging? And for this study here, which is in press, we injected cerebrospinal fluid tracers, and we prepared 30 minutes later brain slices to look at the distribution, basically, of cerebrospinal fluid influx into the brain. And we injected a small and a big tracer, and this is how it looks in a young mice. If you look in the old mice, it's very significantly reduced. Especially if you plot it as a function of aging, you see for the small tracer, you have a reduction of glymphatic influx. That is 85% reduction in glymphatic influx in old mice compared to young mice. We have a gender difference or sex difference, such that female mice have about 30% reduced glymphatic influxes in the young age, but the difference disappears in aging. What triggers these age-dependent suppression of glymphatic fluxes? I believe it's many different things, but one thing that probably have not really been discussed or studied much is that with aging you have the stiffening of the vessel wall. Many people have been interested in how the pressure gap between systolic and diastolic pressure changes as a function of a stiffening arterial wall. But what also happens is that the elasticity of the vessel wall decreases, and thereby there would be less pressure to drive perivascular fluxes in. So we can actually measure that by using 2-photon line scanning, and we can show that in aging you have very much reduced movement or pulsatility of the vessel wall. Finally, I just want to show new unpublished studies where we have adapted a model from Mike Moskowitz's group where we induce small infarcts in the brain. We inject cholesterol beads into mice, and we produce pretty much incomplete infarction. This infarct can be recognized 1 week later by the infiltration of microglia cells in the small incomplete lesion. The small lesions that are labeled here in red are surrounded by huge area of reactive gliosis detected by increase in GFAP. So our question was, if we look at glymphatic influx in a mouse like that, would you have a reduction in influx just in the lesion, or would it include this big area surrounding the small lesion? I hope you agree with me that we have a reduction in influx of cerebrospinal fluid detected as a green tracer that include most of the same hemisphere. So it's a whole hemisphere that's affected and have suppressed glymphatic influx. So the summary is just that I think we should start to take cerebrospinal fluid fluxes more serious. We should look at the perivascular space as an important unit for both discovery of disease mechanism, but possible also for treatment, and glia cells certainly play a role in that, and so does sleep. The lacks in the knowledge is that we basically know very little about this. We need first and foremost to show that this system also is functioning in human brain, and if it is important in removal of amyloid, we need a diagnostic test of it.
Thank you very much, Maiken. a few minutes to entertain some questions. I have a basic question, and that is, so there is no interchange of the CSF at the capillary level, presumably because the Virchow-Froben space doesn't exist that is fused, is that right?
There seems to be, it depends on the tracer size.
Depends on the tracer size.
Large tracers would not reach the capillary.
Okay.
So,
good. Gary?
Yeah, the spinal fluid is actually made by the capillary.
in part, yeah.
60% in a monkey comes from the capillary, and it's released into the CSF through the white matter, where there is white matter in larger animals, and drained out through the lymphatics. But the other thing that happens, so my question is, that could be one site where the reduction is going on with the change in the capillary. The other possibility is that when you absorb the spinal fluid, it goes through the arachnoid granulation. And if you study, and people don't do this anymore, but there was a thing called cisternography, where you would put a tracer into the CSF and follow its flow out of the brain, and it would get hung up in the older patients and stay for a longer period of time. So I guess my question is, you have a source in the capillary, you have a sink in the arachnoid granulation. Can you
so the data I showed here was only cerebrospinal fluid reflow into the brain because we inject into cisterna magna. I did not show data looking at clearance of the fluid from the brain. And you're right that the clearance suggests there's another source, which most likely are the capillary. Yeah. So there's two sources of influx, one exit of it.
And maybe in the aging animals, too, you may have a slow absorption through
all
the
that
arachnoid
is up
granulation.
for defining, and I totally agree. So of course it's complex. It's all complex.
Yeah, thank you very much for that presentation. I haven't thought about this at all, but I was very fascinated with the increase in the circulation during phases of sleep. And I would like for you to just comment on it, whether or not as you go from fast wave to slow wave, is there a pulsicity in the pulsicity in terms of the perivascular circulation? And, you know, this is sort of carrying it to a functional parameter. If individuals are sleep deprived, then you don't have this sort of recirculation of the cerebrospinal fluid that would affect cognitive function or something. I mean, I'm sitting here thinking that's really interesting and how important quality sleep is for the health of the brain. So it's more of a comment, but I would really like you to speculate on this pulsicity of going in and out of fast slow wave sleep.
We actually postulated when we published this work that the purpose of sleep was to clear interstitial protein, and you need to go in this phase where glial cell changes from being supportive of synaptic function and really ensuring that synaptic function is precise in time and space to go to a syncytium-like function, where they might supported by the delta waves, which slowly moving waves that actually follow the glymphatic fluxes. Yeah. They come from frontal and go back and inward, exactly like the glymphatic, that you go into the mode of cleaning your brain. And this is why any animal, you know, and human that die if you sleep deprive them long enough. The other interesting aspect of it is that all cognitive decline in aging and different dementia are linked to loss of sleep quality, fragmented sleep, less deep sleep, and the interconnection by those, what is the chicken and the egg, I think is a big outstanding question. And I think many groups are studying it very well now in the sleep field. And there are pretty good, I think, clinical studies suggesting if you report that you're a poor sleeper in middle age, you're more demented when you reach 75, age of 75 years old.
We have time for, on that thought, we have time for one more question, a very short one, please.
Mike, and I'm just curious about conditions that change the level of alkaporin 4. And I realize it's bulk flow, but couldn't that slow?
So sleep increases the expression of aquaporin 4, so there are studies suggesting that. I don't think aquaporin 4 is a driver of it. It just facilitates, it reduces the resistance towards the movement of the cerebrospinal fluid.
Thank you very much, Mike.
Thank you. Thank you.
I'd like to introduce our next speaker, Dr. Isabella Brownbach from University of Iowa. Can you hear me now? No.
I think they turned it off.
Maybe it's off. Right. I'd like to introduce our next speaker, Dr. Isabella Grambach from University of Iowa. The title of her talk is Calmodulin Kinase as Nodal Integrator of Calcium and ROS-Dependent Signaling in Blood Vessels.
Thank you very much for inviting a practicing cardiologist / large blood vessel cardiologist or blood vascular biologist to this meeting. Now, to make matters worse from your perspective, I received an email about a week ago which said, could you please also talk about redox signaling in cardiac myocytes? So instead of trying to take you through redox signaling in vascular cells and cardiac myocytes, I decided to concentrate on a particular aspect, and I will try to integrate the findings and highlight the opportunities and challenges of the field. Vascular disease, heart failure, arrhythmias, cancer, and asthma. What do all these diseases have in common? I'm sure you've heard this many times. Excessive oxidative stress. And this concept has been around for long enough so that there are now pills, berries, soft drinks, even facial creams with antioxidants which promise to keep you healthy and make you look better, and they lead to a perfect world. Now, the bad news that I have for you is that antioxidant therapies in this global generalized fashion have in clinical trials mainly failed. And that is probably because concise redox responsive disease pathways have remained elusive. Redox signaling. What is redox signaling? Well, it just means that particular signaling pathways can be activated or shut off by electron transfer events such as oxidation or reduction. Now, if you find this too theoretical, I think a way how many of us think about this, there's a disease-initiating event, hypertension, hyperglycemia, which leads to increased ROS production, and then to ROS-sensing, disease-inducing signaling mechanisms leading to vascular disease. But to me, this is more like a big black box. And the reasons for this are somewhat elusive, but some aspects of why antioxidant therapies have failed are that ROS isn't produced everywhere in the cell and at all times. It's really a localized event, and it's a temporarily localized event as well. So ROS are mainly produced in caveolae lipid rafts, in endosomes, in mitochondria. And since many reactive oxygen species have a very short half-life, it's at least interesting to speculate that their main effect will be on the neighboring signaling pathways and structural proteins. So what certainly does not happen, as is depicted in this cute and slightly fuzzy slide, is that reactive oxygen species shown in red are just swimming everywhere in the cytosol and they're eaten up by antioxidants. So, one way to look at redox signaling is to also step out of the limitations of just looking at one pathway and another pathway, which is what many signaling people like to do, but to think about how to integrate signaling pathways and see how they intersect and may, in combination, lead to disease-initiating events. And as you all know, calcium is one of the most important secondary messengers in the cell, and there are numerous ways to see how calcium signaling and ROS signaling may interact. For example, there is good evidence that numerous calcium channels, for example, the L-type calcium channel, may be regulated by increased ROS production. On the other hand, NADPH oxidases can be, for example, NOx5 directly activated in a calcium-dependent fashion. NADPH oxidase subunit P47, for example, is phosphorylated through PKC, which is calcium-dependent. Calcium is taken up into the sarcoplasmic reticulum through SERCA, and there are numerous papers at least suggesting that ROS-dependent events modulate SERCA function. the ryanodine receptor and the IP3 receptor are responsible for releasing calcium for the sarcoplasmic reticulum. And again, there are numerous papers suggesting that superoxide and hydrogen peroxide affect their activity. Lastly, mitochondria are a significant source of intercellular ROS production, and they're also one of the greatest calcium stores. So there are numerous opportunities for calcium and ROS signaling to interact. And there are a number of fairly well-defined effectors in this regard. PKC signaling certainly is a science in itself. Protein tyrosine phosphatases and ChemK2 have emerged as effectors which may integrate calcium and ROS signaling. ChemK2, of a calcium and calmodulin-dependent kinase 2, is actually strongly expressed in numerous cell types, and which also includes the vasculature. And I apologize for including a slide of the aorta. So this is ChemK2 expression, and as you can tell, there's robust expression in smooth muscle cells, in the endothelium, is probably best shown here, and also in cells in the adventitia. Now, what makes ChemK2 interesting in this particular context is that it can be activated through calcium, leading to autophosphorylation at threonine 287, and based on data which is fairly old by now and very, very well established, ChemK2 for the longest time has been thought to be a decoder of intracellular calcium events. Now, it's been known for a number of years now that in addition to activation through autophosphorylation, ChemK2 can be oxidized at 2-methionine residues in the autoregulatory domain, leading to sustained increased activity. In smooth muscle cells, which is the main focus of my lab, we and others have shown that ChemK2 signaling affects calcium homeostasis and has some impact on smooth muscle contraction and blood pressure, hypertrophy through calcium-dependent signaling pathways, inflammation, migration, proliferation, and apoptosis. And the vascular biologists in the audience will, of course, think of migration and apoptosis as ROS-dependent events. And many of the findings on this slide were really made before appreciating the function of chem kinase in redox signaling. Since I was asked to also speak about cardiac myocytes, here one slide on ChemK2 function in cardiac myocytes. There are numerous publications, more than a slide could fill, which have described seminal findings about chem kinase in cardiac myocytes, which include hypertrophy, similarly to calcium-dependent signaling events, electric remodeling through chem kinase phosphorylation of, for example, the L-type calcium channel of the ryanodine receptor, and events surrounding inflammation and apoptosis. And there is now equally good data over the last 10 years that chem kinase is activated through oxidation of these 2-methionine residues in disease states or under treatment conditions which are known to increase intercellular ROS production, for example, angiotensin infusion. Now, we started out using an antibody which was given to us by a long-term collaborator, Dr. Anderson, who's now at Johns Hopkins, and this antibody fairly reliably detects oxidized chem kinase, but does not detect non-oxidized chem kinase. So we were, for example, able to identify oxidized chem kinase in atherosclerotic plaques, but really didn't see much evidence for this in blood vessels of so-called normal patients. In diabetic retinopathy, we detect chem kinase in the endothelium of capillaries in the inner plexiform layer. However, the oxidized form was only present in patients with diabetes. Similarly good is the data on OXChemK2 as a correlate of high ROS disease states in humans in myocardium. Here's an example, the recent publication on atrial fibrillation. So this is human tissue from the right atrial appendage showing significantly more oxidized chem kinase in patients with atrial fibrillation versus sinus rhythm. And the overall expression of chem kinase was not different between the two groups. On the right, an example of a heart failure model. So this is a Western blot, again using the same antibody, showing higher levels of oxidized chem kinase in diabetic patients after MI versus non-diabetic patients. Now, an antibody is only as good as it is, and not surprisingly, for at least one project, we were asked by the reviewers to prove that there is that oxidation of this site occurs in vivo, at least in a high ROS disease state. And so we embarked on a journey of mass spec experiments where we first made a number of peptides which correspond to the autoregulatory domain of chem kinase, and we made different peptides which are oxidized and non-oxidized, and we were actually very happy to find out that an elution pattern which corresponds to the oxidized form of peptides containing this oxidation site were only present in a high ROS disease model, but not seen at baseline. So one of the difficult questions in redox signaling is, does this modification mean anything? So is inhibiting oxidative chem kinase activation sufficient to prevent vascular pathology? In order to show this, we used a mouse model in which these two oxidation sites at methionine 280, 182 were mutated to valines. And I'm going to refer to this model as the M2V or MMVV model. And indeed, we were able to show in different scenarios that just point mutating these two sites is sufficient to affect vascular pathology. So this is a very simple experiments done by a student in the lab who isolated smooth muscle cells and performed apoptosis assay in response to hydrogen peroxide in migration assays, and she found that the MM to VV mutation significantly decreased the propensity of the cells to become apoptotic or to migrate. We used this system in vivo in the large blood vessel biologists' model of arteriogenesis. So this is a model in which very simply the left carotid artery is ligated and the right carotid artery undergoes eutrophic outward remodeling. And while this was definitely present in wild-type mice, in mice which were lacking chem kinase delta, as well as in our chem kinase MM2VV model lacking the oxidative phosphorylation sites, this did not occur. So there is equally convincing data from myocardium. So this is an atrial fibrillation model which is driven by infusion of angiotensin by mini pump, and when the mice undergo an atrial pacing algorithm, and as you can see here, wild-type mice which were angiotensin infused did develop features which remind of human atrial fibrillation about 60% of the time, whereas the angiotensin-infused mice with this MM2VV mutation did not. Similarly to mice lacking NADPH oxidase subunit P47. So this story led to a concept in which angiotensin induces ROS production, which activates chem kinase for oxidation, and oxidized chem kinase then phosphorylates the ryanodine receptor, leading to calcium leakage and a higher propensity to develop atrial fibrillation. So lastly, this is unfortunately very busy. I'm just going to highlight two particular mouse strains. So these are mice which were made diabetic by streptozotocin injection and then underwent ligation of the left anterior descendant artery to induce a form of diabetic cardiomyopathy. And the survival of the wild-type mice was actually pretty dismal. However, this was significantly improved in the mouse model with the MM to VV mutation, shown in blue, as well as other mouse models in which chem kinase was lacking. So one area of surprise, and which has been actually reproduced by other groups as well, is that this is actually not a one-way street. So we didn't only find that ROS activated chem kinase, we also found evidence in different models, both in vivo and in vitro, that that chem kinase also regulates ROS production. So just very simple examples. So these are again the smooth muscle cells from these MM to VV mice versus wild types. And in response to a cytokine mix, they produce significantly less ROS than the wild type mice. So the lessons that my lab and our collaborators have learned from these exercises is that it is possible to identify single redox signaling pathways, and it's really key to develop relevant tools like an antibody which reliably detects the modification you're interested in, and also probably important to have an in vivo model which mimics the redox modification. And very often you need an integrated approach to demonstrate the physiological relevance of the modification you're studying. There are numerous gaps. Some are related to this project, and we are still, are the fodder for discussions at weekly lab meetings. But from the big picture perspective, I think that it is still very difficult to understand different aspects of redox modification. These are both technical gaps as well as conceptual gaps. The technical gaps is we don't really have a good technology to resolve intracellular ROS spatially or temporarily, and it's still very difficult to predict which amino acids will be modified and will indeed impact protein function. Conceptual problems include it is really unknown whether there are cascades of ROS-dependent signaling similar to kinase phosphorylation events, and also most of the focus in redox signaling has been paid to oxidative modification of amino acids. However, fatty acids certainly also undergo modification, and this will probably lead to a far more complex picture than what we are seeing right now.
Thank you, Dr. Kramer.
Jose Lopez from Seattle. So the fact that you can detect these oxidized CAM-K2 in tissues in disease states indicates that it's not really regulatory, but it seems to be irreversible. So why is it not reversed by methionine sulfoxide reductase?
It is.
It is.
It is. Yes. So we've done some studies showing that methionine sulfoxide reductase A at least appears to reverse oxidative activation of chem kinase.
And so is that actually the defect in these disease states, that the MSR is defective?
So that's a very good question. So we have a number of experiments in MSI knockout mice, and they are not the opposite of the chem kinase model. So the answer is it's probably more difficult than this.
Okay, thank you. Next, I'd like to invite Dr. Jose Lopez from our Division of Hematology Medicine at the University of Washington, who's going to give his presentation on an in vitro system for the study of small blood vessels. Jose?
Let's start my own timer here. So thank you very much for the invitation to come here. And I show this first slide for two reasons. The first is to point out a couple of people who are both in the room, Ying Zheng, who developed the system as she was a postdoc. She developed the system as a postdoc and has continued to refine it. And Junmei Chen, who's on my lab, who's done a lot of the work that I'll talk about. So, and the other reason is because usually at this time I'm backpacking in the Cascades, and this is Tank Lakes in the Alpine Lakes Wilderness, just east of Seattle. So the topics, I'll talk a little bit about the endothelial contribution to thrombosis at the level of small blood vessels, the role of flow and vessel diameter and vessel geometry in these phenomena, and use this opportunity to describe this in vitro vessel system for the study of angiogenesis, barrier function, and tissue modeling. So many diseases are characterized by small blood vessel occlusion, and these include thrombotic microangiopathies, and that includes thrombotic thrombocytopenic purpura, or TTP, hemolytic uremic syndrome, but there are others like malaria, sickle cell disease, and sepsis. So as a model, thrombotic thrombocytopenic purpura, a rare hematologic disease that'll kill you if you get it and don't treat it. And it's characterized by low platelet counts, mechanical red cell destruction, when there is no alternative explanation. And what one finds is systemic microvascular platelet thrombi. And if you look at a normal blood smear, what you have nice red cells, and you have a few platelets around the smear. In a patient with TTP, what you have in this particular smear are no platelets and a bunch of shredded, mechanically damaged red cells. And here's just a consequence of that is that there's small blood vessel occlusion. Here's a glomerulus shown. So a few years back, my colleagues and I discovered that a finding that might shed light onto the mechanism of TTP. And that was that if we activated endothelial cells and perfused platelets over them in the absence of plasma, that the activated endothelial cells would bind the platelets and form these beads on a string-like structure. And what we subsequently learned that what it was that the endothelial cells secreted von Willebrand factor. The von Willebrand factor individual molecules, this is a large multimeric molecule, they can self-associate to make essentially endless strands that orient in the direction of flow, and those strands bind platelets. Normally, these strands are removed from the endothelial surface by the action of a metalloprotease named ADAMTS13, and when that is inhibited or that enzyme is absent, then we have trouble, and that results in TTP. So we've used an engineered microvessel system to study this, and this was described. Ying was the first author in a paper that we published a couple of years ago. And essentially, these vessels are made in a collagen 1 matrix from a form, and the vessels can be made in essentially almost any geometry that one wants. The endothelial cells that are seated into these channels in the collagen make junctions. They make tight junctions, overlapping junctions, and adherence junctions, and they have good barrier function. So we can take these vessels, and in this case I have a vessel that has a geometry that looks like this, and we can perfuse blood through them. In this case, the blood has fluorescent labeled platelets. And so you see that when we put this blood through there, you get a little bit of platelet adhesion to the walls, but those platelets do not accumulate on the walls. They bind, and then they are released or embolized off. So we have a very different situation if we activate the endothelial cells in the vessels, and these are from umbilical veins, human umbilical veins. We have quite a different situation here where we've activated the endothelial cells with 4-ball myristate acetate. Now you see that there is a lot of accumulation of platelets on the vessel wall. The platelets continue to accumulate, and you get accumulation, then embolization, but eventually this can go on until some of these vessels are occluded. Now this is whole blood, so it has the intact enzyme. If that enzyme is not there, what can happen is the vessel can occlude. And what we get when we look by electron microscopy is that we have these platelets attached to these strands, and these strands are essentially strands of self-associated von Willebrand factor. Now, I mentioned that von Willebrand factor is a multimeric protein, and in plasma we do multimer gels, and sometimes we can obtain gels that show up to 16 bands. Now, 16 bands, each band is the addition of one protomer, which is about 500,000 Daltons of molecular weight. And it's been estimated that those protomers are about 120 nanometers. So if you put 16 of them together, you'd get a pretty long molecule, 2 microns, which is about the width of a platelet. But in fact, we see really, really enormous molecules. And the reason for that is that the von Willebrand factor will self-associate. Here you see, in a straight tube, in a vessel that approximates a straight tube, flow from left to right, that strands secreted from, so the von Willebrand factor is secreted from viable Pilati bodies, and a bit remains attached. And you see that these strands will come together to form thicker strands. Here you see from adjacent streamlines strands going together. If the vessel is not a straight tube, now this is a bigger vessel of about 500 microns diameter, then you get stranding along the wall of it, and the strands follow the direction of flow. And they accumulate most where the shear stress is highest. So here you see that this is essentially following the flow, and when you get a sharp turn here, that most of the accumulation is on the inner corner of the turn. And here now we have a smaller vessel of about 300 micron diameter, where all we've done is activate the endothelial cells. No blood has been perfused, so all of the von Willebrand factor that we see is coming out of the vessel wall. And you see that this von Willebrand factor will self-associate into strands that follow the shortest distance through this vessel. It's as if you had put a line through it and stretched the line so that, in fact, you had the minimal length that would go through the vessel. And these can go on indefinitely. So here we have a section of vessel that's approximately 5 centimeters long, and there is one continuous strand. Again, there's no blood, there's no fibrin. This is just von Willebrand factor coming out of the vessel wall. And so what we've learned is that what leads these strands to accumulate, or what favors their accumulation, is shear stress and flow acceleration. So what we have here is a grid vessel where we perfuse from the bottom, and here's the outlet. And we've activated these vessels, and you see that there's accumulation of strands at the inlet. Here's where shear stresses are very high. Shear stresses are very low here because we have a very large cross-sectional area, and as one approaches the outlet, the flow has to accelerate, the shear stress goes up, and now we get strands that are oriented in the direction of flow and thickening as one approaches the outlet. And here's just a higher magnification of the outlet, and you can see that there are strands basically going in the direction of flow. There's a region right here that has quite a complex flow pattern, as the flow is coming in this direction, and in this direction. And if you look in higher magnification of that region of what's happening to von Willebrand factor, you see that essentially you've made something that looks like a spider web there, that when blood's going through there, they can capture blood cells. So it turns out that shear stress is quite important for this phenomenon, but even more important is flow acceleration, and that's called elongational flow. And here is just demonstrated from a recent review by Tim Springer that at 2,500 inverse seconds of laminar shear stress, one gets flow acceleration, or one gets unfolding of the model polymer, and that unfolding occurs at much lower forces in the presence of elongational flow. Here's a vessel in which we've either perfused at the top normal plasma after we activated the vessel, or plasma from a patient with TTP. They lack the activity of that enzyme, ADAMTS13. In the presence of normal plasma, one sees von Willebrand factor on the wall, but there's no accumulation of strands. What we really see actually on the wall are these little dots, which are essentially like stumps that are left over after the ADAMTS13 have come along and mowed away the strands. In contrast, in TTP plasma, which lacks the enzyme, you see massive accumulation of von Willebrand factor on the walls here, and in some cases even starting to occlude the vessel, and you see complex patterns of net-like formation on the vessels. So these strands of von Willebrand factor, what they do and why they're bad is because they capture platelets. And just by themselves, by capturing platelets, they can occlude blood vessels. And here the platelets are labeled pink, and you can see that in an activated vessel with ADAMTS13 inhibited, you get a lot of accumulation, particularly in the inlet to this grid vessel. But in addition to capturing platelets, we also show here that there's a lot of capture of erythrocytes. Red blood cells are captured. And of interest, that capture, if one inhibits the binding of the platelets to the strands, you still have the strand shown here in green, but you don't have platelets. And now that you don't have platelets, you don't have red cells. So we think that this has a lot to do with pathology in certain syndromes, such as TTP and possibly also malaria and sickle cell disease. In addition to those two cells, platelets and red cells, this shows that in regions where you capture a lot of platelets and you have a blocked vessel, essentially, that's full of von Willebrand factor, that you not only get platelets labeled here with CD41, but you get a lot of leukocytes, polymorphonuclear leukocytes and monocytes. So the von Willebrand factor can self-associate not only by the contribution of the VWF from the endothelial cells. There's also, von Willebrand factor is also a blood protein. It circulates in blood. And in this case, what we've done is we've perfused fluid phase von Willebrand factor over a transluminal strand of VWF, and you can see that the fluid phase VWF will bind on the upstream surface of this strand, further contributing to the thickening of that band. If you don't stop this, then you get trouble. So we found something that can actually prevent this by accident. And the reason that we found this by accident is because we work with purified von Willebrand factor. If you purify it too much, you lose it if you leave it in a tube. But we found that there was something in boiled plasma that could protect the von Willebrand factor. And to make a very, very long story short, that substance is ApoA1, which is the apolipoprotein of high-density lipoprotein. And that can prevent the self-association of von Willebrand factor under tubes, and then we also studied in a vessel. So in this case, we activated a vessel to secrete von Willebrand factor, and then we perfused a solution that had ApoA1. You can do that with ApoA1 or HDL. What we find that the ApoA1 will decorate the strands way better than the VWF antibody does. I guess it prevents the antibody from binding. And if then you perfuse fluid phase von Willebrand factor, which is at the bottom, you get no association. And the consequences of this are that there are fewer platelets that attach to that vessel, and that's shown here. If there's no ApoA1, you get a lot of fibers of von Willebrand factor, and those fibers bind platelets. In the presence of ApoA1 in an identical grid vessel, you still get a few fibers accumulating, and that's probably because we didn't have the ApoA1 in there all the time. And pretty much the amount of platelets that adhere are proportional to the amount of fiber formation. And we've looked at in some of these disorders that have high von Willebrand factor, at the ApoA1 levels. Here's normal. In sepsis, we have a marked drop in ApoA1 and HDL, and TTP as well. And here we show that the concentration of very sticky forms of von Willebrand factor is highest when the ApoA1 antigen is lowest in these disorders. So just to say a couple of other things about other uses of these vessels, I have about 30 seconds. We can use them for angiogenesis. Here's showing angiogenic media. They make leaky vessels. Here we have angiogenesis induced by perivascular cells. Those are not leaky. We've also studied the effects of platelets on barrier function. They can restore barrier function. Here's a leaky vessel that was incubated with platelets overnight and it became non-leaky. We've also used it to simulate some tissues. And that tells me I'm done. And here we've put CD41 cells. They attach to the wall. They actually change the character of the endothelial cells to make holes. They put through proplatelet processes or these burst-like platelet formation. So to summarize, we've developed this versatile system of ex vivo vessels where you can study all the things that I just mentioned, including thrombosis, which has not been mentioned here and I think is quite important. And this system is good because it allows modification of flow and vessel geometry, assembly of multicellular systems. You can monitor it in real time, and you can do sophisticated imaging. There are lots of challenges because this is a laborious and highly specialized technique, and it has low throughput. Nobody likes to hear this. The gaps in terms of thrombosis, I say that we still have to more precisely define the role of von Willebrand factor and platelets in a lot of these diseases of microvessel occlusion. But there are lots of opportunities in that we can use all human components. We can control the components and the timing of their use. We can be reductionist or we can not be reductionist. And we have the ability to use cells from patients or deficient mice. So I've already mentioned two of the people. I want to mention one other, and that's Dominic Chung, who did the HDL work. Thank you.
Thank you. We have time for a few questions. I have one, and the system that you showed us is sort of a purist system, isn't it? It's using purified von Willebrand factor.
The von Willebrand factor is coming out of the endothelial cells that we've seeded in it.
Yeah. So if you put blood through the system, can you make one comment about thrombosis and fibrin deposition, and also what's the viability of the endothelial cells in that setting?
The endothelial cells are more viable than they are in a planar culture. And we don't know how long you can culture them, but they seem to like to grow in circles rather than in flat planes. In terms of fibrin, that's a problem. And we intentionally anticoagulate the blood, but we are now developing a system where we can induce a fibrin clot within the vessel, but we still don't know. We're very interested in studying contact activation of blood.
Thank you. Yes, please.
There was a suggestion that actually these long strands of von Willebrand factor may be important in shearing RBCs and be part of the TTP pathology. Have you tried to replicate something like that in your system?
We have, and we see that. I just didn't have time to show you. And we actually see, in some cases, the red cells hang up and get sheared right by the strands. I just want to mention that Dr. Mayadas did some of the really great early von Willebrand factor work showing how it polymerized. I love those papers, by the way.
Thank you.
Please.
Yeah, what kind of anticoagulant you use for blood perfusion?
Generally citrate, but you can use heparin or Hirudin or whatever else. But generally,
that means when the BWF stream formation is calcium independent?
It's calcium independent, yes.
Thank you.
Okay, thank you. Please.
The self-association is calcium independent.
Nice talk. If you use ApoE1 Milano, which has been shown to be protected, you have the same effect, or is...
We haven't used it, but I think it'll work. We're trying to actually get fragments of the ApoE1 and see what's doing this, but it works if it's HDL or it works if it's ApoE1. I think Milano will probably work. I think we should try it.
Thank you. Mark?
So I'd like to comment on the relationship between circulating von Willebrand factor that we can measure clinically, and on the one hand, and these strands, these von Willebrand factor strands that you've demonstrated so nicely.
Well, what we've observed, if you don't have the enzyme and you induce Weibel-Pilaty secretion under flow, is that a fraction of the von Willebrand factor is released. Another fraction remains on the surface. I don't know what's the precise relationship, but what you expect would be that in those syndromes where you have a lot of vessel wall von Willebrand factor, it would be reflected in higher levels and bigger multimers of von Willebrand factor in the blood.
Thank you. Short question, please.
I just am wondering about the in vivo relevancy, because when I think of thrombosis in vivo, I think of a thrombus forming in a low shear space. Branch points, low shear, turbulent flow, rather than the high shear that you're talking about.
So we have in the blood two types of polymeric molecules that are involved in the adhesion of platelets. One is von Willebrand factor, which is a preformed polymer, and that one seems to function almost exclusively in high shear stress. The The higher the shear stress, the more it'll self-associate and the more platelet accumulation you'll get. So that's why you get it in pre-capillary arterioles and capillaries. The other polymer is fibrin, which isn't preformed and forms and actually can't form at high shear stress. So it's kind of an interesting thing. So what you're talking about is the formation of a fibrin thrombus that's generally rich in red cells and does have platelets, but it's not dominated by platelets.
But in some acute coronary syndromes, I think the thrombus is platelet rich.
It is platelet rich, and it has fibrin, and it has von Willebrand factor.
Very, very short question.
We see a lot of things in vitro that does not happen in vivo. So I was wondering how relevant is the system for patients with thrombocytic purpura or other related pathology?
I agree with you. Well, we also see some bad things in vivo that this mimics, like TTP. I mean, actually, TTP is the reason that I went into hematology because it was such a dramatic phenotype. So what we're trying to do here is we're trying to make this system where we can understand biophysical events that contribute to those things. I think that what we find is we've found, for example, I think already with this in vitro system, why thrombi accumulate where they do in TTP, the high shear stress regions. 2, we've figured out why in every one of those disorders where you get massive release of endothelial von Willebrand factor, there's low HDL, and it happens very fast. We think that's a sequestration event. So we're using this to help us understand the in vivo. Obviously, we have to go back and correlate all our artifacts with things that actually happen in vivo.
All right.
We're going to carry on for the panel discussion. So, Jose, if you want to stay up here with us, and all the other speakers, Mike and Tanya, Gary, and also I'd like to have Dritten Egalnu and Ira. Yeah. Okay, thanks.
I guess I'll jump into the pool first, and all I can say is, wow. It would have been much easier for me, I'm Ira Herman, to present some of our work on how pericyte chemomechanics teaches endothelial cells to regrow after being growth arrested. We've heard about modeling in animals and in vitro. We've talked about pathogenesis and human disease. We've talked about how oxidative stress might be a player, whether hemodynamics and biomechanics play important roles, how the microenvironment influences cellular dynamics. Professor Aird started us off by teaching us that not all endothelial cells are actually created equal, and that organ-specific differences might be actually dependent or regulated by the cells that are born in those organ systems and the way in which those tissues come together to give rise to the organ-specific functionality that we see. And then, in addition, we have also, and I think importantly, and maybe this is a good point to jump off from, talked about how vascular cells not only talk to one another and how they modify a matrix, and for example, in glomerulonephritis, or whether we're talking about a disease in the brain or whichever organ system, it may be about the way in which the microenvironment, the non-vascular cells, actually talk to the vascular cells that shape the phenomena, whether they be physiologic or pathophysiologic. In the case of, for example, aquaporin-borne channels in astrocytic foot processes that are uniquely organized around special blood vessel features that penetrate the brain parenchyma to help take CSF out of the organ while we sleep. I don't sleep enough. Maybe that's my problem. So if there's not something here for you to talk about, to ask about, we can start asking questions or talking amongst ourselves. But we challenge you, the audience, to perhaps go ahead. Please.
This one. Yeah. Sorry, Tritana Galileo. So I wanted you to think a little bit about something else. And
We talked here about the small vessel cell interactions, but I think there is a theme that I want you to think about, and the vascular is not really in its own. I mean, it interacts on one side with pericytes and the tissue-specific cells to form organ-specific properties, but also interacts with immune cells. And I think one of the themes that I want you to think about here is the interaction of the immune cells with the vasculature. And I think Dorian McGovern talk tomorrow is going to bring back that discussion as well. But, you know, this came from the first talk, thinking about lupus. And even though the lupus discussion was more on the kidney, it actually also has a lot of neurological effects. So I want you to kind of, you know, maybe some of the questions that are going to be thought about really bring back that, you know, thinking in terms of an underlying theme that brings immune cell CNS interactions. The second talk by Gary really described very nicely this particular disease, but, you know, the CNS has sort of an immune privilege property, but there's a lot of interaction of the immune cells with the CNS, and that becomes very prominent in diseases of the CNS, such as MS or this disease. And in particular, I think also some of the stuff that Nika talked about, for a long time we thought that the brain doesn't have a lymphatic system, but yet this particular lymphatic really brings in a new concept of how we think about brain physiology. So there may be really an underlying theme in all the organs that I want us to discuss, because I think part of this workshop is really to, you know, think of a unifying theme of the microvasculature. So, you know, maybe we can sort of streamline the discussion in some of these 3 points.
Okay. Open to questions from the floor now.
So I have a question. I guess it's really to Macon. Okay. So you mentioned that proteins come out of the perivascular space with pulsatile flow. But what drives this? I mean, is this vasomotion? Is this, you thinking of cardiac motion of the vessels? If it's cardiac motion, they all seem to go up and down together. So that means you need valves. So where are the valves in the perivascular space?
Voivella have described valve-like structures in the Wecken-Robin space in human patients. He does very, very good EM studies of valve-like structures in around human arteries. So I would also say that, of course, that the exit of protein from plasma to CSF or to interstitial fluid in brain is, of course, much smaller than it is peripheral tissue because of the blood-brain barrier. But we do have leakage of proteins into the interstitial fluid, and especially, and this is what I try to talk about, is that it looked like all cytosolic protein, even though they don't have a secretory sequence, are released to the interstitial space and can be found both by microdialysis by David Holtzman in the interstitial space, but also in the cerebrospinal fluid.
And what's the pulsation?
Also, we just published a study that showed that pulsatility, at least in part, is responsible. So you can drive these fluxes of cerebrospinal fluid up and down by changing the pulsatility, but it's not the whole story.
But that's vasomotion?
It's vasomotion, yeah. And whether that's just a cardiac pulsatility or whether it's a very slow motion you have described, we don't know.
John Lederer, University of Maryland. I don't understand what is special about sleep that permits, during that period of time, the cleansing of bad proteins. Can you describe this a little better?
It's a good dream. No, we actually showed, and we did quite a bit of studies on it, that there is no, basically sleep is a default of the brain. Arousal is modulated by all the neuromodulators, norepinephrine, orexin, and so on. And what the neuromodulators do is not to drive different level of lymphatic fluxes to sleep and awake by pulsatility, but what happens is that the interstitial space in the brain parenchyma expand 65% when we go to sleep.
So why is that?
Our cells shrink as a function of being exposed to norepinephrine in particular.
So what is contracting and what is expanding? I mean, is all that worked out?
It's underway, but what I know is that there's papers in press in human brain showing, using diffusion-weighted images, that water movement is restricted the less you sleep. So if you sleep deprived patient, you have less water movement in them than during the sleeping state.
So Mike, what's the role of the neuron in all of this? Yeah.
So I am not talking about whether it's astrocyte or known, shrinking or expanding. I don't know, because we don't have the optical resolution to explore that. What I know is interstitial space changes in volume.
Maria Luisa Sequeira Lopez, Charlottesville, Virginia. Continuing on with the same theme, do you see the same changes during all sleep stages, or the rapid eye movement stage?
That's a really good question. We haven't explored it in detail yet because mice have only very short-lasting, REM episodes, and we only have 2 shots of the traces. So we are exploring it in larger animals that have more solid REM sleep to see whether there's a difference or not in that, but we don't know yet.
And another question is, what would happen during exercise? Because your mouse is resting when you are studying it. So what do you speculate that will happen?
It goes even further down. I would expect that. Yeah.
Hi, Dave Antonelli, Michigan. Dr. Lopez, I wanted to ask you a question. What do you think it would be like to expand the kind of system that you're working with so it is more readily available and maybe even commercially available in the scientific community? And maybe associated with that, what do you think of the systems that are already out there, like the cartridge systems and such? And some of your thoughts on that.
So for the second question, I can't really answer because I haven't used them. We either use parallel plate chambers that we can coat with endothelial cells, or we use this system. We are trying actually to expand this to make it more available because it is kind of finicky. There was a company that was interested, IBIDI was interested in this, and they still are, I think, but we don't really have the whole intellectual property issue as a problem. But yeah, I think what we need to do is we need to make it easier to use. And I guess that the complex one, if you need to do anything complicated, then you're probably going to have to, do a lot of work. But there's a lot of simple things that you could use with a simpler model.
Do you see differences depending upon whether or not you use, for example, venous endothelium from umbilical vein, or whether you use capillary-derived cells?
Well, Ying has probably done that more than we've done that in our lab, and she does see differences, although what we also see is we see something that Dr. Ayer talked about this morning. We see a lot of plasticity depending on what you do to the cells. For example, I didn't have a lot of time to show this, but the megakaryocytes completely change the endothelial phenotype, absolutely completely change it, and make it like they wanted to make, as if it were in the bone marrow.
Just back to the sleep conversation. I was wondering if you've looked into patients that have LVAD implants, so they don't have pulsatile flow.
We haven't, but we have reduced pulsatility, and it reduces the perivascular influx of cerebrospinal fluid. So we haven't looked at it.
I wonder if we could get back to the first talk and just talk a little bit about how antibody-mediated injury influences glomerular adaptive responses. And I'm particularly interested in knowing whether or not the modifications that you make using transgenic approaches to the macrophages, or leukocytes in this case, actually are feeding directly to the endothelium, or how does the mesangial cell talk to the endothelium to give rise to the glomerulonephritic disorder that you see? In the PAS localization that you showed, the mesangial matrix was remarkably hypertrophied. Mm-hmm. And so I couldn't help but think that the mural cell talking to the endothelium was a way in which there was sort of crosstalk coming back from the parenchyma. Is that true or not?
Yes, so mesangial cells are sort of akin to smooth muscle cells and parasites and other vasculature. We don't work so much on mesangial cells, but certainly they contribute significantly to glomerulonephritis. But just getting back to just a point on immune cell endothelial cell crosstalk, definitely there's something there, but what I wanted to actually also bring up is that there's something about the target organ susceptibility factors, and this is probably in the CNS and anywhere else, that dictates whether that immune cell will actually even react to the deposited immune complexes. So in our case, we had to actually knock out a receptor on MAC1, which we think regulates Fc gamma 2A, in order to see the injury. But there may be some other endothelial factors, some mesangial factors, epithelial cell factors that also give rise to that ability of the immune cell to sort of see the immune complexes and make them pathogenic. And that also gets back to some model systems that when we talk about, we use naive mice, for example, in all of our studies, and we all do this with C57 Black 6 in our facilities. But many of the diseases we talk about, including lupus, have a lot of comorbidities associated with them. It may now be part of that disease susceptibility, target organ disease susceptibility factors. And so we should also think about mouse models that maybe are slightly more sophisticated and can replicate what happens in humans.
For in the case of diabetes.
Exactly.
Yeah.
Right.
So I had a question related to that, just keeping in the theme of the immune cell endothelial interactions. In your knockouts, have you looked at other disease models where neutrophils play a role? For example, there's very clear evidence of neutrophils implicated in stroke progression. There's also a role in MS.
Mm-hmm.
So have you looked in those knockouts to see if there are profound, changes, just like what you described in glomerulonephritis?
So in our MAC1 knockouts, which we generated a long time ago, MAC1 is normally thought to be pro-inflammatory. MAC1 is a CD18 integrin, is supposed to be important in leukocyte recruitment. But then since then, it's been really appreciated that it can be immunomodulatory. And we think in the context of immune complexes and Fc gamma receptors, actually dampens down. And interestingly, there's a SNP very highly associated with lupus in MAC1. And that SNP leads to a reduction in MAC1 function. So the model that we had developed actually may have some relevance, so that in this case, when you actually take away MAC1 function, it seems to hype up the immune system, versus in other sterile inflammation like with TNF, etc., MAC1 may actually be pro-inflammatory. So there's a yin and yang of many of these molecules.
Do you know which of the ligands for MAC1 is involved?
Oh, we'd love to know that. We think it may be complement or ICAM. We don't know. We'd have to do that experiment.
So one more question from the floor.
Yes, Jose, I was just wondering, endothelial cells make ADAMTS13. Can you just comment on its regulated release and whether you've looked at, for example, knockdown, whether you can make those strings even larger in cells that don't secrete?
That's a very good point. Well, I actually don't know anything about its regulated release. I think it's constitutively released. I don't think there's a lot of it in most endothelial cells. Most of it comes from hepatic stellate cells. Probably, so we have some experiments, and I think these might address this. We haven't knocked it down, but we put high concentrations of antibodies that will neutralize it in there. And the extent of strand elongation and thickening is even more rapid. So I think there is something that's coming out of the endothelial cells.
One more short question.
Third question for Dr. Niedergaard. Comments on effects of fluctuations of serum sodium and water fluxes related to sodium levels, how that might impact your glymphatic system.
That's really interesting, because the glymphatic and the cell volume changes is, of course, all about iron homeostasis. So maybe we can talk about it, but I think it's extremely important, and I think one of the things we're working at now is actually to show that the interstitial iron composition differ in sleep and awake, and that can again basically drive arousal, because if you just increase potassium by 0.5 millimolar, you change the whole excitability of cortex. So it's not just volume changes.
Okay, an applause to the discussants and chairs and speakers. We're going to have a quick break. Please be back by 3:15. We are going to start promptly with the next session.
Hello, good evening. My name is Margaret Bino. I am Associate Professor at Cornell University. First, I'd like to thank the organizers for inviting me to chair the session today. I've learned so much in one day since I've been here, and I can't wait for the rest of the evening. So today, we have several speakers, and I just want to remind the speakers to try to be on time. New rule today, and that's from me. For every minute you go over your talk, you buy 10 drinks tonight at dinner. All right. And the first 2 drinks, I get 2 drinks for each one you buy. Okay. All right. So with that taken care of, our first speaker is Azra Malik, Dr. Azra Malik from University of Illinois at Chicago. And he would talk about signaling pathways regulating endothelial barrier function.
Good, thank you. Yeah, I forgot my credit card, so.
We'll work something out.
Thank you for the invitation. It's a wonderful symposium. I especially like that bit about Galen, and it's so delightful to see Bill Aird so excitable when he talks about Galen. I think he's probably the only one. So, so I'm going to be talking about the sort of the intimate mechanisms that regulate permeability, and more in terms of what little we know, and I guess some aspects of what we know, and perhaps areas that are getting a bit overworked. So, to begin with, I think it's good to talk about a summary of the field, or get a general sense of the field, as I've tried to deconstruct it here. So the main aspect of permeability regulation is really at the level of the junctions. By that I mean adherence junctions, and these are well known to be consist of a series of sort of interconnected. Can you hear me? Interconnected proteins. These are weak adherent, which is very specific to endothelial cells. Then another one, a family of catenins, P120 catenin, beta catenin. So it forms this network of these junctional proteins that are then connected to the cytoskeleton. So it's a very important arrangement, and this arrangement here is really central to the dynamic regulation of barrier function. We've shown that, and multiple other groups have shown that. But there's some important aspects of it that I think have been recent in their understanding. This concept of disassembly of adherence junctions is a fundamental concept because, I like to think of junctions as plastic, in that they can disassemble. I don't mean they rip apart, but I think this is a very ordered a mechanism. It's a very orderly mechanism, and they also have the capability of reannealing or reassembling junctions on the basis of certain signaling cues. And here I've kind of diagrammed what would happen if the barrier function was disassembled. There would be a drop in what we can measure actually is electrical resistance. So there's a drop in that, and these are pretty canonical changes, and this may vary according to the stimulus that disrupts junctions, and eventually, in this case, it's 2 hours, but it may be 4 hours, it may be 6 hours, and so on. But depending on the initial stimulus, there's this reassembly or this restorative phase, and so the two unknown questions, in my view, which I think should be studied in all microvascular beds, is the nature of the disassembly. One important aspect of disassembly is that weak adherence is actually internalized. It's an active event. It's endocytosed. I know endocytosis endocytosis is not a verb, but it's literally endocytosed on the basis of some specific signaling pathways we've recently described. And then there's a return to the membrane where it reassembles and reanneals the barrier. So this V-coherent internalization event is a discrete event, and I'm going to show you that it's controlled by certain signaling pathways. Then the reannealing itself is again a event on some specific signaling pathways, and there are a couple of them because it's a complex process and it's a physiologically, homeostatically relevant process. So one of them involves RAC mediated trans interaction. Remember, V-coherent binds in a cis manner, but it also binds in a trans manner. So it's RAC, this GTPase RAC mediates this, and this other GTPase, CD42, partitions this function of these Rho GTPases, and its function is to elicit by some active mechanisms formation of lamellipodia, which I'll describe for you. So this is what we've shown this is to be essential, this protein kinase C-alpha regulation of V-coherent, and it functions in a way, I think, in all vascular beds. Basically, you have this domain of P120-catenin, which is a binding protein. It's very perimembranous binding protein, right at the edge of this juxtamolecular domain. PKC phosphorylates it, so there's phosphorylation at this serine domain. PKC then, this phosphorylation dissociates it, and this protein AP2, which is an adapter protein for clathrin, binds there, and then you have this clathrin-mediated internalization of V-coherent, and hence the disassembly of junctions. So it's a well-regulated, well-coordinated process. Now, I think that from a perspective of how do we restore integrity, I think that's a very important question for all inflammatory diseases in the heart or the brain or wherever. So what regulates this sort of reannealing or reassembly process? And again, one can sort of partition various signaling factors. So one of the critical ones is CD42. Now, CDC42 functions by inducing polymerization in these newly formed lamellar protrusions, and really allows these cells to meet one another and to contact one another, and you can visualize that, and various studies, some of them shown here, indicate that CDC42 is a requirement, not only a requirement, it's sufficient for the formation of lamellipodia protrusions and for this initial event or this initial step in the formation of adherence junctions. Now, because of the lack of knowledge, we don't know events 1 to 10. We know events 1, 2, and 4, but so there are probably a series of handoffs from this initial event to the subsequent event that leads to the formation of these adhesive junctions. Now, we've probed somewhat in the role of RAC1, which is the other interesting GTPase in this context. And the approach we've taken is that we can photoactivate RAC locally at the junction using these are collaborations with Klaus Hahn, where RAC can be photoactivated using in live cells using this expression of this construct. And when you apply UV light, this unfolds, and so you have activated RAC at the level of the junctions. So in this case, at least, we use the photoinsensitive probe, which is this one, and well, I'm sorry, this is a dominant negative one, and these are the two controls, and this is the actual probe we used. So the question we asked was, what is the consequence of localized activation of RAC in promoting reassembly? And this is a complex graph, but I think it makes one or two simple points. And that is, if you activate RAC at the junctions here, this is where the UV light is shown, and you look at mobilization of V-cadherin at different times very rapidly. So within 3 to 4 minutes, you get increased fluorescent intensity of V-cadherin at the junctions. Whereas if you use the inactive probe, photoactivated probe, there's no increase in V-cadherin. So what this means is that RAC functions, and it only is a function in mature junctions, junctions that already have adhesion complexes, that RAC functions in actually making the barrier stronger because it enables the accumulation of V-cadherin at the junctions. So RAC is, and again, like CD42, which is an early step, RAC1 is a subsequent step that promotes the accumulation of V-cadherin at the junctions. And through many studies, we came across a sort of general model or general concept that RAC stabilized V-cadherin trans-interaction to reduce the V-cadherin dissociation. So the accumulation is the function of the reduction in V-cadherin dissociation. And this is because RAC functions by inhibiting Rho A. We know this cascade, inhibiting Rho A, which thereby inhibits myosin light chain kinase phosphorylation and reduction in tension. So the reduced tension occurring at this junctional domain is the primary factor responsible for RAC-induced assembly of the junctions. And this is clearly an important point, we think, that because it's a way of stabilizing a junction, and it's a way various junctional stabilizing mediators such as sphingosine 1-phosphate function. So this has important relevance to try to understand the biology of junctional re-annealing. So a few words about the other pathway. The pathway I've talked about so far is the paracellular pathway. There are a couple of people in this room, for example, Radu Stan, who know quite a bit about this other pathway. So this other pathway is important, but a bit of a Cinderella in that much less is known about it. But we do know it's important. We don't know why it's important. We know it's important. And this is the caveolar pathway. If you put albumin in the luminal side, this is small particles of albumin, 1 or 2 nanometers. So albumin particles are engulfed in these caveoli and are transferred from the lumen to the basal side. So this is the basal side here. This is an alive animal, and you can see a caveola depositing these albumin particles. There's one out here. This is only 5 minutes after adding these. So it's a very rapidly activated system, and it's a fundamental system for transport of large molecules across the endothelial barrier. Please, you can see here that if you look at the junctions, there's no albumin nanoparticle. Junctions are simply too small. The diameter radius is too small for the transport of these molecules. So albumin and other macromolecules, at least in these type of endothelia, and clearly in the blood-brain endothelial, can only be transported through a caveola, but that's another question. So this is an important pathway. So if you want to play sort of genetic games with this pathway, for example, if you knock out a protein that is responsible for caveoli formation, caveolin-1, in endothelial cells, you can see interesting compensatory mechanism being activated. And you can see here that in fact there is transport. That there is transport across the adherence junctional complex here in the absence of caveolin 1. And this shows another very dramatic picture, also in the absence of caveolin 1. You can see albumin being transported by this mechanism. Am I over time yet?
Just wrap up. You have 1 minute.
Wow. Okay. So there's an interesting compensation occurring. And the compensation is this. This just appeared recently. And that is there's a consilience between the caveolar-mediated pathway and the adherence junction. So when caveolar pathway, there's an involvement between the caveolar pathway, release of eNOS, nitrating this Rho gap, P190 Rho gap. So this is a nitration reaction, which kills the gap activity. So you have Rho activity at the junctions, and then Rho opens up the junction. So there's a very interesting crosstalk between these two pathways. So my final bit, just 1 or 2 slides, is that we've recently begun to address the question, what is the mechanism of endothelial cell repair? This has nothing to do with renewing, but it's repair. So we made this, I think, an interesting mouse model where we have tomato red mice, tomato red as a fluorophore labeling non-endothelial cells, and GFP labeling endothelial cells. So we can then test the idea of non-endothelial cells being labeled red and endothelial cells being labeled green. So the question we wanted to ask, if you injure the endothelium precisely, do other cells take over the endothelial barrier or endothelial function? By other cells, I mean red cells, let's say progenitor cells from the red tissue, or do endothelial cells themselves induce the repair process? And the long and the short of it is, and this just shows the model, so you can green label the endothelial cells, everything else is labeled red. And the long and the short of it is, if you induce LPS injury, let's say, of the endothelium, there's a decrease in the amount of GFP at 24 hours, but importantly, recovery, so indicating full recovery of this injury, and we titrated the dose, et cetera, to arrive at that. Now, what this means is that this is due to a decrease in the green cells or the endothelial cells. It's not due to increase in the red cells, because if that had occurred, this wouldn't have recovered to normal. So what this suggests is that they're facultative endothelial cells. That is, endothelial cells that the green cells that are promoting repair.
To the last 2.
Sure, that are promoting repair are specialized, and they have some specialized functions, and they're facultative. So the unknowns in this area, there are many, and I haven't learned many lessons, such as keeping on time. So one of them is the plasticity of endothelial cells. The other is the notion that you can close adherence junctions. The third is that trafficking of caveoli are important, but poorly studied. Endothelial lineage analysis, I think, is a way forward. And gaps, there are a lot of gaps. Details of the signaling pathway, the precise working of the transcytosis pathway, definition of facultative endothelial cells, and so I mean, this is a very interesting idea. How does liquid and solute, how do they transport across, as well as blood cells and cancer cells? So I'm making this kind of heretical argument. Metastasis is really a disease of the endothelial barrier. Thank you.
We're happy to take questions. Just one or two quick questions, please.
Or half a question.
And you better be prepared to buy a lot of drinks tonight.
Okay.
Can I start?
Yes.
Yeah. Hi, Marty Kluger from Yale.
That was a very nice talk, and I think you're absolutely right that the localization using the photoactivatable RAC1 is a technological improvement over looking at RAC1 in a global way. But you also spoke about maturation of junctions, and I wanted to bring up the point that tight junction molecules are expressed throughout the microvasculature, and more so, say, in capillaries than post-capillary venules. So I think that some of the mechanisms you discussed about reformation and also taking junctions apart should be applied to those molecules and the different caliber beds.
Right. It's a very good point.
Yeah, next question. One more. Okay. Very quick.
Yes, this Irobo Bogu from University of Alabama in Birmingham. I want to first of all find out which cells you are working with, which endothelial cells we're working in particular, because I guess the ability to form adherence junctions and tight junctions will depend on the cell type you're working with.
Well, actually, we use the ones that where we can get adherence junctions. So we don't use the cells. Microvessel cells, for example, microvessel endothelial cells, they form very nice adherence junctions.
And did you look at tight junctions at all?
Tight junctions are in our experience, are not that well expressed in the endothelium, except for the unique brain endothelium.
Okay.
And the main junctional permeability pathway, after much hair pulling and this and that, appears to be the adherence junctions.
So we are going to have to take this up tonight over drinks, especially since he's buying. Okay, so the next speaker is Dr. Berislav Zlokovic. Is that how you pronounce? Okay, great. And Dr. Zlokovic would talk about the role of blood-brain barrier in neurodegeneration.
Thank you very much. And I can go to lessons learned and gaps, because if we don't have time, right? Okay, so let me just try to really briefly review for you. This area is huge, so I'm not pretending I'm going to tell you very much. But I'm going to focus my talk both in animal models and humans on parasites and how they regulate the blood-brain barrier. So this is going to be my last slide, too, because we need desperately biomarkers, molecular biomarkers of function of different cell types within neurovascular unit, including endothelial cells, pericytes, astrocytes, neurons, white matter, and also we need imaging biomarkers. So we tried over the past year and a half to develop a molecular platform for about 40 biomarkers for cell-specific types within the neurovascular unit, and also developed some imaging tests both in humans and animal models, including DC-MRI, ASL-MRI, etc., and relate this to brain structural and functional changes, including diffusivity, tractography maps, but DTI, MRI, and functional load maps. So our model of small vessel disease, that I call it small vessel disease only for you this time, it's usually parasite deficient mouse model, I think is characterized by very strong blood-brain barrier breakdown. As you can see over here, this is fibrin extravasation, and that correlates with the loss of pericyte coverage. More you lose pericyte, you have more accumulation of fibrin. And also there is accumulation of other molecules coming principally from blood, such as thrombin, plasmin, that degrades neuronal laminin, etc. So going back to the slide that I just missed, I want to say that our model is PDGFR beta plus minus mice. That's receptor on pericytes, the green cells, which actually get signals from endothelium, from PDGF-BB, and then forms form vascular networks. So you see age-dependent progressive loss of pericytes and progressive loss of capillaries. So it's a capillary degeneration model. And as you can see here, there are pericytes that are separated and they're dying, and also endothelial brain capillaries are dying. So they're dying together. All right, and so is that functional? So we did characterize that first in my lab by Bob Bell and Winkler when I was still in Rochester. And this was just showing by lectin angiography that we have, actually this is a perfusion, two-photon microscopy of the blood flow, by showing that at 1 month we have reduction in the blood flow already. So this is reduction in capillary length, and that goes progressively with age and correlates with the reduction of the resting cerebral blood flow, here shown by quantitative autoradiography. Very important to notice is that these changes happen very early. They happen at 1 month of age in this small vessel disease parasite deficient model. that has this chronic blood-brain barrier breakdown. So all vascular changes happen very early. More recent work in this area, we were interested in CBF dysregulation following on some work of David Atwell that was published in Nature recently. And we found also in response to stimulation that we have red is capillary response and gray is arteriolar response. And we have the capillary response we also found comes a little bit before arteriolar response. But of course, both responses play an important role in functional hyperemia. But in pericyte deficient mouse model, we have the reverse. Arteriolar response comes first, and we have very, very delayed capillary component. So we believe that this is the reason for low oxygen delivery. And here are oxygen tissue maps, and the blue is bad, I mean low oxygen, and the reddish is better. So as you can see in pericyte deficient mouse model, we have about over 20% vessels that have a very low oxygen, areas that have very low oxygen level, about less than 15 millimeter mercury. So these all dysregulatory vascular problems or neurovascular dysfunctions happens very early. At that point, there is no any problems with neuronal dysfunction. But we are very interested to see whether white matter is affected in this model, because white matter regions have the lowest cerebral blood flow normally and have lower capillary density compared to gray matter regions such as cortex or some other regions, hippocampus and etc. So we've done monosynaptic anterograde tracing using AAV-EGFP in barrel somatosensory cortex, and then determine the overlapping on the contralateral cortex monosynaptic pathways, also sensory motor cortex 2 and internal capsule. So as you can see, there is dramatic reduction of this connectivities through the corpus callosum very early at 2 to 4 months, and that is progressive with age. In internal capsule is less pronounced and comes a little bit later, and there are no such changes on the short pathway, so such as S1, S2. So we then wanted to study by using, DTI MRI functional maps and see how these functional maps that are here beautifully generated by our collaborator, Madeleine Doianu, also from Romania, like Zorina. And they always like these nice things to present. And so this is basically all this different tractography that we used to study pericyte deficient mice. So how the vascular deficits influence different tracts in these mice. So the simplified version of these mice here shown just by corpus callosum, and you can see that these mice, 2 to 4 months, start losing tracts and fibers, as shown below over here. And also, of course, the older mice, they all completely lose these fibers and have a very significant loss of connectivities. What is also interesting that this white matter tract across corpus callosum show the first reduction in the volume, while hippocampus and cortex do not show changes at that stage, but show changes only in a later stage. And this has been confirmed by calculation of a myelin axonal ratio, which is called G ratio. And as you can see, these are normal G ratios at 2 months old and 9 months old. They are parallel. And then at 2 months, this G ratio moves down, which actually suggests a loss of myelin and also number of degenerated axons. And is also confirmed at a molecular level by BMPA. And in an earlier work, we showed that at that stage, hippocampus does not show changes by, for example, a Golgi technique shown over here. But at 8 months, this is CA1 subregion of hippocampus. This pericyte deficient mouse model, because of small vessel disease, develops loss of neurons, loss of dendrites, dendritic length, spine density, and this can also be confirmed by H&E staining. As you can see, great loss in the CA1 region. So what I showed you was white and gray matter degeneration. In a parallel work, we also showed that this model of small vessel disease developed A-beta and tau pathology when crossed with APP mice, accelerated, and also leads to tau phosphorylation and neuronal degeneration and neuronal loss, having a spiral effect. But I don't have time to talk about that. So we moved to human studies, and we use a model very similar that Gary Rosenberg talked to you about, but we extended this model to actually study in parallel several regions in the brain. So we focus on 12 or 14 regions, And what we first noticed with this model, this is, for example, a mask of hippocampus, that we have age-dependent leakiness, normal age-dependent leakiness in normal human populations. So now we have about 36 of these participants, and particularly CA1 region and dentate gyrus are affected. As you can see, increase in K-trans, in permeability, while other regions are not. So white matter is fine, and all other regions are not. So then we compared this with MCI patients, which are with mild cognitive impairment, and also MS patients, which do not have cognitive impairment, but just young patients. And as you can see, there is a progressive increase in hippocampus of blood-brain barrier permeability. In MS patients, there is increase in white matter. I don't show you that, but there is no any changes in the hippocampus. And so this is now about 60 patients altogether. And so we are preparing a revision for our paper that we hope to submit in a month or so. And this relationship holds in CA1 region, dentate gyrus. So these are the most prominent regions associated with cognitive decline. Now, we heard a lot about pericytes, and one model of measuring pericytes is soluble PDGFR-beta, that is pericyte marker, that we show in this group of MCI patients is increased and correlates with increase in blood-brain barrier permeability. And we also did 2 animal models, one pericyte deficient mice and one APP mice, and we did both lose pericytes, and we show also that this pericyte marker is increased. Now, molecular MRI is something that we heard from previous speakers is quite important to develop for monitoring neurovascular inflammation. And my French postdoctoral fellow, Axel Montaigne, has developed this molecular MRI method with MPO muon particles connected with alpha VCAM. So we measure this inflammation in models of ALS, for example, and this is something that I think is important for the field. Finally, I want to mention that our previous work in ApoE, studying the effect of ApoE genotype on a blood-brain barrier, has shown that ApoE4 leads to blood-brain barrier breakdown by activation of cyclophilin A pathway, cyclophilin A MMP9 pathway, and degradation of the Thai junction. But ApoE3 or ApoE2 and murine ApoE can block this effect through LRP receptor and can block the pathway. So this is basically what we showed in mice that was published by Bob Bell, my former student, who is now in Pfizer. And we also showed that these changes happen very early. The blood-brain barrier changes with uptake of, let's say, IgG here, or thrombin and fibrin in neurons, they happen very early at 2 weeks of age. While neuronal activity, for example, cortical depolarization, is completely preserved and show changes much later at 4 months of age. So that's again an example that vascular blood-brain barrier problem precede problems in neuronal function. And this has been repeated in human study. Now, in 90 patients, we first published paper, actually my graduate student published paper in JAMA Neurology, in 50 individuals showing that BBB breakdown is associated with activation of cyclophilin A and MMP9 findings in analytes in the CSF. And interestingly enough, on a larger number of samples, we also showed that E3, E4 patients have correlation with age and blood-brain barrier permeability to albumin quotient, not E3, E3. And this is related to increase of MMP9 and also increase in the endothelial VCAM, for example, marker, but not, for example, astrocytes marker. And we've done all other different markers. So that's about all what I wanted to tell you. And lessons learned are that pericytes lead to BBB disruption, capillary degeneration, and blood flow reduction dysregulation. This leads to white matter lesions, pericyte degenerate in AD. There is blood-brain barrier breakdown in aging human hippocampus and pericyte degeneration, and so implication for neurodegeneration in AD is something which I think would be interesting to study. And ApoE4 BBB breakdown that is mediated by this pathway is interesting also because restoring this pathway in animal models restores the BBB and neuronal phenotype. Obviously, all about markers, I think we need to use in human studies, different markers, vascular markers, tractography, molecular markers, A-beta, and study people at risk for Alzheimer's disease, familiar, and also vascular cognitive, and similar studies in animal models. And this is a wonderful group of people that work in my lab, plus collaborations with Caltech and USC ADRC and neuroimaging group. Thank you very much.
Thank you. So we have time for some questions, quite a few actually. You don't have to buy drinks tonight, by the way.
Okay. Maybe somebody buy me.
Yes, I'll give you one of mine.
Okay.
So that's it.
Yeah. Oh, okay.
That was nice. Can you comment on the kind of the distribution of the pericytes along the cerebrovascular tree?
Yes.
And the difficulties with the fact that at the arteriolar level, you have as you transition to the capillary, you have a mixture of smooth muscle cells and pericytes, which may be difficult to kind of sort out.
Yeah.
And so, can you kind of comment on the complexity of this model?
Sure. Well, I think that's a really crucial question. I know when we first showed this in Neuron in 2010, we showed that about 70-80% of capillary wall is directly invested by pericytes in direct connection, and at that time. Then after that also, Martin Lauritsen and David Atwell showed the same, and then many labs reproduced that. So at the capillary level that are 6 microns, there is no question what it is. Now at 20 microns also there is no question it's smooth muscle cells. But what happens between 20 and 6, and where is this lovely transition happens when pericytes comes closer to his cousin, vascular smooth muscle cells, where is the transition of phenotype? I think this is a dynamic process and really depends on the precapillary sphincters, depends on many, many other roles, and we don't know much. We are trying to construct, by using STP tomography and Imari's software, the branching of all these vessels to figure out what it really is and see if we can do it using NG2 red mice, for example. But it's work in progress. But wonderful question, no definitive answer. For lower and upper part, yes. In the middle, we don't know.
Thank you.
Yeah, here. Very elegant. Two short questions. One is, can you comment if you looked at astrocytes in your pericyte deficient mice? Was there anything unusual about astrocytes?
Yes. Actually, we looked at that, and also Christer Batskell's looked. And Christer reported, we didn't report it, but we also saw the same. Pericytes are important for bringing the position of astrocytes and feet to the wall. And astrocytes communicate with pericytes mostly, direct contact that we show in that Nature paper and ApoE paper, but also directly with endothelium. So if you don't have pericytes, this opposition doesn't exist, and there is a huge space between astrocytes and endothelial wall. So the loss of orientation of pericytes and feet.
Okay. And the second question, so we know that pericyte coverage increases during brain maturation, right?
Yes.
From those almost none in fetal brain and then gradual development.
Yeah.
So, and when pericyte coverage is relatively low, the integrity is still in place.
Yeah.
What do you think about this?
It's a very interesting question because the coverage is not proportional to the loss of the cells. Because pericytes do have what is called plasticity, so they can throw 50 processes, or they can throw 100 or 150 if necessary. So you can have loss of cells 85% or 90%, like in ret model, pericyte deficient, but loss of coverage is only 70%. So it's a huge, and similar if you have loss like 15, 20% in coverage, you can have loss of pericyte 40, 50%. Amazing plasticity. We don't understand everything, but it's amazing plasticity.
Thank you.
Next question.
Sarah Tolonai from Heart, Lung, and Blood Institute. I was wondering, how does the pericyte deficiency manifest itself in other organs? Have you looked at that?
Yeah, we looked in this model. We did systemic. We did analysis of complete renal function, liver function, etc. In these models that we used, that are moderate or modest pericyte loss, we don't see any systemic problems. So now, why is that? Obviously, we asked. It is possible that the ratio of pericytes in brain and eye is 1:1 to endothelial cells. In other cell types, if you read older literature, and I don't know the newer one, there is no much, there is a 1:10 ratio, for example, in heart, or 1:100 ratio in skeletal muscle. So it's less affected than in others. But
so we have time for one more question.
Okay, thank you.
Betsy, hi, Randy Marshall from Columbia. How are you?
Hi, very good.
Thank you for the talk. So you showed a progression of the blood flow changes that may actually precede pathological changes. Do you think this is a common instigator for both processes, or is this a cause and effect producing the clinical manifestation?
Yeah, it is very interesting. It's an interesting question. I do believe that the blood-brain barrier breakdown at first gets leakage of things like fibrin that are vascular toxic, and then you have now degeneration not just because of loss of pericytes, but you have a positive feedback of this toxic elimination. So when this happens, it's very difficult to mount any angiogenesis vital response, so the blood flow goes down. So I personally believe that blood flow changes follow, but also according to our recent work and David Atwell's work, it's possible that they happen in parallel as well.
Thank you very much. Okay. So our next speaker is Dr. Gregory Del Zoppo, and he will tell us about microvessel cell matrix adhesion receptor matrix interaction in permeability, barrier, and barrier integrity.
Thank you very much, Margaret, and thank you to the organizing committee for asking me to speak on this topic. And thanks to the Institute for support over the last years. Finally, thanks to, and probably first of all, thanks to our group in Southern California at Scripps for much of the input in the non-human primate work, as well as some of the cellular work, and in Seattle at the cellular level. This is sort of the knowledge circa 1977 of the vasculature, and it's, as you see, it's basically the intricacy of the neuronal interconnections, and it was appreciated some 100 years ago that the vasculature was very important in its application of to neuronal behavior, but the force that drove us to look specifically at the microvasculature came out of acute work, and specifically out of reperfusion of large artery occlusions in ischemic stroke. And what's clear is that following reperfusion, the downstream vasculature could be reconstituted, but the question was what happens to vessels under the size of 100 microns that could not be imaged by these procedures, and what happened to the cells around them. And virtually at the time of this acute interventional work, there was very little known. What was appreciated, however, in the 50s and 60s and beyond was that the cerebral microvasculature had a particularly complex setup, is a high flow, low pressure system, and an occlusion within the system, and the branching of the system, particularly in the cortex but also in the striatum, allowed flow to reverse and not produce downstream ischemia to a threshold. Furthermore, the structure of these vessels is not as simple as an endothelial cell on a matrix, but includes the brain itself, and specifically the astrocyte end feet, as Mike Nedergaard has pointed out. So the vasculature, the microvasculature, it contains two cell types bounded by an intermediate matrix. The model system that was used for our initial experiments was intended to look at acute problems, that is, acute events that took place following occlusion of the middle cerebral artery in the awake animal. And then specifically because of the vascular arrangement, the neuroanatomy arrangement, as well as the hemostatic arrangement in these animals, they could be used to model a human disease, at least to a limit. This is in the Papio species. I would say one thing about other species, and that is that they have other advantages that this species does not. So we spent a considerable amount of time looking at the intravascular events that take place, and some of those have been described today. They include leukocyte adhesion, events that lead up to occlusion, thrombosis, and hemorrhage. And what was interesting to find was that these events would be initiated immediately upon cessation of flow in the awake animal, suggesting that while there was neuron injury, vascular dynamism was also at play, and this was not appreciated. So for the purpose of this presentation, I'm going to focus on the events within the cell wall itself, and these are virtually silent, inasmuch as the microvasculature in most organs does not announce itself until fairly late when the bulk of microvascular disease has play clinically. These events I'm going to speak about are basically below the level of the radar that most of us have. And they became very interesting because the timing of these events matched pretty much that of neuron injury within the field. So this is a diagram demonstrating some very simple experiments early on. The territory of interest was the lenticulostriatal branches of the middle cerebral artery, so the proximal middle cerebral artery occlusion producing injury in the striatum. It was clear that one could use DUTP incorporation techniques to define cellular injury as a core, and then around the core one could sample and see what events could take place there. And what's important to know is that within 1 to 2 hours of the onset of ischemia, the vast majority of cells that were DUTP incorporating were neurons. And vascular cells did not present until much, much later, when about 2% of the cells at 24 hours were endothelial cells. So endothelial cells are really resistant in vivo to the kinds of injury that we would hope to see clinically. The second point to be made here is that this is an awake primate model. This is an anesthetized rat. So the timing and the duration, the unfolding of events that take place is anesthesia dependent and probably species dependent as well. So it is sort of a tangential to our own exploration of other things, but we began to wonder whether or not adhesion of these cells, how the endothelium is tied to the basal lamina, may be important in how the cells respond, and particularly how the blood-brain barrier evolves its injury. We knew well that tight junctions were an important element in this, but the question is, can you have a tight junction if you don't have a cell that's adherent to its base? And so of consideration were the 3 beta-1 integrins, alpha-3, alpha-1, and alpha-6. And this is just an example of what happens with alpha-1 beta-1 during ischemia. Within the first 1 to 2 hours of the onset of ischemia, there's a significant loss of beta-1 and alpha-1 integrin expression in the core, and that's seen in this region here. And there's a somewhat more modest but still significant reduction in the area where there is no real clear evidence of neuron injury. That is to say, there must be some putative injury, but in the peripheral zone there is some degree of beta-1 loss. In fact, if one looks at all of the beta-1 integrins, except for alpha-5, there's a significant loss in this area. And in the same vessels, or in vessels nearby, there's an increase in integrin alpha-V beta-3, a pro-angiogenic signal, which is important, as we can point out later. The relevance of these structures is that they tie the vessel together, but they're also signaling molecules. As you know, integrins are involved in either outside-in signaling the environment to the cell, or inside-out signaling how the cell will respond to other cues. So these are both structural and potentially signaling molecules, and they're pretty much manhandled by the ischemic event. So for all intents and purposes, we can look at beta-1 integrins as a family, realizing that they're not really identical from one alpha to the other. Second, another point to be made is that in the ischemic territory, that is in the core, as marked in these 3 animals, one sees a loss of beta-1 in the center of the core, but around the periphery, there's an increase in beta-1 transcription. Now, the alpha partner to this is not known, but a good candidate would be alpha-5, which is important for angiogenesis. Well, what about the astrocyte side of this? You can't think of the endothelium in the vasculature of the brain without thinking about the astrocyte. And we should say, parenthetically, the neuron which is beyond that, to which the astrocyte interacts or speaks. So this is the vascular part of the neurovascular unit, of which the neuron, which you don't see here, is also a player. So in order for the vasculature to work properly, it would seem that the infeet would also need to have a strong and firm interaction with the basal lamina. These adhesion receptors involved here are integrin alpha-6 beta-4 and alpha-beta dystroglycan. Now, in GFAP positive preparations, looking at alpha-6 beta-4, one could see a number of vessels where alpha-6 beta-4 was co-expressed on the end feet, but a number of other vessels where there was no alpha-6 beta-4. And this turns out to be territory specific in the brain. For instance, this is a good example of the cortex in which all microvessels of every size, from capillaries all the way up to large vessels, express alpha-6 beta-4 on the astrocyte end feet. But only a small proportion of these express integrin alpha-6 beta-4. So this is alpha-beta dystroglycan, rather, alpha-6 beta-4. In the white matter, however, this is a 1-to-1 correlation. So every microvessel has both alpha-6 beta-4 and alpha-beta dystroglycan. What these 2 molecules are doing is not yet worked out. But there is some idea about alpha-beta dystroglycan. during focal ischemia in the primate, within 1 to 2 hours, there's about a 40 to 60% loss in expression of alpha-beta dystroglycan on the astrocyte end foot. Now, it's important also to note that the astrocyte is the epithelial cell of the brain. It adheres to the basal lamina much in the same way that the skin adheres to the basement membrane. But indeed, there is a rather rapid and remarkable loss of this expression. This can be mimicked in vitro using primary mouse astrocytes. This is C56 Black 6. They can be grown on wells or on inserts. And here's an example of an oxygen glucose deprivation experiment in which there's a loss of alpha-beta dystroglycan, in this case beta dystroglycan, which is not affected by inhibitors of proteases, including a protein and the cathepsin L inhibitors, but is affected, in fact preserved by inhibitors of MMPs. It's GM6001 and 110 phenanthroline. And it doesn't matter whether the cells are grown on collagen 4 or laminin 1. It appears as though there is a protease-dependent loss of beta dystroglycan, which is associated with this change. Now, why is this important? Well, it turns out that it's important because glial cells, which are banging up against the basal lamina, detach from the basal lamina during ischemia, as shown in this 6-hour preparation from the Sprague Dolly rat. And the glial cells detach, and there's interstitial fluid which increases between the basal lamina and the astrocyte. This is an observation that's about 45 years old, but now has a potential explanation. Well, what about the basal lamina itself? And here I'll try to be brief. It was Gerhard Hamann in our group a number of years ago who demonstrated very clearly that within 4 to 24 hours after the onset of ischemia in the primate, there was a significant loss in collagen 4, laminin, and fibronectin content of microvessels, that is predominantly capillaries. And there was, this coincided with an increase in pro-MMP2 expression in this system. Whether or not the pro-MP2 was activated and caused these changes was a question at large. But what's important to know is that this is not the only protease that's generated. In fact, while pro-MP2 expression was also correlated strongly with neuron injury within the territory at risk, other proteases also generated, in this case by endothelium, urokinase, were not so associated. It's possible to make a number of biochemical pathway guesses at how this works out, but suffice to say how these particular MMPs and how members of the other families of MMPs, serine proteases, cysteine proteases, and heparinase are upregulated at the same time, interact with each other, is still unknown and unclear. What's important is that, among other things, is that the microvascular expression of these matrix proteases occurs at the same time as neurons are found to express them as well. An important caveat, again, is that as Eng Lo and I showed some years ago, that the expression of these proteases in the baboon, in this case pro-MP2, may appear different in another species, such as the mouse, where it's MMP9 that's upregulated following focal ischemia. So beware of species of questions. So this leads to the next question of how these are expressed, and this is a hypothesis, a cartoon based on data from our group that demonstrates the co-expression of heparinase, cathepsin L, which I'll allude to a little bit later, pro-MMP2 and urokinase and its receptor on both neurons and on the vasculature, suggesting that for some reason yet unknown, these two structures, the neuron and the astrocyte endothelial portion of the microglia, portion of the microvessel, rather express these compounds. So directions of inquiry that resulted from this were to determine the relation of beta-1 integrin expression to permeability status of the endothelium, and whether or not alpha-beta dystroglycan also plays a role. So I'll try to speak to this swiftly. The endothelium, as you've already heard, is bound together by tight junction proteins in the CNS, and our concern was whether or not these structures, these adhesion complexes, were also important in maintaining the barrier, and whether or not they served for the barrier itself. These three components of the barrier we would now propose are the interendothelial tight junctions, the basal lamina, and the third component would be the adhesion receptor matrix interactions. To look at the beta-1 integrin matrix interaction, we chose an in vitro system using primary murine endothelial cells and astrocytes. These were grown on inserts. I should point out that these cells are plastic sensitive. It depends upon the manufacturer of the plastic and on the matrix protein you grow them on. We use both video imaging techniques, permeability studies, immunoblot studies, as well as flow cytometry studies. So a very simple example is using an inhibitor of a beta-1 integrin. We could show a significant decrease in Claudin-5 expression, the tight junction protein. When beta-1 was blocked in confluent cells, this produced a decrease in the expression of Claudin-5, but also an increase in permeability of the system to both 40 kDa and 150 kDa molecules. This was also true of other tight junction proteins, including Claudin-5, occludin, and Z01. And if injected directly into the brain, this produced a significant increase in permeability. So blocking beta-1 integrin increased permeability across the barrier. An important new piece of information is that in vitro, the loss of beta-1 integrin by oxygen-glucose deprivation or ischemia requires the presence of astrocytes and also serum. So this reduction is most significant when OGD and serum are combined. I think these are mislabeled. And the important point of this is that when plasma percolates across the blood-brain barrier into the neuropill, it's essentially producing serum. So these are serum-free conditions and with serum. So blockade of beta-1 integrin produces a decrease in endothelial tight junction protein and an increase in permeability. Ischemia or OGD produces significant loss in beta-1 integrin. And then finally, I wanted to make a mention about astrocytes. Brian Hawkins in our group looked at the question of alpha-beta dystroglycan blockade. And here again, an alteration in water transit occurred. I can just summarize to say that OGD decreases the rate of water uptake in astrocytes in vitro. There's a direct blockade of alpha-dystroglycan on enteric cells, mimics the effect of OGD on water uptake. This is ERK phosphorylation dependent. And finally, inhibiting the proteolysis of dystroglycan or ERK activation preserves water uptake in these cells. Signaling, I'm not going to speak to, but this is the next step. And then finally, with percolation of plasma molecules into the neuropill, there's an activation of other cells, which I haven't talked about, including microglia, which can make MMP9 and also cathepsinellin. So lessons learned, there are many, and these can be summarized very quickly to say that microvessel responses and endothelial responses, as well as astrocyte responses to ischemia, are everywhere as rapid as neurons. Finally, that the astrocyte function is matrix adhesion dependent. Endothelial cell astrocyte modulation is clearly operating, but we don't know how. Implications for signaling events and clinical events is quite clear, but also there may be potential treatment possibilities. Gaps to be filled are numerous, but I'll just focus on three. One is these are acute interventions, but what's happening in the chronic situation of, let's say, lupus neurovascular disease or amyloid deposition? How are these receptors and how is their interaction of the cells with the matrix altered more or less permanently or progressively? The second is communication within the vascular segments and signaling across the barrier, that is the basal lamina barrier. And finally, what role does matrix protease generation play in all of this? Okay, thank you very much.
Thank you. We'll take some questions. You owe me 2 drinks.
So I'm Elizabeth Hillman from Columbia University. The talks today, it's been really amazing to see everybody's picture of a cross-section of a blood vessel in the brain, because we've seen so many different layers from, the endothelium. Some people have a parasite there. Some people have smooth muscle. We've seen the lymphatic system. We've seen the astrocytic end feet. We've seen all sorts of different things. And this has puzzled me for many, many years, because in the brain, you go from peel arteries, peel vessels, which don't have the glial end feet, to the diving arteries, to the capillaries. And here we are all trying to figure out what's going on from the inside out, from the outside in, how vessels change their diameter, what's the blood-brain barrier. And we don't seem to have any sort of common language or common agreement on what this thing looks like.
Well, I think it's a good point. I mean, some people are smarter than others. That's number one. And other people are just more resistant, and some people have their own kind of thing they look at. I made the schematic without the parasite and without the vessel wall histiocyte, without mast cells, without microglia, which I mentioned at the last, and oligodendroglia, which clearly play roles in how all of this is generated. The purpose for our work was to look at the acute situation. So here, these cells do play roles, the other cells I mentioned. But yeah, you're right. You're absolutely right. I think to schematize it, endothelial, basal lamina, astrocyte, this part of the neurovascular unit is still not worked out in terms of how the communication should take place. And it does, and it's pretty rapid. But your point is well taken.
Next question.
Tanya Mayadas from Boston. I was just wondering, because Elizabeth DeJano's group showed many years ago, like about 20 years ago, that these integrins, the beta-1 integrins, may actually be at not just cell matrix, but at cell-cell contacts.
Yeah.
And I was wondering if you've looked at that in your vessels.
Yeah, we did. This is an interesting observation because I'm not so sure it doesn't depend on the organ system to some degree and the preparation. We cannot exclude the possibility that there's a low level of beta-1 interaction between cells, endothelial cells, in vivo. What happens in vitro is very much dependent upon the setting. Jordan Pober showed very nicely a little bit of hydrogen peroxide removes the polarization of beta-1 integrin from the base. And I'd be willing to take the criticism that we know nothing about alpha-beta dystroglycan polarity on the astrocyte. I mean, these cells, it could be everywhere. So I think it's a point well worth looking into.
Last question over there.
Howard Crystal, Brooklyn. I don't understand something. Is the basal lamina the same space that the lymphatics are in? And if so, how does all that transport happen through all these proteins?
Well, the person you should talk to is sitting in the front row here about that. But I will say this. Lydia Sorokin in Münster has shown very nicely in the last 4 or 5 years that the chemistry of the basal lamina is actually quite complex. And it depends upon where you are in this vascular tree, from the peel penetrating vessels all the way through the cortex down into the striatum. And indeed, the let's call it the abluminal side of the endothelial basal lamina and the astrocyte portion of that basal lamina have a different composition, and they fuse in the region of the capillary. And I think you've already heard today that the capillary has something to say about CSF production. So how this is all worked out, I think, is probably not entirely clear. And you're raising a very important point, and that is that the composition and structure of that part of the vasculature along the whole tree is going to be very important to investigate. Fair enough?
Thank you so much. Okay. Our last but not least and final speaker for the afternoon is Dr. David Antonetti, University of Michigan, and he will talk about mechanisms of blood to retinal barrier permeability and potential links to angiogenesis.
Well, thank you first to the organizers. A really interesting and exciting format to talk about blood vessels. And it's been particularly, I think, great all day. I've gotten a chance to see people that we normally don't get to see, people I've had a chance to read for many years, and now I get a really chance to interact with. So congratulations, and thank you very much for letting me participate. I'm going to try, obviously, in this very short amount of time to speak broadly and use schematics for a lot of what has been published, and then talk about some new work. So we're particularly interested in the blood-retinal barrier, and particularly as it relates to diabetic retinopathy. And here is what's a fundus photograph of what a clinician would look like with a patient with diabetic retinopathy, and the problem is this central macular edema, which is very closely associated with loss of vision. We have these microhemorrhages, lipid exudates that are occurring. And if you do a fluorescein angiogram on the right here, so just perfusing fluorescein, and it tells you a lot. You can see a lot of the changes. So we see these blood vessels that leak. We can see this leak at the optic disc. And then we see some of these very interesting regions. We see regions where there's very low perfusion of blood vessels, and beside them, we can see these hyperpermeable regions that are associated with neovascularization. And so some of the questions that we have is how are these blood vessels leaking and how is this proliferation occurring? Of course, I'm sure everybody here knows vascular endothelial growth factor, or VEGF, is an elevated factor in this disease process. And by delivering anti-VEGF therapies into the vitreous of the eye, about half of patients are showing a good responses, and some even improving in vision, and many having less loss of vision. So we can reduce the permeability in clinical studies looking at this angiogenesis. So, you know, a lot of the perspective so far, and a lot of what I'm going to talk about today, has been how do we block the pathology moving forward. And in a nutshell, the question that I would have going forward for our group is, can we, as we start going forward, thinking about changing that and asking, instead of blocking this neovascularization, why is this pathologic neovascularization rather than a reparative vascularization? And how can we potentially promote a reparative vascularization? So I just basically said that. This is a schematic from a relatively recent review from Harris and Nelson. And Dr. Malik very beautifully talked about a number of these parameters. This is looking at adherence junctions. And this makes a really important point. So Dr. Malik talked very much about this side of the picture, which we see lots of permeability factors that signal through many of the well-studied events have been through kinases, tyrosine kinases, as well as serine kinases that can phosphorylate proteins such as V-cadherins or related catenins, and these are associated with permeabilizing events. Endocytosis appears to be a major part of this process. So that's going on. But this side of the picture shows another important point, which is that this adherence junction complex then impacts the endothelial cell and is also a signaling center in that endothelial cell. And so V-cadherin can downregulate, in a complex, can downregulate VEGF receptor signaling. These adherent complexes can regulate cytoskeletal organization through G proteins, beta-catenin, or V-cadherin through FOXO can regulate tight junction, particularly Claudin, like Claudin-3 expression, and Claudin-5 expression, I'm sorry. And beta-catenin can regulate Claudin-3 expression, regulating barrier formation. So we have signaling going to the adherence junction, regulating permeability, but also the adherence junction can act as a signaling center, affecting things like cytoskeletal regulation and junction formation. We'd like to argue that the tight junction complex also plays a similar role in addition to the adherence junctions, particularly in tissues like the blood-brain and blood-retinal barrier. So there's lots of data that suggests that occludin cloudins are altered in expressions. A lot of this data comes from epithelial cells in cancers. Very interesting data about ZO1, a junctional protein that moves to the nucleus, and ZO also being associated with growth control, specifically through binding proteins such as ZONAB and CDKs, and ZO2 also binding other regulatory proteins in growth control such as human phosphate. Our favorite tight junction protein that we spend a lot of time on is occludin. It's probably a misnomer. It probably isn't really involved in creating a barrier. Knockouts have been made and animals live, but they have a host of defects. And these defects include some cells, mucous cell hyperplasia. Occludin has also been shown to target TGF beta receptor to tight junctions, be required for epithelial to mesenchymal transition. As I said, it's lost in a host of epithelial cancers. And very interesting studies showed transformed PA4 cells can be reverted by overexpression of occludin. And we've shown in ARP19 cells, siRNA to occludin also leads to proliferation. So a number of years ago, our lab and a number of other labs had identified it, started working to identify post-translational modifications, specifically phosphorylation sites. In red are serine-threonine phosphorylations, and blue are tyrosine phosphorylations. And we're just learning about how all these phosphorylation events lead to regulation of cellular function. And what many of us are coming to is that occludin is not really occluding at the tight junction, but a regulator of barrier properties and has a variety of signaling processes in the cell. So just to point out a few, serine 408 was studied by Jerry Turner's lab and was critical in forming a cloud and pore, which allows permeability very specifically to ion flux. And one of the things that, from our perspective as a tight junction biology lab, we've noticed today is that we talked about permeability in general, but permeability is very specific. There are different routes for different kinds of molecules, and this was actually a very good example of a very specific pore formation allowing ions that wouldn't allow large solute flux. Tyrosine 471 looks to be pro-barrier. Phosphorylation looks to be required for assembly of the junction complex, and that's an area of ongoing work. This talk today, we're going to focus on serine 490 phosphorylation. It's in this region of the coiled coil terminal domain of occludin, and it's right on one of these turns. It's a PKC target, much like we know PKC alpha target of V cadherin for permeability control. This also appears to be a classical PKC target. So this is a rather complex slide, but I just highlight a few points. This is endothelial cell culture looking at permeability in response to VEGF, and we see our typical twofold response, and this is looking at large solute flux on the kilodalton dextran. If we put in PKC beta, we get an even larger increase in response to VEGF. And if we overexpress a point mutant at serine 490A of occludin, we can completely block that VEGF effect, even if we're overexpressing PKC. Conversely, if continuing work showed that this phosphorylation led to a subsequent ubiquitination and endocytosis of occludin. Very much like the cadherin undergoes endocytosis, we think both processes are occurring in parallel. And if we put in an occludin ubiquitin chimera, we could make the cells constantly leaky, even if we had a dominant negative PKC present. So this led to a model in which we believe occludin is phosphorylated in a PKC-dependent manner, leads to ubiquitination and internalization, in fact, able to show co-precipitation with a number of these ubiquitin interacting motif-carrying proteins that act as chaperones, and endocytosis of occludin and other junctional complex. But we had an interesting surprise, and VEGF, of course, is not only a permeabilizing factor, a proliferating factor for endothelial cells, and with our phosphospecific antibody, we would notice cells with condensed nuclei that were actually going into mitosis. You can see here the centrosome for gamma tubulin, and we can see very clear staining for the phosphooccludin at the centrosomes in these cells as well. And all through mitosis, metaphase, anaphase, and telophase, you can see really very nice co-localization with gamma tubulin. If we transfected then endothelial cells here with the GFP tag, we could show the GFP tag co-localizing very beautifully also with the centrosomes, and here the anti-GFP antibody around this condensed nuclei. So it wasn't the antibody binding something else, it really was occludin. On the left is a patient that had to undergo an operation for proliferative diabetic retinopathy with an epiretinal membrane. So here, this is this neovascularization growing into the vitreous. It was surgically removed. And from that surgical sample, we stained and were able to see phosphooccludin that perfectly co-localized with centrosomes as well. So we had this unique localization for this tight junction protein. We looked to explore this more, used the tube formation assay. So these are endothelial cells in culture and inducing tube formation with VEGF. And so you see over time, 24 hours, this tube formation. And over time, we see this increasing occludin serine 490 phosphorylation. So does this occludin phosphorylation significant to the process? So if we transfect cells again with this 498 point mutant, the same point mutant that blocked VEGF-induced permeability, you can see we were able to completely block this tube formation in response to VEGF. So here's wild-type occludin. It allowed tube formation just fine. The 490D mutant allowed, phosphomimic allowed tube formation, but the 490A completely blocked tube formation. That's shown quantitatively here. It also blocked endothelial proliferation, so using incorporation of either DUTP or other methods, we can have the VEGF-induced proliferation. Wild-type occludin actually showed a slight reduction, and the 490A completely blocked the VEGF-induced DNA synthesis. Conversely, if we knock down occludin in these endothelial cells, and this is knocked down here, we see a basal increase in proliferation, a basal increase in tube formation, and an increase in VEGF-induced tube formation as well. So it appears that occludin puts a block on the proliferation and tube formation that may be released by phosphorylation and potentially in a PKC-dependent manner. Looking in vivo, We used a model developed by Peter Campicero in which VEGF is expressed in an inducible manner from the outer retina. And so we can induce this, and when we do that, we cause the vasculature to grow down into the outer retina. It's so dramatic, the retina actually will come off in about 4 days. If we look at that, this is a picture, just an IB4 staining cross-section of the retina. We see this increased vascularization occurring. And if we look at this serine 490 phosphorylation, we see again the dramatic increase in phosphorylation quantified here. So what are these vessels like? And they are hyperleaky, just like we might expect with a pathologic neovascularization. So I'm not going to describe this because I don't have a lot of time, but the NHS biotin assay to identify regions of leak. And if we're looking at the deep capillary layer, this is in the control. It's very little leak, but after the VEGF induction, we see a very dramatic leak. And if we look at the occludin localization in this deep capillary and the ZO and the control, it's nicely organized. And after DOC's induction of the VEGF, that occludin is really, really lost in its organization as well as O1. So finally, can we reverse this in vivo? We worked with Dr. Bill Hausworth, who has a AAV virus that is able to transduce specifically endothelial cells, and we were able to do this in the eye of mice, and we use this VEGF induction model. So if we just put in GFP without DOCs, we see the IB4 staining of blood vessels right where they're supposed to be. If we put in the GFP and induce the VEGF expression, we get lots of angiogenesis, pathologic angiogenesis. The wild type occludin did not block that pathologic angiogenesis, but transduction with the S490A point mutant of occludin really blocked that pathological angiogenesis or neovascularization. And quantified, of course, in a mass fashion, we were able to show that we really completely blocked that neovascularization in vivo. So we think that in a short time frame, VEGF induces an occludin phosphorylation, we think at the junctional border, that's associated with endocytosis in a pathway that's probably very much parallel, maybe likely integrated with VE-cadherine endocytosis, and that creates breaks and permeability, large permeability pores that would allow things like dextrans and albumin to flow through and increasing permeability. This is maybe a 30-minute time frame. And then after a 12 to 24-hour time frame, we see again an even stronger phosphorylation of occludin. Here we see it very much associated at the centrosome, and this is associated with proliferating cells and appears to be a required step. We don't know exactly how yet, but it may well be associated with orienting the cell. An effective angiogenesis, especially with cells that have these junctional complexes, really may need to maintain an orientation, and we think that that might be what's involved here. To be very precise and brief, our long-term goal then is, we've been blocking this process. We'd really like to know how we could go from here back to a quiescent vessel. So how can we promote a reparative system? So lessons learned, the junction complex may be regulated by translational modifications to provide a specific alterations in permeability. And formation regulation of the junction complex is intimately linked to growth and differentiation. And junctional complexes may regulate proliferation, migration, and angiogenesis, not just from our work, but from a host of other laboratories that have been showing both occludin, ZOs are involved in regulating the growth process as well as the barrier process. And as we said, gaps in our knowledge, where can we go? Can we think more about understanding reparative angiogenesis? And I thought Dr. Davis brought this up first thing in the morning. I couldn't agree more. Potentially by studying normal developmental angiogenesis and comparative analysis to pathologic angiogenesis, we can identify important differences and learn how to promote angiogenesis that could be effective. And the last point, I didn't get a chance to show data or slides, but with our keynote seminar from Dr. Aird, we completely agree. We see very interesting differences between the different vascular beds. For example, this occludin phosphorylation seems to be very much restricted to capillaries, particularly in ischemic reperfusion in eyes, while we see very different changes, but very important changes in the junctional complex in large vessels. And so there appears to be very important differences that are not going to be apparent by smashing up the whole tissue. Further understanding the specificity of the vessel changes in response to these pathologies, I think, is going to help our understanding greatly. Thank you very much.
Thank you. We'll take some questions.
Dave, really nice work. Congratulations.
Thank you.
Just comment and then a question. So there's good precedent to indicate that post-translational modification of junctional components, i.e. a breakdown of the junction via proteolysis, is a means by which you can activate transcription, for example, in Notch or in catenin signaling, where pieces of the junctional components actually transmigrate to the nuclear compartment and actually stimulate mitosis or other gene transcriptional events. Do you know, have you looked, is the VEGF-dependent phosphorylation on occludin at S490, is that representative of the intact molecule, or is that actually a piece of the molecule?
Great question. I'll just start by saying, we've done some evolutionary analysis of the molecule, and really occludin looks to be a joining of two parts. The transmembrane is in fact, it's part of a family of a MARVL, and MARVL D3 is just the transmembrane part. And along cytoplasmic tail, and the coiled coil looks like it's also called the L domain, and it looks like it's derived from another, and it looks like it was a fusion event over time. We were sure that the coiled coil was going to be cleaved and moved to the centrosome, but we did do a centrosome prep, and we do see the whole occludin at the centrosome. So, we're still trying to understand. There are some other transmembrane proteins at centrosome, so we're not out there alone, and we're trying to understand that.
Thanks very much.
Yeah.
Thank you. Next gentleman over there.
So is it a common feature of the process of the choriocapillaries versus the retinal blood vessels?
Oh, that's a great question. We don't have an answer. So far this has been focused on cells that express a high degree of tight junctions and occludin. I don't know. I think that's a great question. We haven't done anything to ask the question about the choriocapillaris, which is just below the retina and supplies all the outer retina, and part of AMD is the angiogenesis coming up. We have no idea if occludin plays a role in that vessel bed or not.
But after angiogenesis, could they maintain the junction? What is the nature? Because most of the neoangiogenesis process make leaky vessels. So these junctions might be very leaky.
I completely agree. We think these junctions are very leaky, and I think one of our really important questions is to understand why in development that doesn't happen, and why in these pathologic conditions this is very different kinds of angiogenesis.
Okay, we need to move on. Next question, please.
Very nice talk.
Thank you.
My question is about the tight junction that induced by the VEGF signaling towards contact inhibition of endothelial cells, and whether this is conflict with this potentially growth inhibitory factor for you. You see the growth promotion effect.
Do I think it's different? I don't know.
Do you think contact inhibition is different?
In our hands, endothelial cells have some degree of contact inhibition, but it's not nearly as powerful as epithelial cells and epithelial cell cultures that can really shut down really tightly with a contact inhibition. But I'm not sure if I get the essence of your question.
Yeah,
maybe we could talk after. Or should talk after.
Contact inhibition, we will usually interpreting contact with the growth inhibitory effect of the growth endothelial cells, which will be anti-angiogenesis.
Sure.
So there will be conflict of
absolutely. And a recent beautiful modeling paper about angiogenesis showed that the cells in the angiogenic tube have to keep moving, and that junctional complex has to be released and allow those cells to move, and that's an important aspect of angiogenesis. The centrosomal localization we think is important, but the release of the junction appears to be a central part of angiogenesis as well. Yeah.
Next question, please.
Great presentation.
Thank you.
So, occluding knockout mice have brain calcifications.
Yes.
And there is evidence that some patients who have a loss of function occluding mutations also have basal ganglia calcifications. So the question is this a problem just with extravasation of blood leading to secondary, inflammation, and then ends up being into, or there is more of an active matrix remodeling process that affects that?
Yeah, I don't know the answer to that question. I think the brain calcification, and I remember when Dr. Ferrucci first presented, they really didn't have a handle on why that's occurring. I think those are great possibilities, and I don't think we have I think we're just kind of getting to the point now where we're starting to understand that this is not a central barrier, but a regulator of the barrier and how it might do that. So,
Next question, please.
Yeah, I didn't see any cells in your retinal preparations, but
Is this a cellular process, or is the VEGF produced by some other component and then leads to a leaky vessel, or is it actually an inflammatory type process?
In the in vivo system that we had?
The one that you had the fluorescein leaking from.
Yeah. Okay, so it's a system in which it's an inducible VEGF from the rods. So it's an artificial system. It represents a specific type of AMD with retinal angiogens. Normally AMD would be from the choroid. This is a very specific subtype, but it's an excellent model because the outer retina is normally avascular. So when we induce that VEGF from the rod, we induce those endothelial cells to grow down into the normally avascular retina. So we were just using that system.
If you did that in a patient, would you see inflammation, or would you just see?
So I think that's a great question. We are just starting to ask the question of the relationship between the inflammation. I will tell you this real briefly. We've injected VEGF into the eye. We get very little inflammation. TNF very little. We co-inject VEGF and TNF into the vitreous, and we get a bomb of inflammation.
Thank you.
Thank you. Last question.
Thanks. So I'm just curious, one of the things that been missing, maybe just because I'm interested in calcium, calcium signaling is obviously very important in endothelial cells and can dynamically regulate at least their retraction or maybe the endothelial stiffness as well. How would you envision this fitting in? Because this potentially is a mechanism to rapidly change endothelial cell morphology. It's been missing from all the slides I've seen so far.
We think about calcium in terms of, right now, the classical signaling of PKC as a required element for the PKC signaling.
Non-muscle myosin?
I guess I'm not going to, probably wouldn't be best for me not to comment on that.
So maybe I can follow up. There's
Yeah.
actually a really important role that calcium plays in regulating the remodeling of the endothelial cytoskeleton through the work of calpain. M-calpain and mu-calpain are activated locally by calcium, which goes on to cleave cytoskeletal and junctional components that foster the remodeling, stimulate the migration, and induce the proliferation.
Okay, so I would like to have all the speakers at the table, please, so we could carry on the discussion. So this would be like a rolling discussion, right? Right into the bar tonight.
Yes, and I would like to start with the panel discussants, Dr. Ubogo and Dr. Stan, if they would like to give some remarks, if they would like to add something based on the research experience that wasn't discussed today, particularly Dr. Ubogo, if you would like to comment on the blood-nerve barrier, and then Dr. Stan can add on based on his research. Thanks.
Okay. Thank you very much. I think what has been presented today has been really fantastic in trying to understand, particularly the blood-brain barrier and its different components, and how each component interacts with the others in order to maintain the microenvironment of the brain. The blood-nerve barrier is somewhat similar. The big difference, I guess, is that we don't have astrocytes or microglia that provide end feet that contribute to the vascular structure or the neurovascular unit in the peripheral nervous system. So we are still very early stage of trying to figure out how, for example, Schwann cells and the local secretion of soluble factors such as GDNF or VEGF may participate in the development of tight junctions within the peripheral nervous system. I think a very important point that's been brought out from all the conversations here is that there's a very dynamic process that takes place in normal disease, in normal states as well as in disease states, where there's an interaction between cytoskeletal components, adherence junctions, tight junctions, and the cells that support the endothelial cell within the tissue-specific environment, including the matrix, in maintaining a normal function. I guess a big question will be what happens during normal development, and in specific diseases, what perturbations take place that result in the pathology we see.
Okay, so what I would like to add, actually, I would like to congratulate the speakers for a really good and great overview of the field. So I would like to also add that actually things are complicated. And I'm referring mostly to the basal permeability and how that function. Yeah, so we all agree that permeability occurs at the level of capillaries, and you have to have a vessel wall, and the vessel wall is the endothelium forms a barrier. The barrier is maintained by input from pericytes, from the matrix, from the subyacent tissue. Now, for me, permeability is a word, to paraphrase Bill Ayer this morning, he was stressing the importance of terminology. Now, for me, permeability is something that denotes control. So in normal undisturbed tissues, endothelial cells, in cooperation with all the other inputs, exercise this control in which the exchanges maintain homeostasis. What happens in disease or in adaptive processes like in inflammation is the loss of this barrier. So is that permeability? I would really like to throw this out there. I would like to hear some opinions. That to me is leak, is vascular leakage, yes, and that is very, very important. But now, the final point I would like to make is that one, we do not really know how the basal permeability is maintained. Yeah, so we have more than endothelium. We have the basal membrane. We have the glycocalyx that has not been mentioned at all. Now, opinions really differ in the field. You know, by some opinions, the glycocalyx provides the filter, and everything goes through the junctions. On the other extreme, is glycocalyx may provide the filter, but there is vesicular trafficking that actually does the job. There are people in the middle as well. So until we do not really understand at molecular level what happens there and how we can actually modify and manipulate these processes in order to answer the question of permeability and its contribution to physiology. And then Bill Ayer also raised another good point, that we see that hyperpermeability in many disease processes, but is it really the driver? Is it just the collateral? I would end here.
I could just add, from the retina, I find it very, first, there are folks in the audience who understand this far better than I do, Jerry Letty, for example, in the back. But if you look at a retina, it's fascinating that in the disease case, the cystoid formation in diabetic retinopathy is not right by the vessels. It is in the outer retina where we just explained there aren't blood vessels. So clearly the process is not just opening and leak of fluid. We often think about Starling's equation, possibly leak of protein and trapping of the protein leading to an osmotic change. But I don't know that any of that's proven, and I'm not sure what folks think in the brain.
If I may just add a little bit to this, there is a new concept that just started emerging, which is very interesting for the field. For example, there is a paper, 2 papers in Nature published 3 months ago about MSF-T2A, which is actually omega-3 fatty acid transporter that also regulates the blood-brain barrier permeability. So there is a direct liaison between nutrient transport, essential transport, and that. We also have work I didn't talk about GLUT1, which is glucose transporter, that is similarly important for maintenance of the blood-brain barrier integrity. I didn't have time. So I think this is kind of an emerging concept. And in terms of how it happens, it's paracellular, transcellular, or misalignment of the tight junctions. Anything can be.
Well,
finally, right.
I'm not sure I understand Rob's point. I mean, he's intimating, I think, is that permeability may not be a causative factor in disease. I think that's likely not the case, because permeability means a disruption of the junctions, which enables transport or extravasation of liquid and solutes and plasma proteins, and osmotically, I mean, it's a pathogenic feature in edema formation and all kinds of other things, and inflammatory diseases. Now, one of the things I think we've neglected throughout this afternoon is really to define permeability, because permeability is actually a physical term. It's described in units of centimeters second, and so I think we have to get away from the descriptive aspect of defining permeability and really get down, and that may help to address some of the questions.
Gary showed that in his lecture in MS, right? In vascular cognitive, you did show increasing permeability. They measured that.
Well, but transfer is not permeability. Transfer could be due
Can you use the
to
microphone?
convection.
That's the same as a
But transfer, I think, is a different, because you can have increased, I'll give you an example. You can have increased surface area, and thereby you can have increased flux of large molecules, which could be misconstrued as increased permeability. Increased permeability, I think that... Well,
transfer constant is a PS product. That's it.
But
it's that top.
It is a function of surface area. I don't think you can rule out the surface area.
So can I interject a little, 1 minute here? So, so we are talking about permeability as a mechanism of a breakdown of a mechanism as in disease. But that's not the only time that the blood-brain barrier is permeabilized. It's also permeabilized as a response to damage and danger signaling. And therefore, it has mechanism to get cells or cellular factors from one place to another, right? And so permeability, so blood-brain barrier permeability isn't just a breakdown of a mechanism during disease. It's also a response, a homeostatic response to damage.
I think one thing we haven't talked about a lot is the role of transporters. I mean, it's a very dynamic process. I mean, I think traditionally we always thought about, okay, this is a large molecule, it shouldn't cross across the vascular barrier, but there is the receptor-mediated, endocytosis. Depending on the tissue you're working in, depending on the state you're working in, there may be very active transport through transcellular mechanisms. And the question is, if I took my high molecular weight dextran and simply saw more of it on the abluminal side, does that necessarily mean that my endothelial barrier is leaky? Or is there an active transport process because of the microenvironment in which my cell existed?
Yeah, I'd like to address that, because over the years, I found out the important thing to look at is exactly what you're saying, but look at different molecular weight dextrans to tell what kind of process are you actually altering in a disease state or a pathological state or an injury state. Is it transcellular? Is it paracellular? What is going on, number one? Number two is, we talked a lot about degradation, we talked a lot about breakdown. What about repair? Repair is a dynamic system. Repair can happen all the time. And the truth of the matter is, we have the caveoli observations, we have the cavern observations, we have the phosphorylated caveoli observations, and to be truthful with you, they can actually transport the tight junction proteins, and you can have actually a trafficking issue going on, back and forth across a injured, degraded, breakdown blood-brain barrier. You can get trafficked of these tight junction proteins right back up to that point and actually re-anneal that barrier. So it's a dynamic system that takes in all the cell types, but we can't forget the model systems we use, especially this albumin issue with Evans blue. That's a huge molecule. Okay? That's like the hole in the dam and everybody's going to get flooded downstream. It doesn't necessarily have to happen that way. It can happen with a small molecular weight molecule that shows you that you have a small leak. That's very dynamic. And to the point, Betsa was bringing up the point about permeability. Permeability is our index. It's our ability to see. Gary's data is elegant in that. The Patlack plots are real. But the bottom line is that, in my opinion, and this is just mine, and I've been doing it most of my career, I guess, and I'm getting old. The bottom line is, what about repair? It's not just a break and then dike. This is something that's been going on evolutionary for years. So the tight junctions are involved, the adherens junctions are involved, the parasites are involved, the glia, the microglia, neurons are all signaling, and we have a dynamic system. So please, when you study it, don't think that the whole thing's going to break down, because if it did, like I say, we'd have blood running out our nose and out our ears, and we all go home.
I want to harp on this permeability measurement.
Well, you can harp on it.
PATLAC is based on certain assumptions. And I was taught this a long time ago.
Okay.
The true measure of permeability is the reflection coefficient
Right.
of a particular molecule, like albumin, for example.
That's
so,
fine.
so I mean, I think, we need to get our definitions straight. So I want to try to switch a question that bothers me personally is that, there's this interest in the neurovascular unit, but it's been very hard to reconstitute that unit in a cell culture condition.
Well, sure.
And
you know why?
Well, tell me why.
Each cell system's contributing a different factor, and that's why you have different soups all the time, different cells all the time.
That's a cop-out.
It's not a cop-out. You got five different cell types all contributing something.
But you should be able to reconstitute it in terms of putting together the
In
different
vitro.
constituents,
Okay.
whether it be astrocytes, astrocyte condition medium,
This is great.
neural factors. So, why?
That's why 20 years ago I moved in vivo, because we found out in vitro it's all black magic.
Okay, I think we can take
That's a
2
good
more
ending.
comments and then, if it's quick, 3.
I just have a question is when you actually disrupt these cell matrix interactions like Greg just showed. Greg, he's talking. When you disrupt the interactions, the cell matrix interactions, because you change the types of integrins that are expressed. Just going back to what Bill Ayer had shown in his talk, that you may actually change the transcriptional profile of that endothelium, because now that integrin is engaging a different kind of extracellular matrix protein. And have you looked at anything to see that whether there's any transcriptional downstream effects?
Yeah, that's the
in
point of
your
the
schema?
current work, which I didn't describe, and it's a little too early days to say what happens downstream from that. We believe that the endothelial cell cultures, particularly on inserts, are at a steady state, but they're probably not. And so, truth in advertising is that although the viability of the cells is 98%, even in conditions of oxygen-glucose deprivation, there may be some component of proliferation, which changes the dynamic anyway in terms of transcriptional factors that are involved, and also adherence, integrity, I guess I would say. So we expect some change. And if one's looking at signaling events, it really does depend, as you probably well know, on when you've when you've begun the culture, particularly for primary and P2, P3 cells. We've avoided all of the long-term generated cell lines that are immortalized because we want to look at signaling. But there's an acute dynamic, and then there's a more steady state dynamic. And which one is true? And so that's a part of the problem we're dealing with right now.
Well, I was thinking more actually looking in vivo, if you could do some microdissection and take, after ischemia, take those endothelial cells and do some
No,
profiling.
we haven't done this. It'd be marvelous to do. I make a plug for non-human primate studies. The era of non-human primate studies for this work, I think, may be gone. And this would be a horrible thing, because this is a phenomenal model. From the standpoint of ischemic stroke, it is probably the best model on the planet outside of the patient, which we don't have access to material to. I've been able to maintain for about 20 years materials in freezer at minus 80 degrees, but they're going to deteriorate over time. So we could look at this question, but it won't be a complete answer. A few years ago with the Genomics Institute of Novartis, we looked at some of the material from an RNA standpoint, and it's really, really interesting stuff. And we have to do the single cell studies in order to be able to understand what's going on. I hope we can come back to that. I would hope that some group like the NIH would like to have on campus a primate facility run at a very high level to which investigators like those of us in this room could go to and do these kinds of experiments in a controlled way, and the materials would be available to everybody. That would be phenomenal. That's what we're missing.
One more or two, if it's very brief, please, brief questions, brief answers.
I don't know if this is the right venue, but no one's talked about when the cells are really permeable, that is when you have a microhemorrhage. So could someone talk about capillaries that are leaking out red blood cells or arterioles that are leaking out red blood cells?
Yeah, so Gerhard Hamann also in our group looked at the correlation, this is a correlation, between the breakdown of matrix in the capillaries and larger microvessels up to 100 microns and the development of microvascular hemorrhage or minor hemorrhage. And there's a very strong correlation. So, the basal lamina, while it's dynamic in its own way, is also a pretty good force, a good nice fence to prevent cell transit. Yeah. So that's very real.
So the chemical engineers that talk about a 3-pore model, a pore through a closed junction, and that might be regulation of ion flux, pores through broken junction, and I think a lot of what we've talked about with dye markers like dextrans or albumins are following that, and then large pores, dead cells. I mean, and these pathological processes probably include all of those steps, and sometimes I think Dr. Walika is absolutely right. We need to be maybe very careful about defining permeability because we may be looking at different parts of an ongoing process, and that may very well be a longer stage with endothelial degeneration and death, leading to very frank hemorrhaging.
Yeah, in fact, that's true. I mean, recent observations, some from us and some from Dietmar Westweber, have shown that different signaling events mediate permeability, truly measured permeability. A different phosphorylation of V-cadherin mediates permeability of albumin, let's say, whereas phosphorylation of V-cadherin at other site mediates migration of leukocytes or red blood cells. So it isn't, I think it's simplistic to say if you have permeability increase, that's going to transport proteins as well as formed elements. That's not the case. There are discrete pathways mediating each process.
Dr. Davis has a comment? No? Can I just make a very brief comment about microvascular lesions and microhemorrhages, because it's seen in many diseases like Alzheimer's or ALS and etc. There is a recent work in PNAS by graduate student Ethan Winkler, who is now a neurosurgery resident. When you increase microlesions pharmacologically with warfarin in ALS model, you can influence neurological course of disease. And other way around, if you reduce the numbers by closing the barrier, you can control the motor neuron disorders. So I think that's one of the examples how the capillary originated. These are microhemorrhages, 50 micrometer capillary originated can influence the course of neurological disorder. I think it's a nice, he did a nice work. Making you were on his committee, right?
I love animal models. I mean, they're the best thing to anything. But the sense of the discussion, there's an anti-cell culture aspect of the discussion. Well, I think that cell culture experiments are not a charade. They have meaning, and they're if you address the question properly, or if you address a particularly interesting question, then they're obviously a valid tool. Someone mentioned they're getting out of cell culture experiments doing animal models. Well, I think you should get back into cell culture and really try to address questions at a very basic mechanistic level, because animal models, although wonderful, the best things in sliced bread, I mean, have limitations also. And I think that having appropriately controlled endothelial monolayers is a wonderful way to go. And that's what bothers me, that no one has been able to constitute a blood-brain barrier. Because if you could reconstitute it, then you would understand the makeup of the barrier. Because, I mean, your studies are not telling us what the makeup is.
Yeah, I just wanted to make a comment about that, because if you take an endothelial cell from the brain, you won't get the electrical resistance. It'll always be too low.
Why?
Well, because it doesn't make a tight junction. And then if you add an astrocyte, you will bring up the electrical resistance, and it'll look more like an actual cerebral capillary. We're now putting back in pericytes. And pericytes are absolutely critical. Each one of these cells makes a different component of the tight junction. And when they break down, you see the tight junction proteins in different cells. So I think the complexity is we're getting close to being able to imitate it. I don't know if we'll ever get the true electrical resistance that Krona was able to get in his capillaries. I think it's 80 something, but it'll be closer. And you just have to have somehow each of these cell types makes a different aspect of the tight junction protein. And if you don't make the tight junction protein, you don't have the blood-brain barrier. So I don't think it's hopeless, but it's very complicated.
But also you have to incorporate flow. There's evidence that flow makes a big difference in the formation of tight junctions as well.
That's exactly right. And I don't want to leave anybody thinking that in vitro is a bad thing. You complement your in vivo with your in vitro, and you've got the full caveat. But you can't forget flow either, and the tortuosity factors that also affect endothelial cell tight junctions. So it's a very complicated issue. Yes, you need in vitro for mechanistic studies, but by God, stay as close to in vivo as you can because of the specialized nature of all these cell types, as Gary just pointed out. Thank you.
Okay. So I would also like to point out that what some of us are not talking about is the quality of the endothelial cells that we're using, whether they are cell lines or primary cells. That makes a huge difference. And also from work from my observation from work from our lab, apparently what astrocytes produce in culture is even better than using the astrocyte stem cells because we get higher transendothelial resistance if we use the supernatant from cultured primary astrocytes, and much better than we get when we use the astrocytes themselves. So obviously there is some component with that astrocytes produce, and that's tightening, resulting in a tighter barrier. So there are a lot of things to explore here. But what I do know is that some cell lines you use cannot recapitulate effectively what you would observe in primary cells.
Okay, very quick, please.
Okay, getting back to this idea that do endothelial cells in vitro lose their characteristics when the endothelial cells in vivo, when they're put in vitro, do they lose their characteristics? Do they retain some, as Belair said, or lose others? Okay, so if you compare UVEC from umbilical vein to HEDMEC or to HEPMEC from pulmonary microvasculature, you see distinctions that are retained in vitro, and if you measure, as Asser said, by electrical resistance, there's a big difference. And as far as Dr. Rosenberg said, the tight junctions are retained in the HEDMEC, and if you knock down Claudin 5, for example, you lose that barrier advantage.
Can I make a quick comment about cell culture preparations? Because in the last couple of years, there have been several groups that are starting to work with iPS cells. They're working with human cells. And Eric Schuster's group in Wisconsin has been able to put together a brain capillary endothelial cell model, right, from iPS cells that has a transendothelial electrical resistance that is pushing 3,000 to 4,000 ohm centimeters squared. Okay, and it has many of the molecular characteristics that you would expect to see in a blood-brain barrier model. So there's a new dimension to this area. I'm not a great believer in cell culture. I'm probably from the Tom Davis school, but I think this has tremendous potential because you can get cells from people. You can get cells from people with various diseases, and you could make the endothelial cells and see whether or not you get the same characteristics for transport, for electrical resistance that you see in controlled cells from controlled patients.
Actually, you can buy them. There are places that sell primary human brain endothelial cells. Yeah, we don't get them from undergrads.
Okay, I would like to thank all session 4 participants, speakers, chairs, and discussants. And I would like to invite my co-chair, Dr. Zorina Galis, for some concluding remarks. And I'll just start. In addition to Dr. Ramadan and Husman that Dr. Storylandis acknowledged this morning, it took a village to plan this workshop, and there are a few other colleagues that I would like to thank, Dr. Jim Koenig and Dr. Rod Corriveau. Rod is not here, unfortunately, but they were tremendous in this planning effort, and it was really a teamwork. And Zorina will thank her staff in Heart and Lung. Just some remarks from the NINDS perspectives. I think we considered this first day a success based on the lively discussion that we got, and that was exactly what we expected to see. And I thank you all for staying up until this time and for participating so actively in the discussion. I think we covered a lot for the first day. I think some of I can't possibly recapitulate all of the topics, or we would stay here until midnight, but some of the major topics that emerged is the importance of sex differences in the vasculature, the complexity and heterogeneity of the microvasculature, as well as the influence of both genetic and environmental factor. I think there was a lot of discussion on cellular interactions, and particularly the interaction between the microvasculature and the immune cells. Some discussion that in the late session about the unique features of the blood-brain barrier and whether it could be modeled in vitro, and it looks like this is still a topic of debate. Some other concepts that emerged is whether small vessel disease is a unique disease, or if it's just a continuum of vascular pathology and the large vessel pathology, and also the topic of repair. I think what also remains to be discussed is the chicken or the egg issue. Is small vessel pathology causing the disease, or is rather a disease process that is affecting the small blood vessels? And I think we may be hearing more about this tomorrow. And with this, I'll just leave the microphone to Zorina for her remarks. Thank you so much. And also after this, if you think there are topics that were not covered today, but you would like to discuss or mention briefly, we would welcome any additional input from the attendees. Thank you.
So I also want to thank everybody. It's very exciting to see so many people coming from various areas of research. NHLBI is covering a lot of territory in terms of vascular structures in various organs, but when it comes to disease, to the disease state, usually people migrate towards the specialized institutes. So we find that many times we get lost in translation, going from knowing the basic biology or the physiology to the specific changes in the organs. So in order to put this together, it took more than 2 institutes. Actually, it took several institutes. We started a small blood vessel group within the NIH. I would like to thank the whole group, which is too long to call out. I could call out a couple of people who are closer to we're closer to the organization of this meeting, and that was Christine, Marek Belkin, Mark Charette, and Esther Tolunay. They all work at NHLBI, but we requested recommendations from all the institutes. We actually had to lobby and show them that there is this research going on across the institutes, and many of you are recipients of awards from various institutes. So I feel, I don't know if I'm a fan of Malcolm Gladwell. In one of his books describes the best place where innovation happens is a bar in a on an island. Some of you nod their head, they know, where all the ships come from all over the world. They have to refuel and change cargo, and everybody gets into the bar and they all discuss what they learn in China or Africa or wherever they are coming from, and there is this huge information exchange. So I feel like we are like that. Later you can even reproduce the exact bar scene. Right now it's just the interaction of ideas. It's nice to see that some things didn't change much. I have to say that I didn't follow very closely the permeability issue. I see that's not changed so much. I remember my very first manuscript was rejected with one line saying caveolae do not exist. So that was it. There was no way we can prove that. Now caveolin is doing everything and caveolae are everywhere and so forth, but we're still wondering what permeability is. So I agree we have to, probably what this group can do very well is to agree on certain definitions. By the way, for the purpose of our small blood vessel group, we decided that small blood vessels are arterial, capillaries, and venules, anything that's not a large artery, so don't think only microvascular. Think probably even a little bit bigger. So I think definitions are going to be very important, and they're going to be one thing that we could all probably work on. The other thing that I think would be very nice to do is to, in the end, maybe discuss how systemic diseases affect the endothelium. Looks a little bit different, but more or less is continuous throughout our body if the models were right. So it's one big vessel in the end, and how various diseases affect us and how we could potentially learn from differences in organ presentation. Another example that I thought was very nice was the discovery of these nets. George Palade showed these bodies, the variable Palade bodies, and nobody knew what they were doing. It's been 40 years, 50 years, I don't know, but finally we see something that seems to be very relevant. So hopefully we can accelerate the process by talking to each other. I think it's going to be interesting to continue the conversation about what's the effect of using in vitro models versus in vivo models, deconstructing versus maybe splitters versus lumpers, right? So the splitters would be people who take the system and deconstruct it to the simplest units, and the lumpers are using the animals and hope to understand everything that's happening. So I think there are certain themes that will go across. I don't want to repeat the topics. We wanted to give a chance to the audience to bring up some topics that we didn't discuss, but please do not give a lecture. Just throw an idea there and see what comes back. Okay? Thank you.
So I know it's late, and I'll try to be brief, but I want to caution you from considering the endothelium throughout every organ the same.
I
consider
did not
it very
say
different.
that.
Okay.
I said that we have an endothelium that covers all
Okay.
the blood vessels. So
I think of a disease like preeclampsia, and I don't mean to keep harping on this, but when I think of preeclampsia and the manifestation that is so heterogeneous in so many different women, I think of it as the weakest link. So when you start having placental disease, you start having brain disease, you start having liver disease, to me it's the weakest link that's getting hit, and you're starting to have end organ damage. And that's when I think pregnancy is almost a stress test to these different organs. But it's the endothelium, the heterogeneity in those organs, that's actually getting affected. So I think of the endothelium as being actually very different in every organ, and it's going to be very disease specific about how it's getting hit. So Sorry if I misinterpreted what you said, but I just think we need
Sorry,
to think about it.
I actually did graduate work showing the difference in transport, even different molecules are transported in different ways. Capillaries work differently than arterial. I would never say that, but we do have an endothelium that is probably, and that was a question that I have for all of you, how does the differentiation occur? Is it in response to the environment flow, interaction with other cells? When do you form the tight junctions in the brain? What's the role? Is this immunoprotection or whatever it is? And I don't know anybody addressed that. When do you actually get that specialized features? Go ahead.
So we haven't heard very much about vessel dynamics, and especially for the vessel dynamics, right? So these blood vessels serve this purpose to actually tune the blood flow to the organs that they serve, and they tune it according to metabolic need, or to a certain extent. Actually, we have really no idea, right? We study this in the brain, and this is the basis of the signal that you measure in functional MRI. There's actually localized hyperemia when neurons are firing. And we haven't really heard anything about that today. We haven't talked too much about neurovascular coupling and how the blood vessels actually dilate in response. But one of the other things that's really important is that that dynamic tuning of the blood flow is important for the health of the tissue. And so when we've been talking about vascular dysfunction and physical problems with the vessels, everybody said, oh, it must be blood-brain barrier leakage, it must be some problem with the sort of structure of the vessel. But if that vessel is no longer functionally responding, its dynamics are no longer normal, it's sluggish or delayed or doesn't reach a threshold to do what it needs to do, you could imagine that would lead to dysfunction of the organ, death of neurons, even sort of acute performance failure by not getting the nutrients that it needs. So it's another, and we haven't really talked about things like endothelial signaling, vasodilation, vasoconstriction, and all of the signaling pathways that are involved in that. And that, we think, could be a major aspect of brain health and another link between the systemic dysfunction and the brain problems.
Thank you. We have more comments.
I was a little disappointed in the way the program had so little to do with the microcirculation in the heart, because clearly non-occlusive ischemic disease in women, and what used to be called Syndrome X or whatever, and NIH did support the WISE study, is a major health event. And we really haven't really addressed that except for maybe one talk in terms of the microvasculature in the heart. So I think that's an area that needs to be improved and really pay attention to the sex difference. The second comment in terms of cell culture and the difficulty in constructing some of these, I would just, I mean, I hate to be the sex patrol here, but, you know, pay attention to what sex these cells are coming from. And not all HUVECs are one sex. And if you're combining cells from the, you know, the capillaries and the astrocytes and the neurons, are they the same sex? Are you mixing sexes? Does it matter? And the third thing that affects that is what's in the culture media in terms of the hormones that are going to affect those cell cultures. I know for the smooth muscle cells and the endothelial cells that we do when we were looking at steroid activity, we would typically strip the serum of charcoal to get rid of the hormones that are in the fetal calf serum or whatever. I know nothing about what kind of media you use for capillaries or brain cells or astrocytes or parasites or anything like that. But pay attention to that, because if those hormones are there, they're going to have a very different effect depending upon what receptors are being regulated or expressed in the tissue the cells that you're trying to culture. Just two cautions that are so obvious, but we always forget it.
Dr. Miller, we actually have a workshop that's looking at that specific sex differences, sex bias in research, and how the applicants may or may not choose to look at both sexes when they do their experiments. But I do have a challenge for you, and I've been always intrigued by the fact that even male cells have a female mitochondria, right? Mitochondria come only from...
Yeah. That's true. I'm not making the point that you have to study both males and females, okay, in your cell culture. I'm making a point that you have to know what you are studying. and actually report it. And if
That's
you're
exactly
putting-
what I said.
Yeah,
They
and if-
don't report the two genders. They don't plan on doing it, or they don't report it, or they don't pay attention. That's what we call bias.
Okay, so let's not confuse having to do both cell types with the idea of knowing what you're studying. And I think that's a huge misconception within the scientific community in terms of what we're talking about in terms of sex bias. So thank you.
So mine is not a complaint, it's just a suggestion for the discussions maybe tomorrow, and it's the role of developmental processes. As you know, many developmental processes, either in the vasculature or in any territory, get replicated in part or in total during disease. So it would be interesting to think during the discussions that will come up in the next day, to think about the role of progenitors. I mean, we now know many of the progenitors of the vasculature, how do they fade during normal development as well as disease. For instance, you know, the role of the perivascular progenitors, how much they contribute to fibrosis, et cetera, et cetera. So just as a plug, just to keep remembering that, if we think also of building eventually a kidney or what have you, I mentioned that at the beginning today, we need to keep that in mind. And that, I think understanding these kind of processes, developmental processes, may help us harness the biology a little better.
Mas after from Yale. I'm an emergency physician, so
Thank you for coming.
I've actually learned a lot from all the basic science work today. My question really comes from clinical observations that we see a lot of patients with chest pain. The focus of today's talk has been more about thrombi and ischemia. I see patients and we're screening them before they've had their heart attack. And we find a high prevalence of microvascular dysfunction as opposed to microthrombi. So I guess it goes back to the question about vascular hemodynamics, whether that should also be studied, because these patients are coming in with symptoms which are exactly the same as angina or heart attack, except they have they don't have troponins or evidence of end organ damage. The other interesting observation is in these patients, they tend to be young. There does seem to be some systemic process, like a lot of them have either migraine or metabolic syndrome or lupus or some other form of inflammation. And so I think it goes back to that question whether we should be studying it as a systemic disease with specification to individual organs or not.
Following what Ariel Gomez said before, we heard today about angiogenesis many times, but we haven't heard the word vasculogenesis, and all the small vessels, capillaries, arterioles, venules, develop by vasculogenesis. So I think we need to go back and discuss development.
Something worth focusing on?
Now that we are just between friends, I can tell you that we had all those speakers invited, but we were not as interesting as 2 meetings in Europe in the same time on angiogenesis development and vasculogenesis. And we also had the coronary specialist invited, but he had the wedding he needed to attend. It's really very hard to put a program like this. So I suggest that you may want to make a note. You should have done this. You should just say, can we talk about this? Chances are within 300 people who are here, somebody can address the topic. That was our hope. And I was hoping that you will get an answer. I don't know if you want to have a dialogue, if somebody has an answer, please, I applaud an emergency room physician.
So one thing that I think would be worth talking about that we haven't talked about much today is the cells inside the vessels and the interrelationship between the spatial aspects of those cells and the vessels themselves. So for example, every single blood cell's too big for capillaries, right? They're all bigger. than capillaries, and they have to squeeze through. So the difference in blood flow that you get when you've got red blood cells flowing through a capillary versus a white cell that comes in is going to have differences, presumably, in oxygenation of the tissue, and then transporting that white blood cell across some adherence across the cell blood wall. That's going to block flow for a while that actually gets moved. So all this kind of stuff is going to play a role that we haven't really talked about today, and I haven't seen much in the literature talked about either. And it would be worth looking at, because it may be that there's some problems going on with the way that the cells stick to the vessel walls themselves.
I'd like to interrupt. If you had an answer, maybe
Microphone. Yeah.
this could be a dialogue. You don't only have to ask questions. You don't only have to ask questions. You can give answers also.
See a lot of patients who have a systemic problem and then they go into a confusional state, delirium, that sort of thing. And I think if you look carefully, you'd probably find cytokines would be the link between what's going on in one organ and what turns on the damage in another organ. But then it's also age. So if you have a vessel that is not as functional as it should be, and it's exposed to an abnormal cytokine, and TNF, IL-16, those kinds, it becomes dysfunctional for a period of time. And then, of course, it's not structural, so it can go back and we see this a lot in, like, an MS patient gets an infection, they come in with an MS attack. So we definitely have to think about how one organ is influencing the other. I think that's a very good point.
So I would like to follow up on that. I have to say this was a feast today.
Introduce yourself, please.
I'm Connie Ryan from Stanford. My primary interest actually lies in inflammatory blood vessel disease, the vasculitides, and the autoimmune diseases. And I have to say, from our point of view, almost every chronic inflammatory disease is complicated by what we now call acceleration of cardiovascular disease. To be honest, we have no idea how that happens. We can only speculate how that happens. But what we do know is that the phenomenon has two major characteristics. First of all, the aging process is centrally involved. And of course, one can come from the side of the blood vessel and say, oh, the blood vessel is age dependent. But the inflammatory system is equally age dependent. Nothing changes as dramatically as we go through life as our immune system does. And an old immune system, unfortunately, does not only become less effective in protecting us, it becomes much more aggressive in being inflammatory. So we have a combination of an aging blood vessel with an aging immune system. And I think that makes our patients susceptible to what we call acceleration. I think it goes beyond soluble mediators and cytokines. It's probably very, very critical which cells are there and how our immune system is regulated in terms of protective and non-protective immune responses. And the other phenomenon, what we have is this really strict territorial behavior of these inflammatory vascular diseases. I mean, how these diseases know exactly where to go is absolutely fascinating. But we use it clinically every day to make a diagnosis. So there must be something that tells the disease where to go. So if the vascular biologist could help us to understand that, that would be very cool.
So just a quick comment. Just so you both know, she's talking about very young patients, 30s, 40s, not older patients.
Right. Yeah, that's where we're diagnosing. Otherwise, they have regional defects and they have clear coronary artery disease. So the symptomatic patients that at least we're finding are, at 30, 40, 50-year-olds. So they're young. So aging doesn't necessarily makes sense completely, but the vasculitis seems to have some connection, like, you know, they have MS or they have lupus or something else.
So I was going to bring that.
Yeah, why don't you go ahead?
Actually, this penetrates all through the patient cohorts. If you go into patients that have rheumatoid arthritis, their acceleration is by about 10 years. They get their cardiovascular disease about 10 years earlier. And they can't recover as well if they have an injury. It's actually the rare patient that has true vasculitis in the setting of the connective tissue diseases. These are the patients that have vasculitis, and they have a primary vasculitis, but secondary vasculitides are not that frequent. What bothers us clinically is the fact that patients that would normally have their disease at the age of 75 now have it at the age of 60. And I think it's acceleration during the second half of life that happens.
I think there's some also, as I also agree, I don't know, I mean, I'm sure age has a big effect, but at least in autoimmune diseases like lupus, where you have a 50-fold increase in the incidence of atherosclerosis, and these are women between 35 and 40 years old, there is something fundamentally about the inflammatory response in the glomerulus, in the kidneys, or wherever the lupus hits, that really increase, and it's not very well studied and not really known, and I think that's some of the gaps in knowledge that we need to fill.
So I wanted to, I got in line when they were talking about the sex differences, and I wanted to push back on a couple of points that have been made. First off, on the sex differences, the idea that we need to know what the sex of the cells are in our culture, I don't think is warranted. I think it's ridiculous, because I put it out there. I want somebody to tell me what, the Y chromosome has very few genes on it. Those genes are not being expressed in culture and endothelium. I want to know of an example. Certainly there are sex differences in vascular diseases. That's no question about it. But the idea that male endothelial cells and female endothelial cells in culture are going to behave differently, I'd like to hear an example of somebody who's seen that, because I've not seen that. So, you know, I'm going to start, you know, we're going to start saying we have astrocytes from men and the rest of them.
In the body, where they are
Obviously,
baited by
in the body,
hormones and other things.
where there's endocrine effects, you know, there's no question there's sex-related difference in vascular, but the idea that in culture we now need to think about whether or not the astrocytes or the endothelial cells we're looking at are coming from men or women, it's imaginary. So, and the other concept that's come up is that idea that we think endothelium's all the same throughout the body, and we've got to keep that in our mind. Nobody thinks that endothelium's the same throughout the body. I mean, it's obvious, and even in the eye, we've got, within a millimeter, the retinal vessels are completely different from the choroidal vessels on one side of the retina and the other side of the retina. So the retinal vessels have vascular tone, choroidal vessels don't have vascular tone, the retinal vessels have a barrier, the choroidal vessels are permeable. There's no question that there's a tremendous heterogeneity of endothelium throughout the body, the glomerulus. So, you know, just, I think that there's some like a misconceptions that we're not considering, you know, we have to consider sex when we don't consider sex, or that endothelium is variable and we haven't considered that. That's, you know, part and parcel of vascular biology. I think everybody here understands that.
If I can make a brief comment, I was going to get to it tomorrow session, but because of the discussion of inflammation and how aging is the definite factor and everything gets worse, you know, autoimmunity and inflammation, and this is the maybe a U-curve that exists, and this is something that I want to bring up to this community because I study neonatal and childhood stroke and experimental models that we developed, and the statistics is very staggering for childhood stroke, that oftentimes within the 6 months after chickenpox, or after, you know, different infection, it may be viral, it may be bacterial infection, the stroke hits, and it hits mostly the boys, not the girls. It's 1 to 6 approximately, you know, girls to boys, and you know, this inflammatory component, it does present, and it's very profound, and vasculature is the one which suffers the most, because these arteriopathies that several grants P50 mechanism study now that show that vasculature is the one where all these processes begin, and then they just it leads to childhood stroke, to repetitive strokes actually in children. So, and I don't know how many of you know that neonatal stroke is as frequent as it is in adult population, in elderly population, and inflammatory mechanisms are clearly present there too. So that is why we should not, I think, discard the fact that immaturity is also very make the brain and other organs, the blood and leukocyte component is very, very profound, but in different ways than in adults. It does happen. So we need to discuss it. That is why I very much appreciate the comments that say we need to look at what happens with development. Maybe it's a topic for a different workshop. It's impossible to cover everything. It's just too rich for soup for all individual topics. But I thought it's important to bring to attention.
I can't let that comment go.
We knew it.
Okay. Let's just say that I think there is a lack of evidence right now as to what is different and what is not. And I think that gap has to be filled. There may be many things within a cell that are the same between males and females cells, but do we know what those are? If you look at what's on the X chromosome, there might not be many genes on the Y chromosome, but hello, one of them regulates norepinephrine development, and there's some also that be macrophage adhesion, and there are a lot of genes on the X chromosome that affect diseases, including some cancer, especially to melanoma, to mitochondrial function, and the fact that you only have one in a male cell and you have two in a female cell, some of those genes are activated, some of them are not. I was talking to somebody before, there was a difference in response to hypoxia. Just because we don't have the evidence doesn't mean that there's not a difference there. We need to be able, we're scientists for heaven's sake, we have to be able to understand the variables that we work with. I don't think it's too much to ask somebody to report what the sex of the material is that they're investigating. How can somebody else down the street reproduce your experiment if that data are not in your paper, for heaven's sake? We used to be able to report sex, age, diet, light cycle, and all this other stuff back in the day when I was a graduate student, and I'm sure that was 40 years ago. But what happened? The methods got put into the supplemental material, we cut it down because of impact factor, and now nobody can reproduce anybody else's experiment. Okay? I mean, we need to be able to do this. And what's fundamental about scientists, we want precision on this and precision on that, and taking your equipment down to a certain level to measure some, just report the sex of the material that you use. If there's no difference, great, let us know. If there is a difference, great, let us know and move forward.
And I would also say that, to stand here and say, don't tell me that there's heterogeneity because it's so obvious to me, is dangerous. I think we've been working, many of us in this room for years to try to teach the vascular biology community, let alone the non-vascular biology community, that not all endothelial cells are alike. And the problem with the glut of literature right now is that we take endothelial cells in culture and we extrapolate to every vascular bed in the body. So, I think from the standpoint of the comment that was made about, well, of course there's heterogeneity. I think, that's Monday morning quarterbacking, and I think we've got to be really careful to, before we just say, don't tell me what's obvious, because I think it's been a huge limitation in the field. So.
Thank you very much. I think we'll stop here and we'll continue tomorrow. For people who signed up, we have dinner at 7 at Positano, which is a nice Italian restaurant, and we hope to see you there. Bye-bye. Thanks.